So. Yes, they go back.
Okay, testing the mic. Can you hear me in the back? Is that, yeah, in the back? Okay.
So before we start, just an announcement for people that are watching online. So tomorrow it won't be streamed through YouTube. We will have parallel sessions, and there are Zoom links that were added to the program. So people that are interested in the different parallel sessions that we're having tomorrow, please go to the Zoom link that is now shared in the program. So thank you all for being here again this morning. I hope you had a relaxing evening yesterday and you're ready for a full day of conference. So we will start today with a keynote address by Pierluigi Buttigieg. Okay.
Close enough. That's good. That's good.
I tried. And so Pierluigi comes from the Alfred Wegener Institute for the Center for Polar and Marine Research. He's a senior data scientist, a digital architect, and strategist. He focuses on ocean and biodiversity data and societal links. He contributes as well for the UN Decade of Ocean Science for Sustainable Development, and is the chair of the Ocean Data and Information System project of the Intergovernmental Oceanographic Commission of UNESCO. Thank you, Pierluigi, for accepting our invitation, and we're looking forward to hear your talk.
All right, thank you very much. Yeah, so my world is a world of acronyms, and you'll see many acronyms as we go through this. Don't worry too much. The purpose of this keynote is to perhaps give you some perspective on things that have been forming and being created to help biodiversity data in general, but also omics data specifically, permeate some of these global data systems that are being used by nations, by operational networks, by the weather systems, by oceanographers. These systems exist already at different stages of maturity, and they don't always talk to each other. But first, I want to get a sense of the people in this room. So everyone here has submitted to INSDC, right? So ENA, GenBank, DDBJ, yes? Yeah, okay. How many of you have submitted, by show of hands, to GBIF? Any data to the GBIF? One, interesting, two. Oh boy. Okay, how many of you have submitted to ODIS? Sorry, OBIS, the Ocean Biodiversity System. Okay, one. Right, and then, because from the talks yesterday, it's really fascinating that you have, of course, the reference genomes, but there's so much more you have, your later analysis, habitat mapping, impact assessments, vulnerability profiles, trait profiles. And let's see what we can do about that. So the purpose of this keynote is to just provoke perhaps the next round of grants, or the people in this room who have grants or missions right now, to think about joining some of these efforts to make sure that reference genomes can be seen by some of the emerging data systems in the world. So let's just jump in. My time is funded by various European Commission projects. Marco Bolo, don't judge us too much on the name. The other grant that won is called Obama Next, so we're not so bad. Marco Bolo, BioOcean 5D, and World Fair, which closed, when World Fair in particular was trying to connect data from biodiversity, oceanography, cultures, nanoscience, material science, space physics, to try to make sure that these systems talk to each other. All of this talk is going to be focusing on digital ecosystems. Who's heard of this, a digital ecosystem? Yeah, I mean, often we hear about it in a kind of passing way. It's often used as a buzzword, oh, our digital ecosystem. But it's actually quite a serious term in data engineering and data architecture. So how many of you have Samsung phones? One person? Oh, Rob can't be the only one with a Samsung phone. Somebody has to. Who has an iPhone? Or no, okay. All right, a few. And an other, who's an other? Oh, wow, look. Yeah, revolutionaries here, good. Fighting the system. But as soon as you buy, and I pick out Samsung and Apple in particular, because you know what happens. When you buy their phone, they really try to get you to stick inside their digital ecosystem, use their products, their password safes, their et cetera, because they control it. Now, this is happening at the scale of the web too. Google, Microsoft, and others, they try to control chunks of the web, Amazon. We see what happens when these things fail. So those are all different digital ecosystems. They talk to each other sometimes, just like you have ecosystems next to each other, a mountain or riverine zone touching up on a grassland. And it's very similar. They have producers of data, consumers of data, things that reprocess and degrade data. Okay. So we talk about this very, very seriously when we do, and we try to map digital ecosystems. And importantly, they are socio-technical systems, right? The technology is one part, but there's also a whole social machinery behind that, the organizations, their processes, their management forms, their policies, their regulations, their legal frameworks. All of that then contributes to stabilizing a digital ecosystem. And so we're going to talk a little bit about some digital ecosystems that are proximal to what you do and how we can perhaps create one that's stronger for reference genomes. One exists already. You have things in ENA, but a lot, from what I gather from discussions had with some of you, a lot of the assets that you've been generating, the knowledge products, the data sets, they're trapped in some journal somewhere, or they're submitted on an institutional archive. This doesn't connect very easily, and it actually costs a lot of money to get out. So I'll put my conclusions first. I've been dealing with policymakers for the past years. Always put your conclusions first to get it, and then we'll see if you believe me at the end of it, right? So reference genomes, from what I'm seeing, they are in a sense, a keystone species in a digital ecosystem. They are a stable point, a deep dive into an organism that really, really characterizes its ability. That anchor can then be linked out to all of that supplementary data, downstream data, process data, the workflows you use to understand it, to annotate it, to understand its role in ecosystems. That I don't think exists very well, and that's a shame. Because when a machine finds your reference genome in ENA or an INSDC resource, there should be a little cloud around it of all of this knowledge that can then be put right in the front of somebody building an interface. Right now, you have to either email or look, correct me if I'm wrong, but you have to email or look or know a site to go to. And that knowledge is really niche. That doesn't scale in time, right? So that's what we're going to try to see what we can do. We've created some resources and we've created some frameworks and organizations over the years, which I'll talk about a bit later, which I think can help us do this. And I see a lot of skill and talent and interest in this room to make that happen. A blast from the past, when I was finishing my PhD, I did it in bioinformatics, microbial genomics, right, and metagenomics. So I'm not so far away from you expertise-wise. Part of my outlook, this is back in 2012 when the Earth was still cooling and all of that. Part of the outlook was like, I tried to analyze, at that time, 5,000 microbial genomes. It was a big deal back then, not so much anymore. But all of this archive data, I had to put it into a central database, clean it up. It took years to do that. And I only got to do the fun stuff much, much later. So the vision was, wouldn't it be nice if people generating these genomes and metagenomes could have their organizational databases, but then package that data with interoperable metadata and standards using the same standards to be able to create on the fly a virtual observatory that just watches everyone's data as it comes in and does analysis automatically and then feeds off to the big infrastructures that we all talked about, OBIS, GBIF, also the CBD, Living Planet Index, all of those consumers, those downstream consumers, which even now they don't have very strong data flows supplying them. That was the idea. And so that's what I've been doing for the past decade or so. More white hair, more, less verve in life, all of that. But we did something that I hope can be useful here. So part one, let's talk about the socio part. I started working early on with the Genomics Standards Consortium. If you've submitted to ENA, you probably saw some metadata fields recommended from the GSC, like MixS, those fields. So we developed a lot of that in the early times. I was my office mate in the PhD, she was leading the MixS, new idea. And we built a bunch of stuff to help stabilize what at that time was just essentially a dumping ground of data, where just genomes went as you didn't know where it came from or what it was about. And data with poor metadata is largely useless. Sometimes it's worse than useless, sometimes it's damaging. So we did a bunch of work in the Genomics Standards Consortium. Just to note, there hasn't been any extension for reference genomes that I know of. Does anyone know of one here? Has anyone extended the standard space for reference genome? You did?
Yeah, we have it.
In Mix?
No, it's just the Ergo manifest.
Ergo manifest. So this is like that socio key. Standard specifications and things, that's technology. It's not necessarily digitized yet. If we can get that into Mix, then it gets proliferated out throughout the standard space. And if any of you are working on ways and you think, hey, this would be a good way to standardize my genome's information, talk to the standards organizations. They can help you implement it and change things, as we'll see that changes in the technical part. Anyhow, One of the things we were working on was the Genomic Observatories Network. And these were observatories using genomic technologies. At that time, it was quite new. And they said, hey, we should actually partner with GeoBon. Who's heard of GeoBon in this room? Good, yeah. So CBD, they do a lot of work with CBD. It's the Group on Earth Observations Biodiversity Observing Networks, right? And they have a bunch of sub-networks that are inside GeoBon, usually regional, like for Europe or for parts of Africa, etc. Sometimes thematic, like Marine Bond or MBON, yeah? So we said, okay, if it's an observatory, we should partner with GeoBon. So we brought those two organizations together. And there was an all-hands meeting back in 2012. This is work led by Neil Davies, who's the director of the GUMP station out in Moorea in Tahiti, University of Berkeley. And a call was put out. We published a call, said this is aspirational to do this. I was at this time just a PhD student finishing up. I was peripherally involved, but I'm saying, hey, there's something here. There's something interesting going on. So the call was published. GeoBon then identified it as an actual deliverable. And later on, it was founded and a founding charter was created. The interesting thing about this is that it brought together the scientific community with the observatory network community. It's a very different way of thinking, much more operational. They need those results now. They need solid data flows with known standards. Otherwise, they can't operate. When those two kinds of thinking met, something special happened in the way that we approached building larger long-term systems. So it's not just project to project. Every project can feed into something bigger that eventually becomes infrastructure. The same thing was happening over in the marine realm. I was involved in a project called Atlantos to build an observing system for the Atlantic and tried to get all of these loose observing activities that had been, particularly in science, sometimes in operations, but try to get them together into an integrated Atlantic observing system. So we did, and inside that, there was a work package that was about microbial genomics in particular. So we then set up a little consortium called Glomicon, a Global Omics Observatory Network, to sort of work with the Genomic Observatory Network. That was different because omics also included metagenomics, transcriptomics, etc. So it was a bit more broadly scoped. Of course, we said, well, it's silly to have two, because if you keep doing this, you're just creating more competition and reinventing the wheel. So over the years, then we merged them and we approached GeoBon to create a new bond inside GeoBon called OmicBon, right? This is a thematic bond. So it's about omics rather than a region or a particular nation's bond. So it's a little bit different, but GeoBon was very supportive, and then we created this. I say this in like a few sentences, but this took some years of negotiations, of design, of all of that. And again, the idea is to give the international community something stable that if they want to start building regional or national observing networks for omics, they have an overarching framework that helps them talk to the right people and not reinvent the wheel. So the charter of that was published in 2023. It's out there, OmicBon, so it describes it a little bit. And one of the figures is a summary of, again, where we placed it and why we needed it. Because we had things like GeoBon. We have like the essential variables, the EBBs from GeoBon that go to policy, the EOVs that go to the Global Ocean Observing System, those essential variables, including biological and ecological ones. We have things like the UN Ocean Decade, observatories like NEON in the US, this continental scale observatory network. Infrastructures are popping up like GBIF and OBIS that have been operating for a while, but also national ones are created, and also private sector is using genomic technologies more and more for their operations, and those are sometimes closed systems. So OmicBon was placed inside this to be cross-cutting, to actually talk to the other bonds like soil bond, freshwater, or any of the national or regional bonds, and really have a focused conversation about omics, genomics, etc., and connect people with the right standards organizations at the right time so they don't build a new one. And then again, we have N+1 standards. Simultaneously in the ocean, we were also trying to clean up a little bit of the datascape of the ocean. We had an intersessional working group as part of IOC UNESCO's activity, and we proposed an information strategy for the UN Ocean Decade. This is before it started. I'm saying this because these worlds were converging, right? Because of course in the ocean, biodiversity is a major concern. It's one of the highlights in the Ocean Decade too. But this sort of document, there was rounds and rounds of consultation with all of the UN ocean agencies, with private sector, governmental organizations, science organizations, etc. And we basically had some key recommendations. It's like, look, everyone's going to be a bit territorial. If you have your data system, if your institution has their data system, they rarely want to change it. They're not going to use somebody else's. And sometimes they're not even allowed to because of local laws or regulations, etc. So fine, let's do that. But let's make sure that you're able to project your data and metadata out in globally interoperable formats, following some kind of open standards on the web, rather than something that's in a paper somewhere, and project that data and metadata out so that they can be consumed by the right organizations, enriched, and a whole provenance chain developed. Okay, sounds reasonable, but sometimes the shift is very difficult, especially in science, because the reward of science is still not data, it's papers, published papers. This is something that in the decade and other consortia, we're trying to sort of shift a little bit to make data a first-class object, because it will be more relevant for longer than your paper ever will be. That's not to say you're writing bad papers. It's just how it is, right? So then that's sort of folded into the implementation plan of the UN Ocean Decade that has a whole section on data and data. We published a data and information strategy, and we're also going to publish a detailed implementation plan of that. We talked to the Royal Society, the National Academies of Sciences. We had a UK-US meeting to try to advise their scientific bodies and their funders on how to treat data in the future. So that's coming in the way that they're going to program their grants. And we talked to others, including indigenous groups, about how to make sure that indigenous interests and the care principles are complied to. I raise that because I also see in IRGA, you have the traditional knowledge and labels, etc. We did some work with them coming out of Living Data, where Rob was. We are helping them plug those labels into these global systems too. Inside the UN Ocean Decade, we launched another program called OBON, the Ocean Biomolecular Observing Network. Basically, it's like the marine-focused part of OMICBON. This is deliberate because we said, look, we need these infrastructures, these social infrastructures, all to be sort of talking about the same thing. And there need to be different sociological and organizational mechanisms to direct them to the same conversation to stabilize the technical side. That's why I bring it up here, not just for more acronyms. But the objectives and the strategy were published. And as you can see, I won't go through them. It's the same idea. We need these different distributed data systems that have their own territories, so to speak, be able to share information with each other, project it, and consume it to create a supply chain, essentially. Right, so the networks have been set up, right? So we have OMICBON and OBON, and we have a few others that are out there that are stabilizing this digital ecosystem for omics in general, right? But reference genomes and libraries, they've been mentioned many times in all of our steering group meetings and our committee meetings. Everybody said we need more reference libraries, reference genomes. People know about IRGA, people know about these efforts, but we don't know where to look to actually get the enriched data space around it. Yes, we can get the genome, but you all, as you've presented, you do so much more that should be right in front of people when they see it. And that brings us to the second part. Now that we have that social sort of underpinning, you have the right sort of actors willing to talk to each other for longer than a coffee break at a conference, we can start building some technology around that. And this is our philosophy, because of course we can build the technology right away. But technology, what is technology? It's the ability to do work faster. If people are doing random work, you can have the best technology, you're not going to get to a better place, right? So that's what we did. So it also comes like the executive secretary, the former executive secretary of IOC once asked me, oh, we know you're doing all this stuff with the networks. Can you write a state of ocean data report? And I said, no, I can't do that, Vladimir. Vladimir is not a man you usually say no to, but I had to say no. And I said, we can't do it because I can write what I know from my networks and my experience, but it will be biased. It will be very, say, European and US centric. So we need something else to actually tell us what the landscape is. We don't guess, we don't write another review. And then this is in this whole context. We know what the world is like. Multilateralism is sort of failing. This also impinges on our digital space. Countries, the US, China, others, they have their own sort of internets that are being built. The topology of the internet looks a lot different than it did in the 1990s, let's say. Lots more desire for digital sovereignty. And for us in genomes, that goes to like Nagoya Protocol, digital sequence information. All of those regulations are going to start to splinter out our data systems too. So I had to think about how do you cope with that? And Terry McConnell said it really well. We had a Digital Twins of the Ocean, a G7 meeting. And I like this metaphor because everyone wants this, right? Everyone wants to build the sparkly interface, the fountain, the thing that looks great. Not many people want to do the plumbing, right? But the plumbing is the sort of core of this thing. The plumbing is what we need to do to make things more sustainable. And it costs us a lot of money. How much time? What's our time?
About 5 to, we started late, so 5 minutes.
I can do it in 5 minutes. They won't like it, but I can. All right. All right. So it costs us a lot of money, right, to do this because we always have great things. We have great deliverables, and then 2 years later, they don't work anymore, right? We can't do that. We need to build the plumbing because it's costing us millions and billions of dollars to do this. So we built something called OTIS, the Ocean Data Information System, on these principles to network those systems. Same idea, right? The mission was to create discovery and interoperability, so I don't even have to know what I'm looking for. If it's relevant, it will be linked to somebody else's data set, and I will see this cloud of data that's meaningful to me without taking away governance from any of the things connected to OTIS. We started in regions that had lower capacity, lower development, because again, from our member state funding, we said it has to work for the whole world. It can't just work for the rich nations. And so we started there, but then expanded it out, and then created a network of systems that are sharing data and the 5-star linked open data model, the original vision of the web, how the web was supposed to work. So your data is not trapped something in like a PDF or in a proprietary format like Excel. You try to get it into open formats, then you try to get it into, how many people here have used RDF or know what RDF is? Yeah. The next generation of scientists, this is important. This is how you break and melt apart data sets easily, and how when you ask your phone, like for LLMs and things, get me this, that's how they're able to melt things and bring it up. Science, especially biodiversity science, we need to catch up on this. We're putting together some university courses to help modernize that a bit. So this is the stuff that runs the web. So when you search for manta ray or something, you know this little information that pops up on the side of your web browser? This is structured linked data. And it's linked because it can also tell you that you can find this on NCBI, you can find this in GBIF, you can see this in iNaturalist. We could have a reference genome tab there pretty easily. All the technology is there, but we don't have the agreement about how to do that. Omic Bond and things can help you do this. Because we did that, we can monitor our whole network. We can see on our 59 nodes, some of those nodes are continental scale, some of them are just one person in a laptop. We can see who's actually sharing data from the Pacific Data Hub, Western Indian Ocean, what they're sharing, if all of their nodes are live, if there are problems. So we verify that fair is actually fair and not just an aspiration. So essentially we're structuring, this is the internet. Say hi. Oh, that's the internet, say hi. We're structuring the ocean part of the internet a little bit more and overlapping that with other efforts like GBIF and OBIS to structure biodiversity, like INSDC to structure omics, and try to make those networks talk to each other more effectively. So we can do this. This is the actual topology of OTIS. It's not a guess anymore. This is the digital ecosystem. This is OBIS. They're doing very well, right? The biodiversity people. So they're not questions anymore that are abstract thoughts. Now we look at it. We see who's producing, who's consuming, who's behaving the right way or perhaps not the right way. And now I can answer Vladimir's question, but he retired. What can I do? Right. So again, the data network around OTIS is getting denser. When the UK, this is the UK. You can't see it. Yeah, but when they joined with their node called MEDIN, and that federates data from 600 or more organizations in the UK. So when they joined, 600 organizations of data, their data streamed into OTIS, and now people can see it. They can see cultural assets, omics, fish trawls, fisheries data, socioeconomic data, and they can put together this thing. This is why I ask, all the data that you presented, if we have that web and we create a sub interface for reference genomes and relevant information, it's technically very easy to do. We just need an agreement in a European scale or globally to build that. And then the data, then it doesn't matter if like the slideshow doesn't show the UK, the data shows it for you, right? Yeah, and so we're updating the services that we can build on top of that to help people find data density, find the kinds of data that they want, and do science faster. We had a meeting in Ghana, GeoBlue Planet over there, and one of the researchers, I remember what she said, I've been looking for this data set for 2 years and I found it in 20 seconds. That's what we would like for all communities in science and sustainable development. It's connecting, it's growing. As I mentioned, ENA, we're working with Peter Woolard and Guy Cochrane to link ENA into OTIS. But when I say OTIS doesn't use any special thing. It's just the web, right? It's linked data on the web. So as soon as they link to this OTIS thing, they link to all of the other networks too. That's just a thing. But if you have projects in there, they will now be visible in OTIS, right? I hope we can do better than that. If again, we have improved standards for reference genomes, we can build better services like portals and all of that stuff on top of it using the same plumbing, right? Rather than having to reinvent the plumbing every time, then portal development is faster. Yeah. We're also linking it. So we're saying, look, it's not enough just the ocean. We're working with things like the WIS2, the World Meteorological Organization, their information system They have their own little fountain plumbing thing, but we're building the link at that level so that anyone who's shared information via Otis or so automatically shares it via WIS2. There's no need to create new layers of this. So I think you see it. It's the socio-technical vision all coming together. This is short, don't worry, Clau. It's okay. I see you. I see the stress on your face. It'll go. So the vision with the digital ecosystem, we have this now. We have the social, we have the technical components, but what we don't have is you. We don't have your community to say, hey, let's create a reference genome working group. Let's use all of those links to talk to the right standards organization, get those standards out of, let's say, organizations at a national or regional scale, make it global, and then build the technology on top of it. So when somebody searches for something in OBIS, they can then traverse that network and find a reference genome data hub with all of the, yes, the genomes, but also the annotations, trait functionals, vulnerabilities. We saw excellent presentations on that, economic potentials, regulations, all of the cool stuff you're generating does not deserve to be buried in the supplementary information of an article. It needs to be alive in this data system. So when somebody clicks on that, they see this web of information around the keystone species. We're putting that into data fabrics. There's a lot of work doing now with new digital architectures where this kind of metadata flow through the web will help people build products, layers, digital twins. But again, we don't want all of these people reinventing the data sources. This should be coming from an arbitrary array of data sources. But if we're using linked open data on the web with open standards, all of these things can push and pull without having to write another multi-million euro grant and spending some poor postdocs, best years of their life just trawling through data. This is not acceptable. That's it. Conclusions are the same. Thank you for your attention.
We have time for some questions, even though we're running late, but not because of you, because of the.
I did that in 24. I did it in 24 minutes, so I'm okay.
Exactly.
Yeah, so
Perfect.
All right.
Questions, Camille?
First of all, thank you very much for being here. I think we have a lot to talk. I was wondering, in the omic bond, it seems you have subgroups divided by ecosystems. You have terrestrial, oceanic.
Yeah. Those are other bonds. So in GeoBond, yeah.
Bonds.
And so the idea of omic bond, because there was a omic working group in the freshwater bond, there was an omic working group in the soil bond, and we said, please don't do this, because then you're going to come to different conclusions.
Okay.
So omic bond cuts across them and says, if you want to have an omic conversation, come to us.
Okay. So how is omic bond subdivided? So how are there technical groups?
Yeah.
Or?
Yeah, they're technical. I mean, again, it's a small group at this time. We spent our effort to build it, to convince GeoBond that they need it, but now we're all exhausted and we don't have anything in us anymore. So we're looking for people to come and create a working group. There's lots of flexibility with what you want to do. And I sort of like the idea of an early action being the reference genomes, because that's like the backbone of omics in general. So this is really an invitation. Please use these channels that we've built, take over, and run this, and we can provide advice and guidance to build that plumbing.
And do you think reference genomes, because I mean, many say, and when we talk about biodiversity, of course, about the reference genomes will be the anchor for those, but the genomic information at population level, for example, will be the real meat for biodiversity monitoring.
But that's why, again, if we're in the same technical architecture as OBIS and GBIF and others who have the DNA extensions now for eDNA, right? Then that information is much more closely together in this network, right? So that when they find this information, when somebody spots it, when they do an eDNA analysis and they get a fish scale that has a bit of this, the reference genome can advise it, help with recruiting, mapping, assembly, etc. And we just basically can create a whole tooling ecosystem around it, right? So yes, agreed. There are people who do that. We do talk to them, like the major ones, GBIF, OBIS, and others. And also nationally, some are like the Australians, they're also talking with us about this too. It's technically, again, not hard to do this. It's just shifting the consciousness of how we work, especially in science, because we tend not to think at the scale.
So I will start domination here. Just to say, the EBI is building a biodiversity portal for reference genomes. Maybe we can talk about this.
Yeah, absolutely. No, that sounds good.
So we have a question online. So are you collaborating with the Darwin Core maintenance group?
With TADWIG, yes. Also the Darwin Core. I mentioned MIX here, but also we work closely with TADWIG. We actually did an activity to map the MIX standards to the Darwin Core ones so that we're bringing those standards communities closer together. So in general, yes, we do work with Tadwig. On the Omicron founding charter, you'll see a lot of names from Tadwig and from the Darwin Core world. We do talk to them. One of the things is that Mix started as a group of scientists that just desperately needed to do something about the mess of metadata in NCBI. It was a mistake not early on to synchronize activities with Tadwig, but they felt as if they were a different community. Now the realization has come that they should be really far more overlapped, and that's happening now. Again, we're trying to simplify the space rather than create more working groups. So yes, we are working with them, like to work with them a bit more, but that's where we are.
You have any simple question, quick one?
I think open data in genomics was made successful by journals and funding bodies insisting on this data being made public. What are you going to do to bring people to start submitting this quite complex data? Because what you're suggesting is not trivial, actually.
Well, it's not trivial, but it's not as hard as you think it is, probably. Even putting those things on Zenodo, giving them a DOI, right, and linking them in already gets us a lot farther than you think it could, especially if you mention your accession number of your reference genome in NCBI, then the graph links it for you. But let's get ahead of ourselves. So that was an important step when journals did that. The game has changed since that time. It's not just a question of like submitting, because we're not dealing with one kind of data anymore. You don't have the same submission targets. For the genome, yes, ENA. For others, it's all over the place. So our recommendations in OBON or so, it's, well, make sure that whoever you're submitting to is pushing out at least their metadata in this linked open data way, so all of the other systems can detect it exists. That's the first step. So if you know that, okay, we rely on these data services in our country or our region, there's a conversation to be had with us at Omicron or others to help them join so that you don't have to do any additional work. You continue business as usual, and they will federate your data on, right? Again, we had the meeting in Australia where we said, oh, look, here's the Australian omics data. They said, wait, how did my data get there? It's like, well, your continental system linked to ours, so you didn't have to do anything more. The journals worked for that time, but it's not working anymore. After this meeting, well, at the end of November, we have a European Commission meeting for European research infrastructures, again, to try to change that attitude towards this and also change scientific publishing in the future. It'll take a while, but your students probably will come to a very different world where their data artifacts will be rewarded in similar ways. There are competitions now to create a data impact factor, as we have for academic journals. It's not easy, but the fact is there's an urgency seen and recognized that we need to incentivize scientists in their career to share high quality data. You should be able to build a career of just producing high quality scientific data, maybe with a few papers, but that's why we can't. Time's up, so I know.
That's fine. Thank you very much, Luigi. I think we now all understand why ERG is being also so picky on metadata associated with reference genomes and samples.
In
This is really important.
in the Helmholtz world, what do we say? Metadata is a love note to the future, and it's true. All right.
Okay, thank you. Thank you very much.
That would be the link.
Okay,
Even better. So we can
let's move on with the case study session. What's the first? So yeah, first case study presentation from Virat from Hungary. So let's get ready to see some cute stuff. Well,
So again, thank you. I will be presenting our case study with the European or common hamster. A little bit about the bigger background. So the common hamster was once a historically abundant species with widespread distribution, but some.
It is on. Can you hear well in the back?
Is it working? Can you hear?
Try to speak a little bit louder, maybe.
Okay, so the common hamster was once a very abundant species with widespread distribution, but some decline were observed throughout Europe since the second half of the 20th century. And since then, approximately 75% decrease in range was observed, mainly in Western Europe, where this decline was mostly very severe. The reasons behind this decline are very complicated and complex, but most of them are connected with each other. But I would like to say that probably the most significant one is the change in the agricultural practices, because it's affecting both the connectivity of the habitat, the cover for protection for the hamsters. Also, the nutritional values of monocultures are not sufficient, that are both affecting the mortality of hamsters. So overall, this disruption of the connectivity of habitats and increased mortality are observable in the reduction of genetic variability in the species. So in Hungary, the situation is not much better. The idea of an agricultural pest is still persisting, and there is not much research done on this species. From historical observation, we've seen that there was fluctuating population cycles and 10 years intervals of outbreaks and collapse. And recent surveys have revealed that these outbreaks are becoming less intense in the recent decades. So we can say that this global decrease has reached also the area of Hungary, which was still considered in the last decade very stronghold of the species. And at the same time, we observed a shift towards settlements since the 2010s, and it provided an interesting research question whether what is the reason behind this shift toward the settlements and urbanization for the species. So we pinned the goal to characterize the genetic structure of the common hamster and assess genome-wide variation across populations, and to investigate genetic patterns of urbanization. Mostly the questions we were looking at is what is the genetic structure of the city-dwelling hamster populations, and whether urbanization reduced the genetic variation, and also whether this process have left its mark on the genome. So overall, our hypothesis was that urbanization is accompanied by adaptive changes in the genome, which must be taken into account when planning the conservation of this species. For this reason, we used two approaches. One was single digest red sequencing and whole genome resequencing. For this, we carried out very extensive sampling, covering over many locations in Hungary. We used live catching with box traps, and also thanks to our international researchers, colleagues, we also acquired samples outside of our country. So overall, it included 64 sample locations and more than 300 samples overall. But out of those, only 165 were used in our analysis. Also, one sample was used to provide for the reference genome. It was assembled by CNAG, and this reference genome is already available in the gene bank. For the red sequencing, we compiled 3 libraries, and the bioinformatic processing included mapping to the reference genome, variant calling, filtering for different measures, linkage disequilibrium, pruning, and so far we have not finished yet with our analysis, but we have a phylogenetic tree and admixture analysis. For the whole genome resequencing, we were basing our sampling from the RedSec. We used our samples from the preliminary results from the RedSec data from 3 main genetic clusters, and we chose 18 agricultural samples, or samples from agricultural locations, and 18 from urban populations. So overall, 36 samples were chosen, and they were sequenced on low coverage, with 3 of them sequenced on deep coverage. The bioinformatic processing steps were very similar, with the addition of a GWAS analysis. Okay, so the most important genomic insights from the RED analysis, based on 73,000 filtered SNPs, we were able to reconstruct detailed phylogeny and phylogeography of the species. And it's not shown here, but admixture and PCA also confirmed that there is a very limited admixture between those regional populations, and we got the same pattern from all analyses. And this suggests an isolation of those regional populations, what we see. For the resequencing data, unfortunately, with the GWAS, we couldn't identify genomic regions associated with urbanization. However, a neighbor joining tree based on putatively selected SNPs showed some possible local adaptations in those 2 populations which were urbanized or from urban populations. So altogether, our sample size was also very low, so we didn't expect much results to see, but it's interesting still that maybe if we would include more samples, what would we find? So also, there were no marked difference between those 2 habitat types, the agriculture and the urban ones, but the genomic diversity based on observed heterozygosity was generally very limited. And we proposed that habitat connectivity and species protection must be maintained and strengthened, with continuous monitoring to be established. So again, the limitation of this study, that our sample size was also too small, and we didn't include environmental variables in this study. During our project, we had several opportunities to engage with different stakeholders, most notably during field at the sample collections, where we exchanged information with local workers, landowners, also with national park management. We discussed the case of the hamster. We also had a workshop at the annual meeting of Hungarian mammalogists, where over 300 participants learned about the case study. And probably the most important milestone that we proposed to elevate the protection status of the common hamster in Hungary. So this study was used to integrate it into the conservation action plan for the hamster, and it had to refine the strategies for habitat conservation and translocation efforts. Also, the protection status was strengthened for the hamster, and it was modified that culling of hamsters was no longer no longer possible without permit. We had other opportunities to present our case studies with the general public. Most notably, we presented the case study at the largest annual agro show with over 10,000 attendees. Also a similar event where approximately 1,000 people attended the event. So far, we have publication, research news published in the Oryx about the preliminary results of this case study. And we plan to revise these results and publish the further results into in another scientific paper in the long term. And in the furthermore, we also would like to expand our resequencing data set, and which would hopefully improve the statistical power for our study. And we would like to also propose long-term genetic monitoring for the species to better understand how those populations are persisting in this region. So thank you very much for your attention, and I'm happy to answer any questions.
Questions from the audience? I think we have it online. This one there. Online? This. I have a question, just a quick one. I was wondering when you're talking about how you can maybe see the effective population size trajectory match the changes in agricultural policies. Do you think this is something that you can see with the data that you are getting? Studying the effective population size and see how this is shifting with the policies in agriculture?
Not yet, but that's also a goal in the long run.
Question. Okay, let's move to the next one then. Thank you. It's really cool to see this. Exciting work. So next one is from Pedro. Is local. Yeah. It's
Can you hear me? Yeah. Well, I don't know. Well, here yes, but at home. Okay. Okay. So Good morning, everyone. So I'm here, as Diego said, I'm João Pedro. I'm based here in Sibiu, and I will talk about a little bit about what we have been doing on our case study on myxomatosis in lagomorphs. So just giving a brief overview. So the myxomavirus is a double-strand DNA pox virus that emerged on South America originally, is mostly transmitted by vectors, many of them the ones that Mary presented yesterday. And although it doesn't create severe disease on Sylviolagos, on the natural host, it is fatal for many lagomorphs. So based on this property, it was introduced in Europe and in Australia to control the population of European rabbits. And in Australia was particularly bad because in 70 years, the population grows from 24 individuals introduced to 1 billion. So this introduction was very satisfactory in the beginning for the purpose. So it creates mortality rates close to 100%. But suddenly start to reemerge again. So basically, what was discussed at the time is that probably attenuated viral strains appeared, or there was resistance on the rabbits. And the study published by, at the time, a PhD student here in Sibiu proved that in fact there was a parallel evolution on the genomes of both populations in Europe and in Australia. So we had a couple of nice target genes in the beginning. So, but the games changed again a couple of years ago, and we start to see cases also on lupus. And at the time, it was the first occurrence on the Iberian hare, particularly. And so apparently this myxomavirus jump species from the rabbit to the hares. I know that they look similar, but they are 5 million years apart, so it's quite different. And what was founded is that there was a new recombinant myxomavirus arising. So there was an increase in virus prevalence in Iberia, and the mortality grows a lot among hares. And the authorities start surveillance starting on 2018, and suddenly they start to see recovery in the population. So apparently the same situation has happened in the European rabbits in Australia. So our case study is based on this idea and on the study that was done with the European rabbit. And so we want to try to understand which genes or pathways drive this resistance, if it's driven by genetic basis. And as long as the case study progresses, we start to think also on looking basically on using this host data that we have to also inspect the myxomavirus itself and other co-infectants. And so this the starting point was the sampling, and the sampling in our heads should be quite simple, but it took almost all the PhD of Beatriz to define what we could consider a resistant individual and a susceptible one. It's much more complex than it looked like. It has a lot of steps. I will not go into details, but in the end, we managed to have 102 susceptible individuals or samples and 96 resistant ones. And during this time, Beatriz was able to also use bioclimate variables to try to predict what was the risk of exposing of the hares in Iberia. And this information is quite relevant because it defines the priority areas to do surveillance, and our sampling in the top reflects quite well these high risk regions. Then Joanna did the lab work, and we produced whole genome data for all these almost 200 individuals in low coverage. And then we leveraged on the use of the 3 reference genomes generated inside ERGE. 2 of them, and the ones that arise first, are part of Jaco's work, and I think you will heard about them in a couple of minutes, I would say. Then our analysis focus on 2 waves, basically. One, the host, so we target the host and try to understand, we compare resistant versus susceptible individuals and on the virus. And here we take advantage of the team of veterinarians that work with us to define a couple of, a target species that normally co-infect with the myxoma virus. And we create a database, we did a competitive mapping, and then we estimate abundance, so virus load, and it's a roughly measure. And to have some background, we also add samples from pre-outbreak. So, and this is quite exploratory until now. We just have 10 individuals of each population, but I will show the results soon. So regarding the host, so we did the genome-wide differentiation scans, and some genes related with the immunity, with the immune system arise, and some of them are the same as on the European rabbit. So quite nice targets for doing genetic monitoring. And then regarding the co-opportunistic infections, first we proved to ourselves that it was possible to recover the myxoma virus from the host data, which is quite nice, because now we can not only target the host, but also the myxoma virus and try to understand if there are more than one variant circulating. But then we also were able to recover some signs, and it's quite hard to see, I guess, but we can see that pre-outbreak, we almost don't have any of these diseases on, at least on the whole sample size that we have. But this is quite nice because we have more data, and we can try to feed these models with more data and try to understand what we recover and what kind of patterns we recover. And this links with the idea that was developed during the case study for the application. And this is based on Leif-Anderson multi-species SNP chip. So basically, our idea is to use the same principle, but to create a SNP chip that is able to target the host candidate genes, but also the myxoma genome and the co-infectants that we are able to detect in this point. So it's like a tool that for 30 euros per sample can give us a lot of information. Even if the target genes or the candidate genes are proved to not be the ones really driven the disease, we have a lot of data that we can play with. So, okay, we have still some uncertainties. This data is still preliminary. We have low coverage, so we need validation. And for all this being possible, we had a lot of partners, mostly in Spain, because the distribution is mostly in the Spanish side. But these authorities have been involved since the beginning and interested in having a tool to speed up the population monitoring, because this species is a key game species in Iberia. So why this is important? Because, well, it can support EU One Health perspectives and national wildlife health strategies. And another thing that was really nice during this case study is that we were able to establish an invited share in game management and biodiversity. So we have one person dedicated to try to use the knowledge that has been creating inside CBU and to try to implement this on the field. And the nice thing is that this guy has a genetic background, so that could help us a lot in trying to pass this information to the right persons. And okay, and regarding impact and communication, well, apart from the scientific outputs, of course, we had a year-long project in high schools. So we have a project running like a normal scientific project where kids were faced with questions and they need to develop work during the whole year until the end, and they use a lot of data that was used here. In fact, they developed the first screening of opportunistic diseases on this data, which was quite nice. And we're present also in hunting fairs. Paulo was present in the last one just this weekend, and we have several oral and poster communications. And so on the next 6 months, we want to validate these candidate genes. We have a lot of data from other projects that can be used for this. And we want to produce the case study deliver, of course. But for the next 2 years, we really want to put in place this multi-species SNP. And we apply a lot for 2 calls, and we are still waiting for results to try to fund this idea. And we will try to guide mitigation and conservation actions with stakeholders. So thank you, and I'm happy to answer some questions if you have.
Nice talk. I just wonder if you can discuss more about possible bias in DNA extraction since the virus is enveloped and you use a common DNA extraction protocol, I guess.
Yes.
But you can recover both genomes at with the same efficiency.
Yeah, in fact, we were surprised that this works so well because we had PCRs, and so we screen all these samples, all these 1,000 samples that we start with targeting PCR protocols for virus. So we knew that all these samples, in fact, this was an outcome of the project, is that all samples that we have screened had the virus, even the resistant ones and the others. So it's completely spread all over the Iberia. So then regarding bias, well, I can't exclude them, but the virus load is really, really high. And when I say really, really high, it's like 10% of what we could get, at least in the susceptible individuals, because this is a disease, a progressive disease, and in the end, the animal is in really bad shape. So the virus load is quite high. The thing that we are not completely sure yet is if we don't have bias on the opportunistic diseases, because we are based on a reference library. So this should be taken with caution and needs validations for sure. But I think that is a quite nice secondary output of this data. That is a shame not to use it. I don't know if I.
Yeah, one short question.
Yeah, well, it's very short. Is this virus can it turn into a zoonosis? Like, is it dangerous for human health, especially because these are game species, right?
Yeah,
so.
Not really for humans.
Okay, yeah.
Okay, thank you very much, JP. Next one is, I don't have the name, but it's Ruby Capra, an old friend of ours.
Next one. In the middle.
Yeah, this is the big black one.
Okay.
Going.
Good morning, everyone. I'm Vanya Danucic. I came from Republic of Srpska, Bosnia and Herzegovina. I work on the University of Banja Luka Faculty of Forestry. I will present today genetic results. My colleague, Dragan Gacic, also work on the University of Banja Luka and University of Belgrade. Also, he's a project coordinator and covers the part of stakeholders and police. First of all, I want to say that we received this project in April 2024, and we lose hunting season to collecting samples, and we must wait the next hunting season to collect samples, and we lost about 7 months to collect samples for these results. I want to thank my colleagues from my team for hard work. Also, thanks our partners for support, and special thanks for organized for opportunity to show our results. Now I will briefly present our results from our study, a case study on the endangered subspecies of Balkan chamois. On this slide, you can see geographical distribution of subspecies for the genus Rupicarpa in Europe. Two main types are distinguished. It's the northern and southern chamois. Northern chamois including 7 subspecies. In Slovenia, red-colored, this part, small part red-colored, alpine chamois is present, and in other Balkan countries, Croatian, Bosnian, Serbia, Montenegro, and Greece, Balkan chamois is present. Our main pressures is climate change and human activities such as habitat fragmentation and hunting, legal and illegal hunting. It's a problem and leading to isolation, reduced population size, and hybridization. The main scope of the focus, we focus on the population of northern chamois in the Dinaric Mountains with the emphasis on the Balkan subspecies. The success of our project measured through key indicators, improved populations connectivity, reduced inbreeding and hybridization risk, and enhanced adaptive resilience to environment change. Chamois is least concerned species by IUCN classification. The main objective is to investigate the current state of genetic diversity in the defeated area with higher inbreeding risk and assess adaptive genetic variation and hybridization risk in Balkan chamois to inform conservation strategy. One of the key questions is how is genetic structure and adaptive variation distributed among population, and which populations are more vulnerable, and where is the inbreeding risk highest. We hypothesized that the Balkan chamois possess unique adaptive genomic features, but climate change and hybridization treat their genetic integrity and adaptive potential. The reference genome of northern chamois was published in November 2024 by first author Elena Gužan and colleagues. It's available on the link shown in this slide. Sample designs cover 5 countries: Slovenia, Croatia, Bosnia and Herzegovina, Montenegro, and Serbia. In Slovenia, provided the samples from alpine chamois, and another countries is Balkan chamois. We took alpine chamois to comparative analysis. We in the project planning to collect 120 samples, but at the end of we collected 150 samples, and for these results show with 46 samples. All samples we collected following the international national regulatory, ethic regulatory. On this slide, for whole genome sequencing, we use a workflow combining standard informatic tools. You can see this on this slide and this description of that workflow. And this is, lets us over 5 million SNPs across that data set. Mitochondrial diversity of extended population show us distinct patterns. Slovenia, for example, red colored, show some unique haplotypes and indicated isolations. In the central part of haplotypes, central haplotypes is green, is population from Croatia, and show us connection in another group. And we have Montenegro, population is purple, is peripheral haplotypes and indicated two unique line genes. Also, Bosnian population is yellow colored, gray and pink colored. We take three populations from three different mountains, and they show us each of them has a unique haplotypes, but some connections show us some connection from the past, historical links they have. Nuclear diversity in this graph show that population from Croatia has low genetic diversity. It's a red colored. Slovenia show us to low to moderate. Bosnian population show us moderate genetic. Oh, sorry. Moderate genetic diversity. The highest genetic diversity show Montenegro population with green colored, and Serbian also high genetic diversity and show us genetic stability. PCA for show us for completely 32% total variation, show clear geographical structure at the country level. You can see in the left have Slovenian population. Show us high genetic similarity among population differently for another country. Also, the population of cluster of Serbia is near Bosnian and Herzegovina population, and clusters for Montenegro is near to Bosnian and Serbian population. Each region has its own genetic signature. Proportion of the genome within rounds of homozygotes. Developed plots show clear differences in homozygosity across country. We have the Bosnian and Herzegovina and Slovenia generally have a low value of FRO. It's a low in breeding. In Croatian has a widest range, have a individual with a high inbreeding, and some individual is near null. And also Serbia show us intermediate distribution of FROH value. Based on our results, we can say that implication for actions can be focused conservation efforts on isolated population with low genetic diversity. Also, consider translocation between neighborhood population with higher genetic variability, and also our results will be contributed to the development of target conservation strategy and may inform potential reintroduction or translocations efforts, especially from Srpska and Serbia. And now, in my words, my colleagues.
With this, I have this. First of all, I'm sorry for my English pronunciation because I got some kind of allergy yesterday. Our case study included in total 43 stakeholders and their structure is shown in the first paragraph. We included the most relevant and important stakeholders, such as ministries, public companies, institutes, and non-governmental organizations from Republic of Srpska, Montenegro, and Serbia. And also we have many important stakeholders from Slovenia, Croatia, and Greece. We held 3 workshops, the second of which is held on the mountain Tara, and that was truly international workshop. Then we released 2 questionnaires and received answers from 45 participants, and we informed all stakeholders about their benefits of our case study and why is important their participation for us. But more valuable contribution was made by the presentations on the chamois status at the national level in many countries. First from Slovenia, Croatia, Bosnia and Herzegovina, Serbia, Montenegro, and finished with Greece. Also, chamois status, chamois Balkan status was presented for many national parks and many hunting grounds. At this workshop, we promoted ERGA and BG, and we also have a possibility to collect many useful informations during the field trip to the chamois habitat in Tara National Park. This second international workshop represents a good example of a successful way to collect information from stakeholders and in terms of cooperation with stakeholders and to join work to the implementation of genetic research. Some stakeholders from Republic of Srpska and Serbia plan to reintroduce or translocate chamois individuals in the coming period. The key message and the most important result of our case study is that discussions and consultations should be held first and mandatory, and only then should start release of initial chamois stock, first in small enclosure area and after adaptation in the surrounding free nature. It is necessary to explain in detail the well-known guidelines about reintroduction or translocation from domestic or international experts and organizations. For example, IUCN guideline for reintroduction and translocation. Also, it is important and necessary to analyze together with stakeholders previous programs and to determine the important mistakes and problems and prevent their repetition. The primary challenge in successfully revitalization of Balkan chamois in Bosnia and Herzegovina, Montenegro, Serbia, such as in other countries, Macedonia, North Macedonia, Greece, Albania, and Bulgaria. Is controlling illegal hunting and implementing penalties for the offenders. Our case study was promoted on many places. We had impact on over 100,000 hunters and many local communities. For example, we participated on the most famous hunting, fishing, and tourism fair held in the Novi Sad. Before the third workshop, we participated in the very popular morning program on the regional television. Also, information about our case study was published on many websites and in two very popular magazines, first of which is in Serbia and called Predator, and second is in Bosnia and Herzegovina and called Šume. In addition, we prepare and make 20 short programs about chamois and about our case study, which has been shown in this year and last year on the many local and regional television stations. In the future, the most important action is to start studies on the interactions between new established red deer population in West Serbia and their impact on the indigenous chamois populations in Bosnia and Herzegovina. Also, we have in plan to finish our genetic analysis and publish that results. And also we have a collection of many data about characteristics of chamois horns from Republic of Srpska and Serbia, and we hope that results also be published. And it is very important to point out that thanks to the participants on these workshops, we have prepared a set of conclusions and recommendations, in total 27 conclusions, and they will be sent to the most relevant stakeholders, and we believe that will very significantly improve current status of the Balkan chamois in this part of Europe, and also help stakeholders to better manage by populations of chamois. At the end, I want to thank all organizations who finance our case study, and also to my colleagues from other institutions who participated in our case study, and also to many stakeholders who help us to get these valuable results. Thank you for attention.
Very quick, short question. Otherwise, we can leave the question for the coffee break. And the same for the folks online. They can drop the questions in the chat, and we are going to do our best for replying throughout the day. Question? No? Okay, let's move on to the next one.
It was everything clear.
Yeah, but this is because
your microphone.
Yeah, the microphone. And but the next one is online. It's a familiar face for us in Berlin. Justina, are you there?
Yes, hello, can you hear me?
I think we need to do something on the technical side. How do we increase the volume?
Can you hear me?
Yes, but just wait a second. The audio is going to the.
We have maybe to change.
Yeah. Can you say something?
Are you talking to me? Yes.
Yes, perfect. Now we're hearing you fantastically. Okay, wait a second.
So shall I share my screen?
Yeah, the screen is. Can you? I think now you can share. Will you try share the screen? I don't know if it's coming. Justina, are you there?
Yes.
Yeah. Can you share the screen?
Yeah.
Yeah, perfect. Maybe we can. 1 second. Maybe we can make it that. Yeah. Yeah, perfect. Okay, the floor is yours. Thank you very much.
Thank you very much. So let me introduce my case study species. This is the aquatic warbler. It's basically a small brown bird. It is a long-distance migrant that winters in Africa, and most importantly, it is specialized to breed on fens. And fens are a type of wetland from temperate Europe. This is a wetland with low grassy type vegetation without too many shrubs and trees, and it is permanently waterlogged. There's a lot of water in there. And this kind of habitat used to be quite widespread in broad river valleys, unchanged river valleys in temperate Europe. However, over centuries, most of such wetlands got lost. And this happened for several reasons and started as early as in the Middle Ages with peat excavation to produce heat, and then it continued with river regulation, building dams, and also draining wetland for farmland purposes, such as hay production. And finally, because many such wetlands or fens were overdrained, they got overgrown by reeds, trees, and bushes. So as a result, the breeding habitat of the aquatic warbler, which once stretched from the Netherlands in the west to Western Siberia in the east, got shrunk dramatically, and nowadays the species breeds only in East Central Europe, in just 3 countries, in Poland, Belarus, actually 4 countries, but most of its population is only in 3 countries: Poland, Belarus, and Ukraine. And this dramatic shrinkage of the breeding habitats corresponded to a steep decline in the population size, which just in the 20th century went down by over 1 order of magnitude. So nowadays, the aquatic warbler is among the threatened animal species according to the IUCN. So in my case study, I used whole genome sequences to examine genomic diversity in breeding, demography, and structure of some past populations to inform current conservation programs such as translocation. I used existing DNA extracts from samples collected in 2 populations. The first population was the Biebrza marshes, which is marked with a red circle here. It was and it still is a large and stable population with about between 4,000 to 8,000 mature individuals. And this population is quite well connected to other populations within the breeding range, which are marked in yellow here. Biebrza was sampled in 1997 and again after around 20 years. And the second population sampled was West Pomerania, which is located around 600 kilometers westward from Biebrza, with scarce habitats in between the two populations. West Pomerania was sampled in 1999, and at that time it was a moderately sized population, declining quite fast, and it was quite isolated. It actually went to extinction just a few years ago, and in 2023, a translocation scheme was started to restore this population. So using information from this genomic data set, I will be able to provide some information for the translocation project. And the sequences were produced by an external company, and then I analyzed them using a typical pipeline, arriving at nearly 6 million SNPs, or a bit less than 1 million after structure filtering. And then I fed this to a population analysis program, and the sequences are now public, and the pipeline is public as well. So when I looked at the genomic diversity, I saw that it is quite comparable between the three populations sampled. It seems to be slightly lower, at least in terms of the percentage of sites polymorphic in Western Pomerania. And in objective terms, it is overall quite low and comparable to one of the rarest birds, such as the hihi, which is endemic to an endemic island bird from New Zealand. So this shows that the aquatic warbler with this low genomic diversity could have lowered adaptive potential. Then I looked at genomic inbreeding with runs of homozygosity. And this reveals that inbreeding is higher in West Pomerania compared to Biebrza. And there seems to be also a slight temporal trend in Biebrza with increasing inbreeding. And compared to another bird endemic to islands, the birdlot's pipit, which breeds on Canaries and on Madeira and has elevated inbreeding. So when I put this in this context, this shows me that the aquatic warbler also has quite elevated inbreeding, although it's by no means an island species. Then I reconstructed the recent history of the effective population size. And this revealed that the Biebrza population went through a genetic bottleneck around mid-19th century, which is when at the time of the Industrial Revolution, during which wetland degradation accelerated. So this is consistent with the history of degradation of the habitat. And recently, there also seems to be that the effective population size also seems to be declining in Biebrza, although this requires more genetic monitoring to confirm. And in turn, in the isolated West Pomerania, I basically found a low effective population size in general compared to Biebrza, and most likely two bottlenecks, suggesting that genetic drift operated more strongly in that population. And also, I found out that the effective population size at the time of sampling was around 1,000, which is just as the recommended effective population size for a population to retain its evolutionary potential. So this shows that in 1999, evolutionary potential in this population was at risk. And then finally, I examined the genetic structure and differentiation. At first glance, West Pomerania clustered apart from Biebrza. However, admixture did not show more than one cluster So all the populations actually belonged to one genetic population, and differentiation was very low. So either there was still some gene flow at the time of sampling, or too short time has passed for these populations to diverge. So overall, these results show that for West Pomerania, genetic factors could have enhanced extinction. And this is important for the translocation scheme that is now being carried out. So in order for the story of West Pomerania not to repeat itself, then it is important to restore some stepping stone habitats between West Pomerania and the nearest largest population, and to increase the amount of habitat for the aquatic warbler. And this is central for successful translocation. In Biebrza, diversity could be declining, although the signal is quite slight. So it is important to continue genetic monitoring, but nevertheless, it is crucial to restore the degraded habitats within the whole metapopulation to which Biebrza belongs. And here, an important limiting factor is, of course, the war in Ukraine. I spread the news about the project and its results among the most important stakeholders, the NGOs involved in the translocation scheme from Poland, Germany, and Lithuania. And I promoted results and the project on social media, during conferences, as well as to laymen at local community meetings. And these results, of course, they will be important to guide translocation and genetic restoration of the aquatic warbler, and they can feed some protection plans for Natura 2000 sites in several countries where the species breeds or where it used to breed, and protection plans in national parks and reserves, as well as spatial planning. Like I said, these sequences are now public, and the manuscript is under construction. And in the coming months, I will communicate the results to a group that is dedicated to protection of the aquatic warbler to guide translocation. And later on, the pipeline can be used for further genomic monitoring of translocated populations. And I would like to thank all the collectors and curators of the samples and the institutions that were involved in this research, and also to my collaborator who did the analysis with me. And thank you for your attention.
Yep. Do we have some questions for the audience? Quick one. I have a question for Justina about the runs of homozygosity. Did you analyze the length and try to fit the size of the length of the size of the runs of homozygosity in time? Try to pinpoint in time where these events generating the inbreeding are happening.
So this was done indirectly by reconstructing the trajectory of effective population size, because it relies on linkage disequilibrium. Mm-hmm. But to look at the change of runs of homozygosity in time, I just had 2 temporal points, so I could only compare Biebrza from these 2 time points.
Okay.
Hi, thanks. It's Jaakko Poismaki. Great talk and super interesting to hear. I was wondering, you simultaneously see high levels of inbreeding, but then on the other hand, no population structure. And I was first thinking lack of population structure would imply that there's kind of no breeding site fidelity, but then on the other hand, you get inbreeding. So do you have any explanation for this discrepancy?
Generally, in birds, birds are quite mobile, especially migratory birds. So structure is usually low across this geographical scale. I think this inbreeding that I found is mainly due to the recent decline in population size and increased action of genetic drift. So that's probably the reason.
One more one more question, please. You mentioned that the West Pomeranian population went through 2 bottlenecks. Can you exclude the possibility that what you can see on the effective population size reconstruction is not the sign of historical immigration into that region instead of 2 repeated bottlenecks?
Yes, that's a very good question. So it could either be that there was a decrease in population size, or there was a founding event followed by expansion, or there could be, as you're saying, some immigration from some distant population. So with this data, with this reconstruction of the effective population size across time, it's impossible to distinguish.
Okay, thank you very much, Justina. I think you have a couple of questions online on the YouTube channel, so please take a look. And then
okay, thank you.
With the next it is yeah, it's, you need to, yes, that's the let's go. So Lynette from Copenhagen. Yeah, first. And
yeah. Thank you. Hi, everyone. I'm from Copenhagen Zoo. My name is Lynette Rasmussen. And of course, I didn't do this project alone. I also have Christina Wilson, who did the keynote yesterday, as well as Hernan Morales, who's from Copenhagen University. Oh, I guess I should tell the group about the title. Pretty much what we want to do is what the title is saying, using genomic data to guide conservation effort for ex situ populations. So for many of you guys, I'm not sure if you're used to using words in situ and ex situ, but ex situ means outside of the natural habitat. So this is referring to captive populations, and in situ is referring to efforts within the natural habitat. So this is usually referring to the wild populations. And I work within the zoo world. And in the zoo, we are divided up often in regions, and the Copenhagen Zoo belongs to the European Association of Zoos and Aquaria, and that stands for EAZA. And within EAZA, then we have what we call these EAZA ex situ programs, and for short, which I will be referring to for the rest of the presentation, is EEP. Many people think of this as breeding programs, but that's not only what they do. They have many other roles. So some things that they have, and I'm sorry, this is very small, is that there, for example, could be ARC populations could be a role, and that means that they're extinct in the wild, but we still have them in captivity, and so we're keeping them going. It could be insurance populations, so we have captive populations that are going to eventually, hopefully, be set out again. Or maybe even population rescue, where we take them in temporarily. Maybe there's some habitat destruction that will be restored again, and then we can put them out again. There can also be a lot of other things, such as introductions, reinforcements, both demographically, if there's populations that are lacking maybe males or something like that, or genetically. They can also be used for things such as education, to educate everyone about maybe an endangered species or another species that is similar. Capacity building, so we can give our expertise out to other conservation efforts, as well as research such as this project. So they have a lot of different roles in just a breeding program for captive populations. So I'm going to talk to you guys about two different EEPs. One is the European mink. This is a quite old EEP. It's very established. It's very stable. And right now they have them divided up into a Western and Eastern subpopulation, which is similar to what we see in the wild. And this is the idea is to try to keep it as similar to the wild as possible. However, because we only have so many individuals, there's current talks about, do we start combining these subpopulations in our captive population? In the wild, we can see, so in the in situ populations, we can see that there is a decline within the populations, as well as we can also see there's a decline in the genetic variability. So the role of this EEP is, of course, insurance population. They're actually already working on population restoration in Germany, Estonia, as well as Spain, and they're doing introductions. They're using these species as an education to show how important ecosystems are and what important roles they have in the ecosystems, and of course, research and advocacy. Additionally, very recently, there've also been talk about cryopreservation of the ex situ population to think for the future use. Do we want to start taking gametes now and cryopreserving them so we can have them for later use? Then we have the Atlantic puffin. This is almost the complete opposite. It's a very, very new EEP. We're only on generation 1, 2 generations. So we don't have a huge population either. We had a very nice founder population. However, they haven't been very good at breeding. So we don't have a whole lot of new individuals coming in. There is also a lot of individuals that we've received that we don't know the origins. And if we don't know the origins, they do not participate in the breeding program. So this also limits our numbers of how we can pair these individuals. So a little bit of problems there. In the wild, we know that there is climate change is affecting the species quite a bit. It's affecting their food source, which then in turn we believe is also what is causing the lack of hatchlings each year. So each year there's reports of there's fewer and fewer eggs and even fewer and fewer hatchlings. So the roles here for this EEP is insurance population for the future. And this is very new. And so we're thinking, okay, we don't need it right now. Introductions aren't needed right now. But we know introductions will be needed probably very soon, considering the lack of hatchlings over the last few years. There's advocacy involved in this one, as well as capacity building. We are sending vets and zookeepers to the various rescue centers in Iceland so they can disseminate their expertise to them, how to care for these species, as well as fundraising. So those are two that I'll be talking about today. And then the idea for the success metrics is understanding the EP genetic composition to ensure the roles of the EP. And kind of what I just said with the success metric, that is kind of our idea is how do we can use genetics to understand what we can use our populations for insurance populations for the wild. So some of the questions we have is what is the current genetic composition of our EEP? Where does the ex situ population align with the in situ population? And how can we ensure the role of the EEP? I used, obviously, 2 different reference genomes here. So for the Atlantic puffin, it was a pretty decent reference genome, as well also for the mink. But the first one there you can see is the Atlantic puffin. It has a BUSCO score of 97.3%. It's divided, it says it's scaffold, but it's actually chromosome level. And so we have 24 autosomes, 2 sex chromosomes, 1 mitochondrial genome, and then 1 unplaced scaffold. The mink genome is also very similar, also a great BUSCO score of 98.2. And it also is in chromosome level. So we have 20 autosomes, 2 sex chromosomes, Unfortunately, no mitochondrial here, but there is with the same individual on a different reference for just the mitochondrial, which is a bit weird they split it, but yeah. And then there's actually 3 unplaced scaffolds, which are actually designated to a chromosome, but they don't know where it belongs in the chromosome. So interesting structure there. So for sampling efforts, we want to get 16, we got 16 EEP individuals to represent our founders. And then we also actually included historical. And why we include historical samples is to get an idea of how it actually looked in the wild in the past. So we understand what diversity was there before maybe everything went to shit. We were able to get 23 historical samples for the puffins, primarily from the Natural History Museum in Denmark, as well as the Museum of Comparative Zoology at Harvard University. And we were very lucky that there was already actually 117 recently published genomes available online. So we were using those as well in our analysis. Then we have the European mink. We have 15 EEP individuals representing our founders. And we have, unfortunately, only 6 samples. It was a lot harder to find historical samples for the European mink. And we received those from the National History Museum in Paris, as well as the Finnish Museum of Natural History. There is currently not very much published data for the European mink that we could use, but we are in contact with other researchers who are promising to share, and they're also going to be publishing very soon. So we're waiting on that. We hope to get, or we're planning, or we have done, sorry. We have done whole genome sequencing. We were hoping for 10x. For the EEP samples, we hit pretty well. We got between 8 to 16x coverage. However, the historical samples didn't go as well. We only got about 3 to 8x. So as you probably could maybe figure out, this is still a work in progress. We actually just got our data back a few weeks ago from the sequencing center. So unfortunately, I've only managed to map and clean up the data, and I was really hoping my SNP calls would be done yesterday, but they weren't. So instead, I'm showing you actually the published data from the puffins that I was talking about with the samples. And this is the structure they've seen. Our plan is to use this and project our own samples on top of it to see where the different puffins land, and especially the ones we don't know the origins from, where we really want to know so we can include them into the AP. So that's some of our future data analysis to come. We pretty much, our main goal is to identify unique and key genetic material that would be vital to the EP to make sure that they maintain for future introductions. But also, we also want to make sure that we work with the EP coordinators to ensure the roles. So for the mink, this is one of the main priorities, is trying to figure out which individual minks do we want to use for cryopreservation. The other thing is we want to help figure out if combining the Eastern and Western population is a good idea or not. And then for the puffins, a big thing is how do we create a better, more stable breeding program for them. For stakeholders and engagement, we've already been in contact quite a bit with the population biologists at the executive office for IAZA. And they are the ones that also help assist the EEP coordinators. We've also been in contact with the EEP coordinators to make sure that they understand what's going on and how our information can help them, as well as the holders. And the holders are the people who actually have the animals. Of course, we're also in contact with other researchers who have worked on these species, so we can work together and find out more information from each other. For policy, all this information will be implemented in the as a long-term management program, which is the program that the EEPs follow. And also, we Their EPs are in a lot of communication with the MINC release programs, both in Spain and Germany, and we work very closely with them. So they are also part of this and then being informed, and they are also altering their policies based on what we find. Then we have the impact in communication. Of course, we hope to make some scientific articles out of this. It will be in the long-term management program, as I mentioned. Then there will be dissemination for the holders and the EP coordinators, as well as conferences like this. For the next steps, obviously the next year or so, we'll be doing data analysis and publishing as well as dissemination. We do have one risk we're a little worried about, which is if our coverage for the links are going to be high enough for the historical data, but we will figure out how to deal with that as we go. Thank you.
So if we have questions, maybe we can leave them for the coffee break and also online. Okay, so we can apply that line, and then we have the final. Yeah.
Oh, sorry.
We have the final. Thank you so much. Final case study of the session.
The red one.
So are you waving me what time, or you will wave me? Okay. So thank you for listening to this last talk. So I'm going to talk about our case study on the conservation genomics of the sand dune shrub Armeria pungens at the latitudinal margins of its distribution. So probably you didn't notice, but some Armeria plants were appearing in many of the slides of puffins, but probably you didn't pay attention because many of the species are coastal. So we are going to talk about a Mediterranean one here. Okay, so I'm working at the University of La Coruña as an associate professor, and here we go. So the genus Armeria, it has a center of diversity at the Iberian Peninsula. So in this genus, there are 54 species in the Iberian Peninsula, and many of them are endemic species from mountains on coastal areas. So here we're going to focus on the coastal ecosystem, and in this ecosystem we have 12 species which have either endemic or fragmented distributions, and they are all along the Atlantic coast of the Iberian Peninsula. So you can see the distribution ranges, which are restricted to many of these coastal species. And today we're going to talk about one of these species, so Armeria pungens. And this one has a fragmented distribution. It has a main distribution range along the southwestern coast of the Iberian Peninsula. And then it has a distinct population in the offshore archipelago of Thies Island in southern Galicia. So this population is 400 kilometers away from the closest continental population. So this is quite a disjunction. So this is sand dune shrub, typical from stabilized sand dune. So I don't need to remind you that these ecosystems are very valuable, both in terms of hotspot of biodiversity and the unique specialized, very specialized species that occur there. And also these habitats suffer increasing pressures from human disturbance, mostly from housing and tourism. I don't know to tell you about this. So I did study this species many years ago for my PhD with the Sanger markers. So I studied 5 different Sanger markers, and we identify remarkable divergence in 2 lineages in the southwestern continuous distribution range. So there's a lineage in southern Portugal and a divergent lineage along the entire Gulf of Cadiz in pink there. So I've been working outside space for many years, over 10 years, and then I never worked again with this genus. And then when I came back to Spain 3 years ago, I started again, and I started working with next generation sequencing data. And the motivation for this was when I was granted the sequencing of the whole genome of this species, and that motivated me to keep getting new ideas. So the aim of this particular case conservation study is to estimate genetic erosion using like modern indicators, okay, that indicators that are based on next generation sequencing and that are benefiting from whole genomes. So in particularly, we will study first the contemporary effective population size, which is related to genetic loss by drift, and it has actually included in the last convention of biological diversity of indicator of genomic erosion. And second, I will focus on the estimation of deleterious mutations from genomic data, which might accumulate, as you know, at the margins of the species populations and in the small populations when genetic drift overpowers purifying selection. So we are, this study is aiming at estimating these two parameters and also more standard genetic diversity statistics. So our formal hypothesis is to test if there is a genetic signal of erosion in marginal populations that might compromise their long-term adaptability. In particular, if there is reduced contemporary NE, reduced genetic diversity, or accumulation of deleterious genetic variants. So for the contemporary effective population size, there are not many studies on plants. It's pretty hard to estimate with a high quality reference genome. Here we are attempted to estimate contemporary NE from linkage disequilibrium in genomic data based on these two approaches, NE estimator and current NE. In the case of deleterious mutations, they are also understudied in wild plants, basically because we need annotated genomes to be able to estimate them. So we can estimate them bioinformatically from genomic data based on two main approaches. The first approach is the approach that is based on the genetic change at the protein level. So we are planning to do this with a SNP-F. In this approach, deleterious mutations are those that introduce, for instance, stop codons or frame shifts. That would be deleterious mutation according to this approach. And the second type of approaches, they're focused on, they're based on the evolutionary conservation. So deleterious mutations would be those that occur at very conserved sites, which are assumed to be essential for gene evolution. So mutations occurring at this size are very likely to be deleterious. So we are implementing this in the frame of another study, PhD project in our group with a large language model, so protein language model, so AI methodology that, to our knowledge, hasn't been used in plants, and we are optimizing it for plant data sets within another project. So then, so to study this genetic erosion, this is the sampling that we achieved. So we sampled 100 individuals of Armeria punyens and 7 outgroups in 3 populations: so northernmost populations, central population, and southernmost population in CS Islands, Sines Beach in Portugal, and Trafalgar in Cadiz. And then we sample 31 individuals per population, taking care of have a good spatial representation of the range of the population, which seems to be very important to estimate NE. And the three areas are natural parks. Okay, this Islands is a national, belongs to the National Atlantic Park, Atlantic Islands Park, and Sines and Trafalgar are within two natural parks. So as I mentioned, we made like an effort really to cover, to get a representation of the total distribution of the individuals in the population for these estimations. So the protocol that we used was the RAT-seq. So this species has a kind of large genome, 4.3 gigabases. So that becomes not very affordable for shotgun sequencing. So we are using DDRAT, and maybe we will use HiveSeq capture for the projects. And we sequence in a NovaSeq with a high from our for our we aim to sequence quite a lot per sample, like 2 giga. And so we managed to get an assembly with a mean coverage of 31x and genotype quality of 35. And the number of SNPs that we genotype, depending on the filtering scheme, ranging between 31K and 16K SNPs. So this is the bioinformatic assembly pipeline, so pre-processing FASTQ and Chemomatic, then the assembly with stacks using the reference genome. We made a parallel removal with the HD plots, and then we work quite a lot on the filtering based on the quality and different thresholds of minimum allele frequency, and allowing a missing percentage of 20%. So we were mapping those against the reference genome that was sequenced within the second round of the BGA ERGA call, which, as I said, is 4.3 giga. So this genome hasn't been annotated yet, but within the frame of this project. So so we are working to manage if we can get. There is a preliminary annotation now, since that I very recently. So we analyze the genetic structure using admixture plots and principal component analysis. And these plots confirm that the three populations are actually well differentiated, as we already knew from the Sanger data. So this is also confirmed with a more detailed sampling and many markers. So we have an optimal number of 3 gene pools based on admixture. And here, we have well differentiated groups. Maybe CS, the northernmost population, and the central population seem to be a little bit closer, but still well differentiated. And we confirm this good differentiation with the structure plots with very stable assignment across repetitions and delta K of 3. And these are our results regarding genetic diversity and contemporary effective population size. So for the genetic diversity that it was measured with the higher stats and stacks. So we found a higher genetic diversity in the central populations of CNS, like both the heterozygosity and also the rarefied allelic richness and nucleotide diversity. Okay, so the central population of CNS show a higher diversity than the northernmost and southernmost population. And the inbreeding estimations were negative or close to 0, which makes sense because it's an obligate outcrosser. It has a sophisticated mechanism of stigma and polymorphism to force outcrossing. Then we estimated the contemporary effective population size with the two softwares, N estimator and current NE. So we found that CNES had showed a higher NE than the northernmost population, but we found a very high NE from the southernmost population, no? Even though that genetic diversity is small. So we are still trying to figure out the interpretation for this result. One of our working hypotheses is that this is biased because this population seems to have been experienced a recent expansion. So that might be bias in the estimates, but this is a working hypothesis for now. Also, I have to say that in some of the filtering schemes, we had higher NE values for the northernmost population. It was not totally, it was not 100%, but most of the results show, yeah, a very low NE. So, so we did quite a lot of, yeah, engagement with stakeholders and outreach within this project, because this plant is very popular. It's known by the public, and it's very easy to do outreach. So we are involved in citizen science project. We've been involved in this for 4 years now. We coordinate citizen science events in the region of Galicia, which also happens at the national level for the identification of plants using apps. So either we organize like fieldwork ourselves for the public, or we encourage universities, high schools, or different NGOs to organize their own events. So we collaborate mostly with this NGO, this naturalist group, Grupo Naturalista Habitat, very, very active and yeah, popular in our region, and also with the outreach project, scientific outreach project, which is called Camino Conciencia. It's a project that combines pilgrimage, so like hiking with scientific outreach. So we have used those also to make outreach of Armeria pungens. Okay. Also, this Camino Conciencia is organizing hikes along the coastal paths of the Jack's pilgrimage. So it's ideal. We also have collaborated with the national parks, where we made the collections, particularly very much with the Atlantic Island National Park, which is the closest to our region. And we hope we can make some panels together with them for information for the public. Also, we hope we can attend the annual meeting for the guides of this national park, so that we can teach them our results, and they can talk about the importance of this species. So the reason why it's so easy to do outreach is because this species is very well known in northern Spain. It's the herba de enamorar, or herba enamorada. So it's a plant that is attributed properties to make fall in love. So this is included in some popular song books, where they describe that this plant can be needs to be introduced in the pocket of the loved one, and then that's how it works. And actually, there is a collection of San Andresinos, which are figures made of bedcombs, which are symbol of hospitality and friendship. And the one that represents love is really like Armeria, one of the coastal Armeria. So it's really well known by the public. So that's sorry, I'm very lost in. Okay. Sorry. Yeah, I'm almost. Yeah. So that's so we feel that when we do outreach actions, they have a kind of a lot of visibility. So the release of the sequencing genome was published in 4 national print media and 10 digital media. And we have also participated in science fairs, in particular the Scientist Night in 2024 and the Street Science Day in 2025 in collaboration with the scientific museums of our city. So this project has also involved training. So we have been lucky enough to attract a master student who is actually the one who has Carmen Soane. She has done all the bioinformatics assemblies. She's a master student in bioinformatics. And she has been also involved in some of the outreach activities. And we have been also very active posting our fieldwork trips in social media. We got about like 1,000 views per post. So the next steps, just to wrap up, will be to estimate the deleterious mutations based on the annotated genome. It has just been annotated based on homology by Aureliano Bombarelli, which is involved in the ERGA annotation committee and is working at the Research Council of Spain. We will intend to do an estimates with GONE, which is the most demanding program in terms of number of SNPs. And then we will be holding Carmen Seoane defense, master's defense, and and then we will launch a new Armeria project funded by results of the Carmen analysis on this case study, and a second paper based on the genome. Maybe we'll compare it with the Limonium bicolor, which is the closest genome available, which is not very close, but yeah, we still don't know. And also we are planning to hopefully to generate RNA-seq data to get a like high quality annotation. Hopefully we can get support from ERGA to yeah. Sorry. Yeah, okay. Sorry, I am.
Do you have one last?
No, I think.
Okay.
Yeah.
Okay, thank you very much. The coffee break and the talks online, please drop the questions online.
Okay. Thank
Yes,
you.
so we are running 20 minutes late. We're behind schedule, so let's try to reduce the coffee break to 20 minutes and be here at 11:40 local time, and so people online it will be 12:40 CET. So, hi everyone, welcome back. So just one announcement. So at the end of this session and before going to have lunch, please meet outside so that we can take the group picture, okay, for the conference.
Perfect. So we can get started. So we have a series of case studies also now. So we will start with one from Gabor on this very beautiful beauty birch mouse. So the floor is yours.
Thank you. Thank you very much. Hopefully I will be able to move it.
Just click on the slide.
Oh, okay, it works now. Thank you very much. So thank you again. I would like to say thank you to the organizers of VGE and ERGA because they made it possible for me to come. And in my talk, I would like to present you our results with the Hungarian birch mouse, which is one of the rarest mammals in Europe. Why I'm saying it's one of the rarest mammals in Europe? Because it used to be a little bit more widespread because it was registered from several locations in Hungary and also in the neighboring countries, Austria, Slovakia, Serbia, and also in Romania. Having said that, after the end of the 20th century, only two populations remained from this species. One is in this part of Hungary, and another one is in Transylvania, very close to the capital of Transylvania, Cluj. And because of the big genetic distance between these two populations, they are considered to be different subspecies. And actually, we have quite a lot of interesting evolutionary history data. And it seems that this is the refugial territory for this group of mammals. And probably because that was the refugial territory, this is the explanation why we have so big genetic difference between these populations. So the thing is, we have only two populations for the species, and for the Hungarian subspecies, we have only a single population existing. And although we have only this population, it's considered to be endangered by IUCN. I believe this is an over-optimalistic or so it's too much, it's not a fair classification to be called this endangered. And this endangered classification comes from the fact that according to IUCN, the area between the two existing populations could hold some unknown populations, but I don't think so. Okay, so within Hungary, the last population exists in around 12 isolated habitat patches, and we estimate the census population size around 1,000 individuals. So actually, we really have a super endangered mammal species here. What were our goals? We wanted to, well, the goals are very, very basic because we really need to learn as much as possible from this last population, and we wanted to understand population structure, and of course we wanted to give exact management indications for the nature conservation. And also what I must add here, these species have a very, very hidden lifestyle. It's only nocturnal, and it's very, very difficult to observe the species on field. So we also don't know quite much about the ecology of the species, or what we know is not enough. So evidently we need quite a lot of genetic information to try to extrapolate from the genetic data to ecology. For example, we don't know how much the subpopulations, the 12 subpopulations are communicating with each other. So what we wanted to do, we wanted to generate a reference genome, and we had some basic information to do that. No reference genome exists in this rodent family where this species belongs to. So actually we were really in the darkness. And what were some lights in this darkness are the chromosome count from Serbia. The diploid chromosome count is 26. And we also have the karyogram of the closest relative, the sister species, which is presented here. I need to draw your attention to this dwarf Y chromosome, which is quite normal in rodents. And we also have a genome estimate, which is around 2.8 gigabase pairs. We obtained from commercial service provider a suboptimal amount of hi-fi reads, and we also obtained high-C reads, and we used a very, very, I think, general pipeline to assemble the genome. And beside the whole genome, we collected samples from 60 individuals from all subpopulations, and we did a classical RADSEC analysis with single digest RADSEC data. Let me please don't go into the filtering details. So these are the results. We managed to assemble, I believe, a chromosome level reference genome, because we found in haplotype 1, 13 large scaffolds plus 1 small that correspond to the 12 autosomes and the sex chromosomes. And in haplotype 2, we have the 12 large scaffolds, which are corresponding to the 12 autosomes. The Busco statistics are not that bad, although I would say could be better. This is probably because we had a suboptimal number of reads from hi-fi, and but that was evidently a useful reference genome. This is the insight from RADSEC dataset. This is the PCA. You can see that we have 3 groups of individuals. This is 1, 2, and the third one. And this is, of course, also mirrored in the admixture plots. And what conclusions we can draw. One important conclusion, I think, that there must be connection between the subpopulations, which are colored differently on this plot, because they are admixed. So this is a kind of indirect evidence that those subpopulations, what we see on field, must be communicating with each other. But we can also see some fragmentation. And if we check the fragmentation, what is the cause of that? There are some large drainage channels, but they dug in the 1970s. And most probably these started to separate some subpopulations, and they simply started to drift away from the rest of the population. So what are the actual recommendations for our collaborators? We need to improve the connectivity between these subpopulations, and there must be some corridors established. And yes, the main stakeholder is our tight collaborator, the Buk National Park, who is managing this protected territory. And we also have to say that Budapest Zoo is also our main stakeholder, because they are planning to do some ex situ conservation program. and very close, very similar to what we have seen in the first part of the sessions. We would like to provide genomic data to establish the ex situ populations. This year was the year of birch mouse in Hungary, and together with this program, which can be translated as Wild Watch, there's a lot of public awareness now in Hungary about this species that has accumulated in a fashion company commercializing a side bag which features the Hungarian birch mouse. I need to consider buying one, but I must confess I have not bought one. Yeah, so policy relevance at the regional level, of course, we will give quite a lot of recommendations to the national park, and certainly in frame of this project, we completed the species conservation action plan of the species. This is the official action plan to conserve the species in Hungary. And at the EU level, I need to say that we must save one of the rarest mammals of Europe. And this is what we need to communicate. And the world's eye on us to not to let this species go. And at the international level, I think IUCN will reassess the endangerment of this species. About impact and communication, yes, I mentioned you that this is the year of the birch mouse in Hungary. We had some events. We published some popularizing articles, and one has made onto the cover of one of these weekly popularizing science magazine. And finally, yes, of course, we would like to publish this reference genome, although some annotation must be done before that. We would also like to publish the RATSEC data, and further, we are committed to start collecting high-density SNPs using genome skimming or resequencing data. Yes, there will be some conferences. So these results, of course, these are not just my results. I'm working together with my PhD student, Sándor Jordan and Virág Nyíri. You have heard Virág before. And we are also collaborating with fine researchers from National History Museum and the Bükk National Park. So thank you very much for your attention.
Thanks a lot, Gabor. We have time for one question. We have time for one question from the audience. Is there any online? I can maybe ask one question. I'm curious about the IUCN. So you said that, of course, they have to reassess. Are you planning to actually show them the power of indeed genetics, and that it's only thanks to your work that, you know, they should indeed reconsider the status?
Honestly saying, we were not thinking about the genomic results. We were thinking of talking about the steep population declines, so losing so many populations within very limited time. And I think that qualifies for critical endangerment. But it's a good idea. We must emphasize what we have found with the genomic results. It will be a nice opportunity.
Yeah. Perfect. Okay, thanks a lot.
Thank you.
The microphone. Okay, and now we move on to another case study about not just one species, but 12 species. Very ambitious. Come in. The floor is yours.
Yeah, it's there. Can the people in the back hear me properly? Awesome. So, first of all, thank you so much for the organizers to invite us and also bring together such a great group of people. I'm very excited to talk about our case study, in which we generated annotations for or reference genome for 12 alpine butterfly species of the genus Arabia. And the context, why are we interested in alpine species, and in particular butterflies? Because let's imagine this hypothetical species that has this green area of distribution in a changing climate with temperature rises and habitat loss. The scenario that we can expect is that species will try to go up to colder temperatures, leading potentially to a smaller and more isolated population, loss of genetic diversity and connectivity between populations. But also, it's possible that some species might be able to adapt and to cope to these particular challenges, maintaining or even expanding their distribution ranges. And it's very important to know if our species of interest are in the first or second scenario to be able to make informed conservation priorities. So the question that we asked is really about adaptive potential. Are our species able to adapt to climate change? And for that, we asked, is genetic diversity in functional genes lower for threatened and high elevation species compared to more widespread and common low elevation species? And more particularly, do high elevation species retain genetic diversity directly at the genes involved in cold adaptations? Those are, of course, very big goals. I'm not pretending we managed to answer those questions, but the very first step to get there is not only to have a high quality reference genome, but also high quality genome annotations. And not going to spend too long on the data, but we had already chromosome level reference genome for 12 species with also the 6 chromosomes assembled. And we chose those species because, well, we can compare some high elevation restricted endangered species to some others that are more low elevation, common, widespread. But also, and I'm highlighting it here, one of the reasons that we're very interested in Arabia is that they have huge variation in chromosome numbers. In this data set, we have species between 8 and 51 chromosome pairs. Coming back to that later. So very standard assembly pipeline. We have hi-fi reads. We used Hi-C for scaffolding. And then and that's where we are very grateful for this case study, because it allowed us to have long reads, RNA-seq, so PacBio iso-seq data for the annotation. Not gonna bore you too long with the assembly statistics, but just to show you that we have very good genome assemblies. We're very fortunate to work with species that are super easy to assemble. But yeah, what I want to talk a little bit longer about is genome annotation. And so we had the expected number of about 13,000 genes per species for a number of transcript of 16,000 per species. But so we were able to compare the quality of our annotation with and without RNA-seq using also the breaker framework in those two cases. So without RNA-seq just means that we used hints from online protein databases. And interestingly, here's the annotation BUSCO score. Seems a bit lower with RNA-seq than without, which only puzzled us for a single second, because when you look at the number of annotated genes, it all makes a lot of sense. That with RNA-seq, you have this consistent expected number of genes, and without, you actually have an aberrant, super high number of annotated genes. So this led to the conclusion that at least for our system and using a breaker for annotation, RNA-seq is necessary to avoid false positive gene prediction. So for the results, there's really just one thing that I could not resist to show you, and I've mentioned the huge karyotypic diversity in Arabia, and this is what it looks like when you do a synteny analysis. So it's a beautiful mess, isn't it? Basically, if you're not familiar with those kind of plots, looking at the color, you can follow how ancestral chromosomes have separated and fused and separated again along the phylogeny of Arabia. And this little black star represents the tenaros clade, which is the youngest, most species-rich and karyotypically diverse clade in Arabia. And those species are barely 2 million something years old, well, divergent from each other. And so, well, this, first of all, I'm showing this to you because I could not resist showing you this plot, but it's also very important for conservation. And I think Alessio made the point very clear yesterday that if there is intraspecific variation in chromosome number, probably those should be considered as different management units. But that's an entire different story. Let's go back to our actual conservation question. And here I'm showing you Arabia Pluto. Let's say it's a case study within the case study, one of our 12 species, which is a particularly high elevation species, particularly cold adapted. In the Alps, you can find it up to 3,200, 400 meters in elevation. And what's interesting about that species is that it has a huge morphological variation in the different mountains that it inhabits, leading to the hypothesis that maybe it's actually a species complex, which of course is important for conservation, because if those are multiple species, they are probably much more endangered than they would be considered if they were a single species. And that's, of course, very timely because we're in the middle of the revision of the Swiss Red List of Butterflies. And that's a document that's produced by the Federal Office for the Environment. And they have asked our friends and colleagues at InfoFauna, who are the people that are doing the long-term monitoring in the field of butterflies, among other populations directly. And that's where we come in, because they've actually asked Kai to help them with the genomics part of the evidence to provide to this red list, including genomic analysis of Arabia Pluto. This is of high relevance for policy, because this is the Swiss Red List of Butterflies is actually a legally binding document that the cantons are going to use to implement regional policies for conservation. And also, hopefully at a larger scale, these country-specific red lists might be taken into account and considered to inform the more global IUCN red list. So just want to say a little bit about how we try to spread the word. And I was very lucky to be invited to present this locally to the entomology community at the Natural History Museum in Neuchâtel and at the Swiss Entomological Society meeting, where the main message that I tried to convey was the importance of having reference genome and reference annotation for understanding and protecting biodiversity. Now, scaling up a bit, I think it's important for Impact to mention here Project Psyche, in which we're very involved. We're the Swiss collection hub, because Project Psyche is this European project that's trying to generate reference genome for all Lepidoptera in Europe in the coming years. And we've just reached a super big milestone together with the genomes previously produced by the Darwin Tree of Life and the Origa community. We've now reached 1,000 reference genome for European Lepidoptera, which is huge for conservation, but also for bioeconomics. There's a lot of endangered species, pest species, and so on. And the goal is, of course, to reach 10,000 in the coming years. I don't think I need to convince anyone in this audience that those huge collaborative projects are beautiful, because they're bringing together a community of people with common interests. All those data is open access for the community to use. And just mentioning also particularly Genome Notes, is a little publication announcing a new reference genome, because they're allowing us to acknowledge all the people that have played a role in generating those genomes, the people that did the sampling, the sequencing, the curation, and so on. And well, I know I've talked about a lot of things today, but just to go back to the first questions that we asked, what I think might be a future for conservation genetics is to look directly at adaptation potential by finding the genes involved in response to current threats of our species of interest, for example, here climate change, and then assessing the adaptive potential by looking at the functional diversity directly at those genes. And hopefully maybe someday use that for making informed conservation priorities. These are very, ambitious futuristic goals because that comes with a lot of challenges. Well, first of all, functional annotation in non-model species is still very challenging. And well, it also needs to make this link between genetic diversity, ecology, phenotype, what's adaptive, what's deleterious. I mean, we don't even know what are the genes involved in cold adaptation yet. So, but yeah, my main point is to give you a little bit of food for thoughts, also maybe for your system, if that can be applied. And of course, I want to thank everybody that has helped us since the beginning of that project. And here is just a bunch of logos, but really who I'm thanking are all the people behind, without whom it would not have been possible. And of course, thank you all for your attention today.
Thanks a lot, Camille. We have time for one question. Yeah, Enrico.
Thank you for the talk, really nice. I have actually a technical question about breaker-free because, it's struggling to work with breaker-free, no? And so for RNA sequencing, you produce just one sample for each genome, more than one?
A single one, but we made sure that it came from the same population as the individual for which we build a reference genome, to at least make it as close as possible. Unfortunately, we don't have like different life stages. We merged a bunch of tissues, but yeah.
And did you change any parameters?
We had long read RNA-seq, right? And there's a specific new breaker version for this case in particular that we used.
Okay, thank you.
No worries.
Perfect, then we can move on to the next one. Yeah, thanks.
Are there questions online?
No, not for now, but I would say monitor it, and they might come later.
Thank you.
Okay, so our next presenter is... siete in due. Oh, sorry, I switched to Italian. Sorry. Two people. Okay, so Enrico and Fabrizio are gonna talk about... Oh, yeah. So we have Fabrizio and Enrico that are gonna talk about the wood ant.
Okay, so good morning. Thanks everyone for being here. And so I'm going to talk about Formica paralugubris. First of all, a little bit of context. So paralugubris is a native to, yes, of course, native to a small area in the Italian Alps, also a small population in Switzerland, the Jura canton. And is the smallest range among the redwood ants, so the Formica rufa complex. And also another interesting thing is that Formica paralugubris was introduced across all the Apennines and the major islands, and also Germany and Quebec, even Quebec, by a professor of the Pavia University named Mario Pavan in the 1950s, because he wanted to use the species to control conifer pests. And so he transplanted this ant everywhere. So the project started in 2023. It's a collaboration between 3 universities and the museum, Firenze, Bologna, Pavia, the university and the museum. And so we started sequencing the first primary assembly. We got the first primary assembly, and then we submitted for the BGE case studies in order to bring the assembly to ERGA quality, high quality like assembly. And basically the problem with this species, the main problem with this species is very simple. The solution is not as simple. So this is a keystone species, but it's facing population decline. So this is the problem. And there is no consistent legal protection because there are legislative gaps. And so this is the main, more on this later. So the main goal was to get a high quality reference genome, and also we can do, thanks to wide sampling, we can do a lot of population genomics, which is ongoing. I'm not showing diversity data, unfortunately, because we are late, and I will explain why we've been late. But basically, we have all the population in the Apennines, in the Alps. We got this year also something from Sardinia. And also we have museum samples, so we can add also the temporal dimension to the analysis. And basically, our data will contribute to both to conservation efforts for the declining populations, and also we want to study the consequences of introductions in the places where the species was introduced. So this is the pipeline, the assembly pipeline. I'm not talking about that. It's a pretty straightforward and normal pipeline. What I want to focus is the sampling, because this is the only big problem we had in this project. Because in order to get high quality DNA and high quality DNA, we need to use pupae. And those are available only for 3-4 weeks in the whole year. And unfortunately, in the Emilia-Romagna region, we had like natural disasters for a couple of years, like floodings and landslides and some biblical like plagues. And so we couldn't really sample most of the time. And so that's why we are a little bit late. We got the IC data like a few weeks ago. So just in time. And so we used a single pupa for the PacBio I5 reads for the primary assembly, and also we used pupa for IC, and then we're going for isoseq as well, which is ongoing, so no annotation as well. Sorry about that. And okay, so the genome is turning out quite good. Especially, I come from bivalves, so that's a dream for me. It's a beautiful genome, and the size is about a little bit more than 500 megabases, which is pretty big for an ant, because of course. And then the repetitive content is almost 70%, which is quite a lot for an ant, because of course. And but we are managing to get, like, we have 33 scaffold longer than 1 kb, and we expect 29 chromosomes. So we are doing the manual curation right now, and we are also getting the karyotype. So we will make Alessio proud. So, and the Busco stats are pretty good, 98.7 complete genes. So it's very good stats for an invertebrate. And also one interesting thing, we did some Felgen densitometry. So we are also getting estimate of the genome size in vitro. And the good news is that the Apennine population has a genome size which is compatible with the bioinformatic estimate. And the interesting thing is that the Alpine population has a bigger genome than the Apennine population. So this is interesting, and we are going to investigate that more in detail. With more analysis, like also densitometry in around all the range, all the populations. And okay, what we are going to do with this data? So first, most important thing, we want a new UCN assessment for this species. We have two collaborators, Giacomo Santini and Alberto Mazzoni from the University of Florence, which are working on this species for a long time. And we are going to use this new data. These guys are assessor for the UCN and species group. So they will use their data and this genomic data to try to get the species to the threatened category. And also there is an important thing, which is the transplanted population are doing good. Despite, I mean, they're doing good, the Alpine population are not doing good. And so the transplanted population have a huge genetic and physical reservoir. And so basically we would like to use the genome, the reference genome, for monitoring campaigns, for control and conservation, to do targeted genetic studies, to see the difference between native and transplanted populations. We are doing also comparative genomics. We are getting 2 more, like an Alpine reference genome, high quality reference genome. Also, we are sequencing the parental species. This is an hybrid, by the way. And also, we want to do comparative studies to find about the adaptive capacity, which we heard about in the previous presentation, luckily enough. And so I don't have to talk about this. And also, we want to develop simple genetic markers that parks and regional bodies and institutions can use to assess diversity. And also, we would like to estimate the population status, especially in the Alps, because we have fragmentation, habitat fragmentation problems for anthropic like actions, like ski slopes, and stuff like that. And also, bark beetles are a problem. And now I call Enrico is going to talk about the stakeholders and the amazing job they did for dissemination.
Thank you. So those are just a few of the stakeholders that we got in touch with during our year of experience. So first of all, we started from high schools. We offer them didactic labs and lessons focusing primarily on myrmecology and on genomics. And that helped us a lot because they spread our name, the name of our project locally. So since we're working with the Foreste Casentinesi National Park, which is one of the few places on the Apennines where the population of Formica paralugubris still flourish, we're working with them on our citizen science app. We decided to introduce to our lessons some labs about citizen science, which we co-designed with the Emilia-Romagna region digital agenda, which has our citizen science education repository. And we presented the results to the Citizen Science Italia National Conference, which was held in Bologna last April. I'll stop saying citizen science right now. And so for what regards communication, we participated to a few workshops, the most important which is Foro di Scienza, which was held a couple of weeks ago, and that's the most important convention for science communicators now. So we had the opportunity to exchange contacts and opinions with some of the most important actors in the science communication field in Italy. And we also offered them a workshop about myrmecology and genomics, which was open both to the general public and to the science communicators. So the premise for policy relevance is that neither of us is an expert in this field, which is followed by the University of Florence, but the general issue for redwood ants as a complex in general is the lack of an European protection framework. So the protection is demanded to on a national level, on the single countries, but most importantly on single regions, which is Italy's case. Those laws follow, unfortunately, an outdated taxonomy and do not take in consideration many of the new risks these animals face, such as climate change. So we would like our work to be set as a trailblazer to introduce genomic data in a new legislation, in a pan-European cohesive framework for the protection of these keystone species. And also, it's interesting, the fact that it could be an incredible rewilding opportunity, because right now, the few projects of rewilding regarding redwood ants are just on an ecological scale, so they do not consider at all genomics. So it would be interesting to add this, this part to those projects. So for the impact and communication, we rely primarily on live events and live lessons and workshops. So used our social media platforms and website more as a repository, let's say. So I don't have enough time to talk about the more than 50 hours courses that we got in our schools that are going to double in 2026, or all the events we participated in. But they are, you'll find everything you need to know if you want to know at the QR code at the end of the presentation. I want to stress out the fact that we're working on a documentary that talks about the fascinating story of the introduction of these redwood ants and the history of the scientists who thought it was a good idea to bring them to Canada, which was not. But but it's a very long story. But I'll be glad to tell to anyone who's brave enough to ask me anything about it during our coffee breaks. So finally, something very important for our dissemination campaign was wildlife photography. So together with the extremely talented photographer Jacopo Nerozzi, we produced the first 2 sets completely dedicated to Formica paralugubris. And this helped us loads because it gave us incredibly quality material that we could use in presentation, as you could see, during lessons and during live events, and helped the general public to immediately immerse themselves into the microworld of our ants, which was crucial since we wanted to guide them into an even smaller realm, which is clearly the genomic one. Also, we got published in a book because one of our shots was prized by the Wildlife Photography Award with an honorable mention, and they produced a catalogue, and they quoted us and explained a bit what our project was. So I would recommend to anyone who's trying to get better with dissemination to invest in wildlife photography, and most importantly, on wildlife photographers. So if you want to...
Yeah, just to conclude, thanks, Enrico. If you want to learn more, this is the QR code. Next, we are very excited about this genome and all the data we are gathering. And so we are working to the very final assembly and annotation. We are working on the PopGen, more schools and labs and science communication coming, a lot of it. Again, comparative genomics, we're sequencing more genomes, high quality, and then the UCN assessment is really important for us. The documentary, I can't wait to see that. And also, we really would like to create a European framework for redwood ants, a cohesive unit, and of course, which is also the problem or the threat, whatever, fundings. We need money, of course. Okay, so I want to thank a lot SIB and DG, because we also, not only for the support, but also because we learned a lot about how things have to be done. And this is what ERGA is about, which is the best community ever, for my opinion. It's not like that. And yeah, so that we learned a lot, and we try to pass this on to students and other colleagues, which is difficult. With the colleagues, I mean, students are super receptive. And so, yeah, that's thank you very much.
Thanks a lot, Fabrizio and Enrico. Do we have time for one question from the audience? Of course, the arthropod person.
Oh, so about the biology, what is the colony size and what is the colony range?
Okay, so the size is huge. These are highly polygenic species, so sometimes there is not just one nest; it's a network of nests, and so it's really huge. And so we expect quite a high diversity in the same nest, but not so much between nests, because it's something that is really widespread. And about the number of individuals,
for colony, yeah, it really depends, but generally they are obligated super colonialists. And so they range from some hundreds of thousands to tens of millions. So, yeah.
Well, thanks a lot for the presentations. Move on to the next one, which is from Silvia. Yes. Hello. Yes.
So, good afternoon, everyone. My name is Silvia Garcia Juan, and I'm a PhD student at the Evolutionary Genomics and Bioinformatics group at University of Barcelona. Today, I'll be presenting a recent work about 2 new reference spider genomes to assess phenotypic convergence and adaptation in coastal habitats threatened by anthropogenic pressures. The diversification of the different species of spiders of the genus Dysdera is one of the most spectacular examples of a species radiation on an oceanic archipelago. Out of the approximately 300 species in the Mediterranean region, around 60 of them are endemic to the Canary Islands, which suppose 20% of the total. This makes the Canary Islands an ideal system for studying how these species diversify and adapt in isolated environments. 2 of these species are Dysdera curviceta in Tenerife and Dysdera mahan in Lanzarote, and they colonize intertidal pebble beach environments. This is an ecological niche that is very unique from this genus because it has only been found in the Canary Islands and in Madeira. All the rest of species from this genus are either terrestrial or cave spiders. And this colonization happened independently because, as you can see here in this phylogenetic tree, they belong to different lineages. So this makes that these 2 species are the perfect candidate for studying adaptation to environmental stress in these conditions and at the genomic level. But these coastal environments are one of the most vulnerable ones across the archipelago because they have suffered from urban expansion, tourism pressure, and also rising sea levels. As you can see in this map, this environment has suffered major changes, and this is mainly due to the intensive tourism that began in the 1970s and that continued to this day. So understanding how this adaptation occurred is crucial for predicting the resilience of these species and also to inform conservation strategies of these coastal environments. The BGEK's main objective is to generate a high-quality chromosome-level reference genome, in this case for Dysdera curviceta. But this objective is marked in a larger project that we have in our group, where Dysdera mahan reference genome was also generated. So in this larger project, our 2 key questions are about what genomic mechanisms enable adaptation to intertidal environments, and if curviceta and mahan show convergent genomic signatures. Our hypothesis is that independent intertidal lineages evolve similar genomic solutions to cope with saline stress, desiccation, and fluctuation conditions, among others. And to test this hypothesis, we are performing a comparative framework across 2 islands and lineages. To obtain the data, we perform the sampling of the individuals alive. As they are nocturnal, we only have to go and try to picking them up from under the rocks. And when we can find them, it's not too difficult because they are like not in the best part of the day. So after this, we did flow cytometry for estimating the genome size of the species. The rest of the individuals were transported alive and then flash frozen in liquid nitrogen, and then preserved at -80 degrees to ensure the optimal preservation of the DNA. Then we conducted the extraction of the high molecular weight DNA and also the RNA with Qiagen kits. And for the sequencing, we decided PacBio Hi-Fi together with the ARIMA Hi-C in Novogene, and the sequencing of the Illumina RNA was done by Cenad. Moving to the assembly pipeline, the steps we did were, first of all, the Hi-Fi assembly for the haplotype resolve assemblies, followed by the Hub Hi-C for scaffolding and Juicebox for the manual curation. After this, we assessed the quality metrics with Busco for assessing the completeness of the assembly and Quast for the contiguity. And then we moved to the genome annotation. Firstly, before this genome annotation, of course, we mask the repetitive content of the genome, and then we could annotate using proteins and RNA evidence. And after this, we also check the boost code for the completeness of the annotation. And finally, we could do the functional annotation with homology-based approaches. So the results of this assembly and annotation are, for the flow cytometry, we estimated a genome size of 1.5 gigabases. And the assemblies that we got was one for each of the haplotypes. So they are high quality chromosome level assemblies. And we could confirm that our individual was a male with a XO sex determination system, which is very typical for this genus of spiders. We can see that we have 6 autosomes and 1 large X chromosome. Then for the assembly quality, we found that it has a high contiguity with an N50 of more than 200 base pairs and L50 of 3. And the length of the haplotypes matches with the estimation of the flow cytometry. And for the annotation quality, the BOOSCO results were quite high also. And in the case of the haplotype 1, we can see that it's lower, but it's because we are lacking that X chromosome that is quite large in the case of this species. Then for the preliminary comparative results, we have seen that the heterozygosity of these 2 species is lower compared to the other species from the genus that are in these islands. So this could constrain some future adaptive responses to the change of the environment. And regarding the sampling size, 2 of both species are found only in a few locations. And in the case of this Deracorviceta, we had to go sampling several times because we were very unsuccessful the first times. But however, we could find 2 new sampling sites. So we think that if the sampling effort continues, and for that money is needed, we could find more sampling sites. And in Mahan also there are few sites because Lanzarote and Fuerteventura were some 1,000 years ago one single island. So it's not something rare that we can find them in both places. And also it's found in some small islands around. And this project has also a strong stakeholders and engagement content. First of all, we are assessing the risk status of different species from the genus, and this Deracorviceta and Mahan are included in this assessment from the Atlantic Island Invertebrates Specialist Group. And we are also doing collaboration with the Canary Islands government to see if these species could be coastal umbrella species, so other species that live in these environments are also affected by this protection that we will give to this one. But this assessment focuses on species distribution, habitat, and potential threats. So we would like to incorporate this genomic information in future assessments. Also, all the information that will be released from this project will be transferred to the different databases, like Biota, that is a database belonging to Bioscan, that is a larger inventory project, and also to GEVIF in the DESUL collection, that is a collection from the University of La Laguna in Tenerife. And also will be transferred, like all these case studies, to the ENA database. Finally, we are in relationship with several local conservation actors, like NGOs, like for example, this one, Asociación para la Conservación de la Biodiversidad Canaria, which has a key role in transferring the scientific information to the social community. And these relationships with the different actors help us to make this information available to the broader public, and also to raise awareness of the society about the importance of the conservation of these environments. When the ICUN evaluation will be finished, we would like to move to the Canary Islands government evaluation to include these species in the Canary Catalog of Protected Species that is okay, so these catalogs comes from this law for 2010, and this will be very important to actually protect this environment, but it is quite difficult because there are other economical interests involved. But we think that the genomic information can make this proposal stronger and more robust. And finally, this project is enmarked in the EU Biodiversity Strategy for 2030 for using genomic information together to the ecological one for the policy planning. It's not working? The scientific outputs of this project will be mainly the one chromosome level haplotype result genome assembly, the one reference genome with a structural and functional annotation, and in the future, a comparative genomics paper that will be, of course, open access. So this project has also been actively communicated across different media. For example, this report for the IUCN specialist group, also disseminated in different press from University of Barcelona, University from La Laguna, and in different conferences. We also share in social media all the process of the sampling campaign and also the procedure in the lab. And finally, we are waiting for a report in the Asociación para la Conservación de la Biodiversidad Canaria to spread this message to the social community. The next steps in the short term are conducting the comparative genomic analysis and also obtaining the results of risk status. And in the midterm, we could turn to other coastal disturbed species because we know about a new coastal species in La Palma. But we have some risk here. First of all, the habitat loss could outpace the policy implementation and be lost. And also that we could have limited funding for possible long-term genomic monitoring of this species. And our proposal is to integrate genomics indicator into coastal management plans and also to secure co-funding with networks like this BGE. And I would like to acknowledge all the partner institutions, and of course, I will be happy to take any questions.
Okay. Thanks a lot, Silvia. We have time for one question from the audience. Okay.
Oh, thanks. It's like a very simple one. How do you imagine that this genomic monitoring could happen?
Yes, the idea of a possible future genomic monitoring could be in partnership with the Universidad de La Laguna, that they can have different experts there also for the identification of the different species. And the idea could be or taking different measures a long time, or also moving to the population genomic analysis to understanding the demography of the species and also how this can be changed through time.
Thanks, Celia. Then we move on to the next one. So the next one is Sophia. And she's going to talk about hairs.
So welcome everyone. I'm the second person today who is going to talk about hairs. And I've been doing this work together with Jacopo Hioismati and also with the Portuguese colleagues here at the CBO. So our problem is that due to the climate change, there is a growing pressure on the mountain hare, and as its range is contracting, while the brown hare is spreading towards the north rather rapidly. In recent years, it has gotten so far that at this point, we have brown hares observed north of the Arctic Circle. And since the divergence of these two species is relatively recent, there is active hybridization going on, and there is fertile offspring possible between these species. So what we want to do here is to assess the scope and scale of this hybridization and its influence on the adaptive success of the brown hare. Here we have a map showing in blue all of our Lepus timidus sampling location, and in red all of the Lepus europaeus, the brown hare sampling locations. And we are trying to see what does the hybrid zone look like, if there is any increased genomic introgression in some genomic regions, any positive selection that we can detect in the brown hares in Finland, and if any of these findings, this introgression, this selection, can play a role in the cold adaptation of the brown hares. So for this, we have generated two reference genomes. Both of them are chromosome level assemblies, and both of them have 23 autosomes plus X and Y chromosomes. And we also have ddRAD sequencing of over 300 individuals. Huge thanks for that to our colleagues at the CBO who did all of this sequencing. And about half and half of these are brown hares and mountain hares, respectively. I will not go too deeply into the workflow. If anyone wants to know about it, I'm available, but we don't have that much time to talk about it. So about the population structure, the first thing I noticed was on our PCA plots that there was a definite genetic gradient in the brown hares towards the north. And there came the idea that I would check the FST between all the subpopulations that we have. And the brown hares are in the lower right corner. And you can also see there that that bigger, darker blue block also shows some substructure, which are the Finnish brown hares, some southern and some northern groups of them. We also checked the hybrid index and intergross heterozygosity, which is shown on that triangle plot there. And you can see that in the brown hares, the lighter purple population are the Finnish ones, which show a bit of hybridization, like overall, with the mountain hares. So this seems pretty widespread. And also in here, if you compare the admixture bars with the map, you can see that towards the north, in the brown hares, we definitely have more admixture with the mountain hares. And on the other hand, we have on the right the mountain hares, which show definitely lower levels of introgression from the brown hares than the other way around. And also, which is not very obvious from this one, but in the mountain hares, we have a lot more uniform distribution, and they seem to move a lot more between Finland and Siberia, and are a lot less structured than the brown hares between Europe and Finland, and even within Finland. So the main direction of introgression is from the Timidus towards the Europeus, and the mountain hare towards the brown hare. Sorry for sometimes switching the names. But we didn't see any introgression on the X or Y chromosomes, which is, I will get to that later, and also more introgression towards the north. So about the lack of introgression on the X and Y chromosomes, we also have a lot less data points on the X and Y chromosomes, which is, I think, a limitation of our DDRA data, that if there is no restriction site there, then there will be no data. And of course, the restriction sites can be different between the species. And if it's missing data in one species, then we can't compare it. But also, in here, I marked with orange the most often introgressed genetic locations in the brown hares, while also illustrating the levels of genetic differentiation between the Western European brown hares and the Finnish brown hares. And I actually found some quite interestingly coinciding genomic regions where there is a really large genetic difference between the Western European and Finnish brown hares, and also quite frequent introgression. We have, by the way, about 100, bit more than 100 Finnish brown hair samples, and over 30 of those was at some level introgressed. So that's quite a lot of hybridization there. And so overall, our Finnish mountain hares retain quite broad genetic variation and with little geographic differentiation. And the Eastern Finnish mountain hares also carry ancestry from the Russian Far East. The gene flow is, as I mentioned, mainly from the mountain hare towards the brown hare, with a lack of introgression on the sex chromosomes. And regarding hybridization, it results in a lower reproductive output for the mountain hares, since the back crosses usually go towards the brown hare. And it also, so it's an increased stress on the mountain hare populations, while it's a potential source of adaptive variation for the brown hares. So in this way, it's there is dual threat for the mountain hare, both for their habitat contracting due to the climate change, and also the hybridization isn't favoring them. And as the brown hare is expanding, they are pushed back north, I mean the mountain hares. So so basically this is the current standing of what we know. And our stakeholders here are the National Resources Institute in Finland, the Finnish Food Authority, which surveys wild animal diseases, and also obviously the Finnish Hunters Association, since these are both game animals. And we are planning to be involved in the national hunting legislation and some species-specific management under the game animal degree. There should be some regional game management plans that would be coordinated with the Finnish Wildlife Agency. And also there is the potential inclusion in the EU biodiversity Strategy Monitoring and Habitats Directive, which is reporting on species of northern distribution affected by climate change. We have produced some social media engagement, blog posts, LinkedIn posts. We have presented our project on the Finnish Molecular Ecology Conference last year. And also there is media coverage on our genome publication. There are popular science articles which were shown here actually. And we have a manuscript in preparation and 2 major grants coming up for continuation projects. So the next steps are obviously the manuscript submission, starting up the continuation projects, and being involved in discussions on hunting regulations for the mountain hares. And in a somewhat longer range, we are planning to work with stakeholders on national mountain hare conservation strategy, trying to increase public awareness through popular articles and promoting mountain hare as a sentinel species for climate change. And yep, that was the last one. So thank you everyone for your attention.
Thank you, Sophia. We have time for one question. Okay, I already see one there from Elena.
Will be short. I know that there is a historical introgression on mitochondrial DNA between the brown hare and mountain hare, but I'm wondering, how do you distinguish between this recent and historical introgression? I haven't seen this from your presentation. And if this is also seen on the nuclear genome or only mitogenome?
So for the historical introgressions and the recent introgressions, when I was checking the hybridization, the hybrid index, so here I was only considering SNPs that were completely differentiated between the 2 species. So any historical or introgression was pretty much excluded there, I would think, because for defining these SNPs, I took the 2 parental populations, Western European and the Russian populations, and only considered the SNPs that were completely differentiated between these 2. So any ancient contact that would cause shared SNPs was pretty much outruled for this one.
Thank you very much. Perfect. So the last presentation of this morning session is the one from Jean-Baptiste on corals.
Thank you, Pierre. So yeah, I'm Jean-Baptiste Ledoux. I'm working at CIMA, which is half an hour from here, close to the sea. And I will talk about the conservation genomics of the Mediterranean red coral, Corallium rubrum, species I'm working on since a few years now. So Corallium rubrum is an octocoral with a highly fragmented distribution from the Mediterranean Sea and also from the neighboring Atlantic, including the southern coast of Portugal. It is found from 5 to 1,000 meter depths. As you can see on this picture, this is an habitat-forming species, which increases the habitat complexity. And the species has long attracted scientific interest with, for instance, the life cycle was described in the 19th century. And sadly, or not, it not only attracted the scientific interest, but also the economic interest. Indeed, the species is used for jewelry and handcraft, as you can see here. So it is submitted to a strong fishing and even poaching pressure. You can see here the strong decrease of annual yield of red coral fisheries between the 1970 until 2011. In addition to that, a second pressure is the direct impact of extreme climatic events in the name of marine heat waves, which are triggering mass mortality events in the whole Mediterranean in shallow population until 40-meter depths. And this is so strong that this can reach to the total extirpation of the populations. So no need to say that this species is a highly relevant model for conservation biology, adaptation or not to climate change, and also biodiversity management. So thanks to a long-term demographic survey, we have a relatively good view of the impact of these two pressures on the demography of the species with strong demographic declines. But what are the impacts of these two pressures on the species genetic makeup is still an open question. And in addition to that, crucial data regarding, for instance, admixture among lineages, demographic history, or effective population size are still lacking. While these data are critical when we talk about the implementation of effective conservation policies. So this is basically the very general framework of this. Oops. What happened? Yeah. It's the general framework of this case study. And the case study was designed around two main questions, or two main objectives. The first one was to set up the evolutionary stage of the species working or trying to work at the scale of the whole Mediterranean. And the second one, we focused on this particular area in the Catalan Sea, where you can find two marine protected areas, and we wanted to use this case study to show how biodiversity genomics can be used to improve or at least to help for the management of marine protected areas. I will come back to this later, but we interact with two different kinds of stakeholders. For the first objective, we interacted a lot with the General Fishery Commission for the Mediterranean, while we interacted with the biodiversity manager of the two marine protected areas for the second objectives. Okay, so regarding the sampling, two different methods. Thanks to the interaction with the GFCM, we obtained a sample from the partner of the commission, which was very nice because we were able to cover large parts of the Mediterranean Sea, but also a bit challenging because we had very different qualities of samples. And for the second objective, we, thanks to the support of the project, we organized a sampling field trip in June 2024. So we ended with a bit more than tissue biobank with a bit more than 2,050 samples from around 30 different locations in the Mediterranean Sea. From this sample, we extract all of them, and we selected 220 samples that were sequencing for whole genome resequencing at high coverage. So this project came in complement with a project that was started a bit before inside the Catalan Initiative for the Earth Biogenome Project, which help us to sequence the reference genome of the red coral. This was done in collaboration with people from CENAC, Taylor Alioto and Jessica Gomez Garrido, and with people from Rosa Fernandez lab at the Institute of Evolutionary Biology in Barcelona. So we used an hybrid sequencing strategy, combining long read, short read, RNA-seq, and IC for scaffolding. And we published at the beginning of the year chromosome level reference genome in the species. You have here some parameter with 20 chromosomes and relatively good metrics for an octocoral. And I have to mention that this was one of the first chromosome level in this taxonomic group. Okay, so we have the data set, we have the reference genome. We went through a usual pipeline for mapping, SNP calling, and filtering, and this is only preliminary results. So, but I will show you some of the results we obtained with based on 1 million, a bit more than 1 million SNP in 136 individuals coming from 20 locations in 12 different regions of the Mediterranean. So here you have the PCA, and the first thing is that we have some structure in the data set, which was expected, and there is some sort of gradient from the Greece to the western part of the Mediterranean Sea on the first axis, and on the second axis we have here a clear separation with the population from the north of the Mediterranean with population from Tunisia and Portugal and Gibraltar here in light blue. The result of the clustering of the admixture analysis confirmed the PCA with 5 to 6 different genetic clusters, with some cluster like the Greece and Adriatic one very well, totally isolated from the rest of the species distribution. And to end on this first preliminary result, just a graph showing you the level of genetic diversity. And here it's quite nice because we have this increase from Portugal, Gibraltar to the northwestern Mediterranean, which then decrease again when you take the coast of Italy to end in the Greece and Adriatic. So quite interesting results, which, yeah, we have a lot of work to do now. I guess this is only the beginning. But regarding the implication of this result for the management, I guess that with such a high level of population structure, the management can not only be done at the scale of the whole Mediterranean. There is a need to define some regional policies. And to come back to the fishery topic, this needs to be clearly formally tested, but I guess with such different level of genetic diversity, we can think that there is some variation also in the effective population size and so in the fishery stocks. So once again, this remains to be formally tested. Okay, so stakeholder engagement, as I mentioned, we had a very good interaction with people from the General Fisheries Commission for the Mediterranean, and more particularly with their research program on the red coral. And here there is Federica, who is also part of this program. So this group of people is composed with GFCM and FAO official, NGO, scientists, and more interestingly, with people representative of the JWL industry and the fishermen. So this is always very good to have all these people in the same room. And from this particular project, it helped us to obtain the sample, as I previously mentioned. From our side, we give them the reference genome because they are conducting a rat-seq analysis with even more samples, something like 700 or 800 different samples. So they use the reference genome to map their rat dossier. And we are now in a step where we are starting to discuss the results and try to see how we can translate those results into conservation and management policies. Regarding the two MPA, it was mostly The interaction was mostly done when to obtain the sampling permits, but we are now also in discussion with people from the University of Barcelona, where because they are conducting a monitoring program on this demographic monitoring program on this population. And I think that we can find some way to interact, to combine the genomic data on connectivity and potentially local adaptation we'll obtain with their data on the demographic monitoring. And yeah, for the future, the GFCM is highly interested in the development of a kit tool for traceability of red coral colonies. So I guess with all the genomic resources we now have, we are in a good position to start working on this. Impact in communication. So as mentioned, there was this paper that received a good reception in the scientific community. It was used the genomic resources, the genomic reference genome was used in this beautiful paper by people from the Scripps institution in California. There was a lot of noise because when you talk about corals, and so these are good keywords for the press, and so a lot of engagement towards the general public. Also thanks to the communication office of CIMA, who did really a great job on this. So I did more of 10 interviews, TV, press, internet, radio. It was sometimes good experience. Sometimes it was a bit, well, let's say that some titles were not really intelligent because Portugal will probably not save the coral from the Mediterranean Sea, but at least we are trying to do something. And I also put here the QR code of the video we managed to produce during the project. So if you want to see some beautiful images of sampling, and if you want to have a framework of the project, you can go there. And yet the next step, well, I still have to upload all this data on INE, update the GitHubs, and for longer term, paper submission for sure. And we are really excited by this traceability toolkit. So yeah, hopefully we will also have fun to do this. And that's it. Thank you. Clearly, this is a huge teamwork. So yeah, I have a lot of colleagues to analyze for this. Thank you.
Thanks a lot, Jean-Baptiste. We have time for one question. Oh God, there are too many now. Jacob, go ahead.
Maybe we have something too.
Great talk and fascinating. I was wondering, corals have these mutualistic dinoflagellates. Were you able to separate those and assemble?
So this one is aposymbiotic, so there is no dinoflagellate.
So I wasted my question.
So we take another one. This one was short.
Until 1,000 meter depth.
And thanks for the talk. I was wondering for your population sampling, if you sampled at different depths and if you could investigate local adaptation among depths at the same site.
So all the samples we had from the GFCM are below 50-meter depths because this is the depth where you can start fishing. And they really want to have an idea of the fishing stock. So all these samples are below this depth. And all the samples we went to sample in the 2 MPAs are above 40-meter depths because we are only usual divers, so we stay. But yeah, we clearly, this is something that we want to check. One thing that is a bit tricky with that coral is that we have a high population structure. So yeah, looking at or identifying the species that are in the local adaptation can be a bit tricky because the FST are always really high. So yeah.
Perfect. Then we can end here the session. Thanks a lot to all the presenters for these amazing presentations. And yeah, another round of applause. Thank you. And we are ready for lunch. If
Yes, just
Giselle doesn't
a
have-
couple of announcements. Yeah. So we will keep the 1 hour lunch break. So that means that we will start at 2:15. So that would be 3:15 CET for those who are on that schedule. Don't forget now, well, lunch in the same place as yesterday, but before going there, please join us outside to take the picture, okay? The group picture. Thank you.
So who will say where we can start? We can start, or who will say? Okay, we can start. I hope you can hear me well, despite I'm so small. Okay, today I have a pleasure to introduce Alexandra Weber and her talk about, she will talk about genomics of sex determination in invasive muscle. Alexandra is evolutionary ecologist, and she's interested in understanding the molecular mechanism generating and maintaining biodiversity in aquatic ecosystem. She used a wide range of omic data from the field and experiment to uncover how organisms adapt to different environmental conditions, and also ultimately infer their vulnerability and resilience in the face of the rapid environmental change. She also focused on native and invasive freshwater beaver species in Switzerland to conduct also her current research, but also to have some collaborative project in other aquatic invertebrates from various habitats. So Alexandra, now floor is yours to hear quite a lot about mussels.
Thanks a lot, Elena, for the kind introduction. Thank you to the organizer for the organization of this really interesting conference and for the opportunity to talk about my research. So as mentioned today, I will talk about genomics of sex determinations in 2 invasive mussels. So I will start with a very quick introduction on these 2 mussels, if some people in the audience are a bit unfamiliar with these mussels. So they are freshwater invaders, very successful. They both arise from the Ponto-Caspian region, so that the regions of the Black Sea, and they colonize, say, continental Europe and also North America within the last, let's say, 30 years. If you're interested about these mussels, you have 2 really good reviews that were relatively recently published that talk about these mussels. And they are invasive, so they have very negative impacts on ecosystems. So basically, since they're mussels, they're filter feeders, and they're very efficient at removing phytoplankton and zooplankton from the ecosystem. So they basically drive really big changes in food webs. But they're also infamous because they cost us a lot of money, and this is why they are so famous. Basically, they grow millions of mussels. You can see beds like this. This is really impressive. And what they do, they damage water infrastructure, and eventually they clog pipes, which leads to millions of costs to replace the infrastructure or just develop infrastructure that are mussel-free. And as with many invasive species, it is currently not possible to eradicate these mussels once they have colonized the environment. And then now what has been developed in the last years is genetic biocontrol that could be a solution to eradicate invasive species in general, not just mussels. And what do I mean by genetic biocontrol? It's about disrupting the reproduction of invasive population by releasing genetically engineered individuals in the wild. So if you are interested in the topic, there is this recently reviewed here, it's difficult with this pointer, that talks about the various techniques of genetic biocontrol. And one of these techniques is called sex ratio distortion, and it is exactly what it sounds like. Basically, you would release males, let's say genetically engineered males in the lab, then you put them in the environment, and then this would lead to increase in male frequency, and eventually, when you only have males in the population, then the population crashes. And there is a really nice example of this with these invasive brook trouts. So genetically engineered YY males are produced in the lab where they have actually 2 Y chromosomes. And when you see that when those YY males reproduce with, let's say, XX female, the progeny is 100% male. So that the increase in male frequency is much faster in the environment. And so there have been modeling studies and also pilot studies that show that this is
could be a really viable solution to drive like populations to invasive population to extinction, and there are ongoing tests in the fields. But as you can see here, this technique relies on the existence of sex chromosomes, so genetic sex determination. And we touched a bit upon that in a few of our presentations, where we saw a bit of chromosomes, but there are actually a large variety of sex-determining systems in eukaryotes. So the most famous ones, the ones that we all know, is our sex-determining system with sex chromosomes, X and Y, with a male determining locus on the Y chromosomes. But we have also the opposite, that was also mentioned in some other case studies, that there are some ZZ, ZW sex chromosomes where there is a female determining gene on the W chromosomes. Then we have things that can get more complicated with polygenic system with different genes on different chromosomes that together act to have a sex phenotype. And this is also something most people have heard about, is environmental sex determination in some reptiles and turtles, where typically temperature can drive to the development of males and females. And to make things even more complicated, we even have some organisms where we have genetic components and environmental component that drive sex determination. And I think you can get to the point where I'm going, that is, we don't know what sex determination mechanisms are in quagga and zebra mussels. So, genomics is actually very useful to uncover potential sex determination mechanisms. I think maybe some of you have resequenced populations and suddenly you find weird patterns and you realize, oh, I found the sex chromosome or sex locus. You can do this with a bit more intention. And this is also a really nice review by Beatriz Picoso that was published in 2019. That shows that depending on the differentiation of sex chromosomes or sex locus, let's say, we will see different patterns. So in, let's say here, and where we have just one sex locus that appeared, but then the rest of the chromosome X and Y is still recombining, we will find differentiation on a very, very small region. So these sex mechanisms are really, really young. So we have an undifferentiated X and Y. And to detect this, the most common thing to do would be to calculate FST between sexes. Then when there is a bit more differentiation between these two, then recombination stops. And then we start seeing that there are differences in male-female heterozygosity, where the heterogametic sex is heterozygous. And then when the chromosomes are very different, when there is a lot of degeneration, we see physical shrinkage of the sex chromosome harboring the sex locus. There we can see differences in male-female coverage here. And this is typically in systems where sex chromosomes are very old. And in those cases, we say, let's say when the X and Y are undifferentiated or very young, we say that chromosomes are homomorphic, so they have the same morphology. And when they are differentiated, when they are morphologically differentiated, we say that they are heteromorphic. And what do we know now? Moving on to bivalves. So they are, it's a big, diverse group of mollusks with over 25,000 species. We have been talking about karyotypes yesterday, and so there are a few karyotypes of bivalves. There are 150 that are published, and what we know is that all of the bivalves that have been studied for that have homomorphic chromosomes, including the zebra mussels. You see the karyotype of zebra mussels there, and we didn't have data for quagga mussels yet. So the hypotheses are either we do have sex chromosomes, but they are young, or we can have polygenic sex determination, or we can have even environmental sex determination, or we can have several factors, genetic and environmental factors. What do we know as well from bivalve sex determination from this very recent study that has just been published this year? In this study, the authors looked at an evolutionary sequence of some gene families that are known to be involved in sex determination in vertebrates and also in insects. These are the DMRT families, SOX and FOX families. They have been investigated in 40 bivalve genomes. And there the author found that there are some usual suspects, so some genes that seem to be recurrently involved in sex determination in bivalves. So for male determining genes, these are the DMRT1 genes. So this is a gene that is specific to bivalve. But there is actually another gene, the DMRT1 gene, that is a master male determining gene also in mammals and in fish. And SOX-H are, let's say, usual suspects in male sex determination. And one gene was FOX2. This seems to be a master female determining gene. So please remember this name. It might come later. Female determining gene. What we also know from that study, and I found that particularly interesting, is that this master male determining gene, DMRT1L, is absent from the zebra and quagga mussel genome, and actually from the whole family. So it is really a true gene loss that happened a long time ago. So if there is male sex determination in these mussels, it should involve another mechanism. Yes. So we started with generating a reference genome for quagga mussels because, well, there was one draft genome, but it had like 80,000 scaffolds. So we decided, some colleagues of mine decided just to do their own new chromosome scale genome. I just go very quickly over it, but we managed to have a decent genome with 16 linkage groups, relatively good Busco scores with over 92% completeness, quite a lot of gene models, but this was using transcriptomic data. And some collaborators also generated the karyotype for that study, and they found that they also had 16 chromosomes like zebra mussels. For zebra mussel, this was already done, so we had a chromosome scale level genome and a karyotype that you saw before. So then we first compared the genomes of those two species, and apparently they are very stable because we have the same number of chromosomes. They are all homomorphic, and no differences in size among them, so no obvious sex chromosome. And also similar genome sizes with 1.6 gigabases of genome size estimate. The assembly was a bit bigger for zebra mussels, but this, for quagga mussels, this is the assembly we reach with also over 96 and 98% of the scaffolds that were put together on those 16 linkage groups. So then what we did is a synteny analysis where we do this, whole genome alignments, and we thought, oh, everything will kind of be the same. But actually, no, we found a lot of genomic rearrangements. So some chromosomes were quite, let's say, stable, like this, the red one. But then there was another one, you can't see, but basically coming from the right side, here you have linkage group, that's what, 5. And it has like genes, like homologous genes that were on 10 different chromosomes in quagga mussels. So it means that there are lots of genomic rearrangements happening between those two species, despite the fact that they diverged relatively recently, about 10 million years ago, that they have the same genome size and the same number of chromosomes. Well, which leads us to think, what are these sex determination mechanisms conserved or are they divergent? So what did we do next? Moving to zebra mussels. We collected males and females from one Swiss lake, and we performed whole genome resequencing. Then we did a classical pipeline of mapping to the reference genome, SNP calling, and we had quite a few SNPs in the end, about 24 million SNPs. And then we identified candidate sex locus by doing first a frequency-based inference, by doing this male-female FST that I showed you before, and also by identifying sex-linked SNPs for both types of systems. So for ZW SNPs, that would be SNPs that are heterozygous in females and homozygous in males. And for XY SNPs, that would be heterozygous in males and homozygous in females. So this was just frequency-based inference, but then we wanted to use this latest model that had been published not so long ago. It's called SDPOP. You have the reference down there if you're interested. And there you can calculate the likelihood of a model following a ZW, versus the null model, which is the model of no sex chromosomes. And then you can compare the likelihood of those two models, and we did that over windows of 10 kilobases. And we did also the same for the XY model versus the null model. Sorry. So moving to the results, this is the results for zebra mussels. We have the linkage groups here on the y-axis, and in black and gray you have the FST values, and we would expect that FST is about 0.5 for sex locus. And on top of that, we plotted the ZW SNPs, so the ones that are heterozygous in females and homozygous in males, because it seems like it's where we could see really clusters and peaks. We did that also for the XY SNPs, but it didn't really look like that at all. It looked a bit cluster all over the place. And then we did the model-based inference here. And here it seems like we have those two linkage group 5, and linkage group 13, that had really signatures of, let's say, ZW sex determining system. So it seems that in zebra mussels, we do have a polygenic ZW system, because it's on 2 different chromosomes, and we have several genes on those different chromosomes. Now, looking at what those candidate genes are, this is linkage group 5, and you see those 3 really nice peaks. And the first one, so those 3 peaks corresponds to 3 genes, so it's really, really small regions. And this peak was annotated as a Tudor domain containing protein that has been shown to have a role in sex determination in other species. Then the second one is this FOXL2 gene that is a known female sex determination gene, so the one that I was showing you in the introduction. So when we saw that, we're like, okay, this is really strong evidence that the candidate we have is the right one. And then we had a third gene that was known to be regulating transcription, which is what usually these sex determining genes do. And then moving on to linkage group 13, here it was a bit more messy. It seems like there was especially this first region that has strong differentiation between males and females, but still we had a few clusters of ZW SNPs in there. There the region was a bit bigger with 21 genes that were annotated in there, and we found 5 long non-coding RNAs. And there are a few studies showing that these long non-coding RNA could have a role in sex determination in other species. We also found a few a bit further down, and here 7 genes with 1 transcription factor, and here in the end we also had 1 long non-coding RNA, which seems to indicate that we have a ZW polygenic system in zebra mussels. So now what did we find in quagga mussels? So we sampled also 20 males and 20 females from a different lake, because they did not invade yet the lake where we had the zebra mussels, but I hope we managed to protect that lake for a few more years. We did the same pipeline, basically whole genome resequencing, alignment, and SNP calling, the new reference genome that we had. And there we had many more SNPs. We had 42 million SNP, which was very diverse species. And we did the same pipeline, we done the same analysis with the frequency-based inference and the model-based inference. And here we were really surprised to see that we didn't have evidence for a polygenic ZW system like we saw in zebra mussels, but here we had more evidence for an XY system. It seems a bit messy, but if you look a bit closer, there is like a region here on linkage group 13 that is really a nice FST peak. It has really a nice cluster of XY SNPs, and using the model-based inference, it is also a very, very strong peak for the XY model. Yes. Yes, and then we were wondering, okay, this is on linkage group 13, maybe it's the same thing as in zebra mussels, but actually, if you remember this synteny plot, linkage group 13 of zebra mussels and linkage group 13 of quagga mussels are really not the same at all, because here linkage group 13 of zebra mussel is the green one, and here you can see that the one of quagga mussel is composed of many, many colors, like blue and yellow and red and everything. So it's just by chance that it's number 13. Let's say. Yeah, now looking a bit closer at this region, there the region is much bigger. If you remember in zebra mussels, it was regions of maybe 1 gene, of 10, 15 genes. Here the region seems to be larger, it's about 1 megabase. And there we had 52 genes in there. And that is also what might happen with your annotations when you do annotations of non-model species, that you have a lot of gene models, but you don't know what they do because those genes don't exist in your, they haven't been annotated. So we had a lot of, I have to say, there are a few genes that are not annotated in this region. But what I was most puzzled about is to find FOXL2. If you remember, this gene is a known female sex-determining gene. So when I saw this, I stopped and I checked everything again. I went back to my notebook saying, oh, did I sex them wrong? And then I checked the sex files. So if I didn't, you know, by chance inverted the males for the females, I even did some reference-free analysis with k-mers that I don't have time to show here. But basically, it seems that this is solid. This is true. It's an XY system with the males heterozygous and females homozygous. And the FOXL2 annotation is also correct with the electronic annotation. So what I did next is to look at if this was really the FOXL2 gene in this region. So I took the FOXL2 zebra muscle sequence and I blasted that against the whole genome of quagga mussels. And then I found that the true homologue of FOXL2 was not on this linkage group 13, but it was actually on linkage group 8, so somewhere totally different. So what's actually sitting in the middle of this sex-determining region? This is a new gene that I call FOXY, because I think it's male determining, so let's call it like that. And this is an alignment of the FOXL2 true gene, the quagga one. And here in green, it means that the alignment is really good, like 97% identity or even more here as well. And this is why the electronic annotation picked the FOXL2, because this region is actually very, very well conserved. And then in the middle here, this was not conserved at all. And this didn't lead to anything. When I blasted it, it was like something completely new. So this FOXY is very likely a paralog of FOXL2 after gene duplication. And it's just sitting in the middle of the sex-determining region. So what does it do? So then we investigated the DNA binding domain, because this is really like the important part of the gene in a transcription factor, which is called the fork head domain. And what I saw is that it was kind of sitting here in the middle of this weird region that didn't have any homology to anything. And so when I checked if this fork head domain was also in the FOXY gene, actually no, there was the beginning of the fork head domain, and then there was an early stop codon here. So it means that there is this is probably not likely that it's expressing a repressor FOXL2 protein. So what is our current hypothesis at the moment? And I admit it's very speculative because I don't have much data to support this. But we think that this FOXY acts as a negative regulator of FOXL2. And how would it do that? When we have here an XX system, then FOXL2 would lead to the female development. And then when we have an XY with this FOXY gene, that would repress through a non-coding RNA mechanisms, the expression of FOXL2 early in development, which would lead to the development of males. So this is our working hypothesis. Yes, so to sum up, I think I showed you that we have zebra and quagga mussels have very dynamic genomes because they have opposite sex-determining systems. So first polygenic versus unique region, and then ZW to XY. And I think why this is promising is that for quagga mussels, we have a single region. So that could be a good target for genetic biocontrol. For zebra mussels, I think a polygenic system would be too difficult to tweak to have like good GMOs. So I would not go down that route. I would try different things for zebra mussels, but sex ratio distortion might not be the way to go for zebra mussels. Yeah, but before going to genetic biocontrol, we would have to actually validate this candidate locus in quagga mussels using experiments. And to do that, we need to rear these mussels in the lab. And so far, my lab has not managed to do that. They're really difficult, but I will get there eventually. Yes, and anyway, if we want to develop genetically modified organisms, we need to master the life cycle. We need to have these mussels reproduce in the lab. And just a few words about stakeholder engagement. So when I talk about this to cantonal authorities or people who are working in the drinking water companies, they're all very excited. to have like a sustainable solution to the quagga problem, because quagga mussels is a big issue in Switzerland. Zebra mussel, not so much. But yeah, still, we're not there yet. There are still many barriers to overcome. So first, there are lots of technical aspects to overcome, like rearing them, mastering the genome modifications. And then there are some regulatory aspects, like can we actually release genetically modified organism in the wild? So currently, the legislative landscape in Switzerland does not allow it, but we will see what happens in the future. And there are also big discussions with this gene drive about ethical aspects. How ethical is it to drive a species to extinction? Are we really allowed to do it? Should we do it? Yeah, so there are still a lot of things to develop before we can actually have that working and going. And with this, yeah, there are many people I would like to thank for this project, mainly my co-authors, but also many people involved in this project. And I thank you for your attention.
Thank you, Alex. Now we are open for questions. Are there any questions?
Thank you. Really nice talk. Actually, for zebra mussel, I don't know if the genome is still the same, but when I downloaded the genome 2 years ago, and I found a lot of problems with that genomes, like 200,000 annotated genes.
Okay.
In the NCBI annotation. So, I mean, Fox, I didn't get if actually you can find the Fox L2 also in zebra mussel, or you cannot find it at all. I mean, maybe it can be something related with the gene annotation actually.
Yeah, no, actually, I did find it in zebra mussels. That's one of the candidates. But I can check if it's also a different copy of it, and I will check the annotations again. Yeah.
Thank you. Any other?
In your homology plot, it wasn't clear. Are you talking about amino acid sequence, or it's DNA or exons that are missing or not aligning between these two?
No, it was orthologs that were aligned. Yeah.
So the protein sequence.
Because I didn't do those analyses, but no, I think it was amino acid sequence.
Okay.
Yeah.
And then this kind of interference mechanism, I guess you're not suggesting like RNAi, but what, recruitment of other factors, or?
What I'm confident to say at the moment is that it doesn't seem it's protein-based. I think it's non-coding, some kind of RNA, non-coding RNA mechanisms. Yeah. But I'm not a molecular cellular biologist, so I don't know much about this. But yeah, that's the hypothesis I have at the moment.
So we have an online question. So do you happen to know the sex of the zebra mussel, quagga mussel that was used for the genome?
Yes, I think they were both males.
Okay, because yeah, they were saying this: if it was a female, you would like the EM31 gene. So, and another one.
No, but this gene absence, that's yeah, you mean that, yeah, but it was done in a phylogenetic context, and there you could see that almost all of the species that were in this clade did not have this EM31 gene. So I think this is a gene loss that happened much earlier in the past.
In a polygenic sex determination system, so I guess that how well GMO-based control works depends on the architecture of the sex determining the lossy. So can you check with your data whether they are dominant?
Whether what is dominant?
The GMO-based control works.
Yeah. So in the GMO, the genetically modified organisms?
Yes, if the sex determining architecture is.
I think in GMO it's never polygenic. I think when they do it, it's a classic XY system with a strong degenerated sex chromosomes. Because actually true examples of polygenic sex determination are actually fairly rare. So there are a few in fish that I know of, Yes, I think all of these genetic biocontrol with sex ratio distortion that has been done is just on classic sex chromosomes.
Okay, thank you. There's a last question.
Given Switzerland's current thinking on GMOs, are you gonna just continue this conversation for the foreseeable 100 years, or what is your confidence of actually beginning to change perception especially in a situation like Switzerland?
Well, maybe that's because I'm an eternal optimist, but I think anything is possible. Yeah, no, and I mean, in my research institute, we are fairly close to policy and practice. So I think it is possible to have at least this conversation going. And this quagga muscle is such a big problem that it might be, the exception to the rule. Nobody wants this genetically modified maize in the plates, but when it's about saving millions and billions, maybe they will change their mind. Yes.
Money talks.
Yeah. No, it's sad, but that's how it is. Yeah.
Okay, thank you again for a nice talk, and we can move on.
You just put it. We can hold it. I know. Okay. Thank you all. So we're diving back into our case studies. Big breath and buckle in. I was told yesterday evening by Gabriella that my threats were a bit aggressive yesterday. So I'll warn you at 10, 12, and at 15, I'll stand up and sing Lady Gaga or something, and it's time to move on. Yeah. Okay. Cool. And we have two people presenting online. So. Senya, I can see you, and hopefully Hannah Solucia will join. We'll see. Otherwise, take it away. Yeah. This one is the big button to go forward.
Okay. So hello everyone. I'm Pier Giorgio Massa, PhD student. PhD student from the University of Bologna under the supervision of Alessia Cariani. And this project is on the application of ancient genomics to the conservation of the bluefin tuna. So this species indeed has one of the oldest commercial exploitation history, and as it is testimonied by this, and it may have started already in the 5th century before Christ, as it is testimonied by these coins and some other findings. These coins that have been found in Turkey, and which dates back to the 5th century before Christ, where tunas are represented on them, highlighting the importance to the Greek and Roman societies. So this figure here comes from a recent review on the commercial exploitation history of the bluefin tuna, and based on the archaeological records, and the dotted line is the number of Sicilian tuna traps. And what is important here to notice is that the tuna trap catches may have, what they suggested here is that the tuna trap catches may have reached industrial scale levels already in the 19th century, to levels comparable to the most intensive decades of the bluefin tuna exploitation. Then, with the advent of industrial fishing, the number of tuna traps decreased, and these have been replaced by these new techniques, fishing. And by the end of that century, the 20th century, the eastern stock has strongly declined. So some catch quotas had to be established to make the stock recover. But this has been done according to the 1970s baseline, as this is the period from when the routine fishery data collection started. But according to this study and some other previous other studies based on fishery independent data, this baseline corresponds to an already altered state of the stock, a phenomenon that is quite common in marine fisheries management, and that has been already described by Daniel Pauly in the 1995, and yeah, which leads to the gradual accommodation of the disappearance of species. So what we wanted to do here is to basically check with genomic data if actually this baseline correspond to an already altered state of the stock, and when the impact on the genetic diversity of the tuna has started. So this project builds on the results and on the dataset that Adam John Andrews has obtained, a former PhD student of our group that is now a researcher at the Norwegian Institute for Water Research and is a project member, obviously. And so he has collected these samples from 80 samples from the Eastern Atlantic. In blue, you can see the modern ones from Norway, Western Mediterranean, Central and Eastern Mediterranean, and then in red, the ancient ones. The most recent one is from 1941, the oldest ones from 3000 years before Christ. But as the sample size of this one was very low, 3, we had to exclude them from the analysis. And so the oldest one are now from the 9th century. Okay, so he has mapped the whole genome sequence, all of them. Then he mapped to a highly scaffolded reference genome that was available at that time. And he already found some evidence of a genetic erosion in the modern populations. But now we have a new chromosome reference genome that has been produced under the ERGA pilot, which is also annotated. And in this project, we have resequenced the same libraries to further increase their coverage and to make more comparable the coverage of ancient and modern samples. And we did so by obtaining a mean depth of coverage of 17.7x for the ancient and 17.8x for the modern ones. And with this new data, we wanted to further to get more accurate genotypes and to further corroborate the genetic erosion and to focus on the non-neutral portion of the genome. So, okay, we applied a stringent, so this pipeline here to obtain genotypes from the FASTQs, we had to rely on some stringent and ancient DNA aware filtering, which is there reported, and we obtained this data set of oops, no. Sorry. Okay. This data set of around 700,000 samples. Oh, okay, it disappeared, but it's okay. Okay, so we obtained this data set of 700,000 samples from 34 modern and 34 ancient samples. From this data, we managed to corroborate the weak genomic erosion that has been already observed by Adam John Andrews. And as you can see, on the left, we have the ancient sample, the observed heterozygosity of the ancient samples. Here, of the modern samples, there's a slight decrease, which is statistically significant. And when looking at the single temporal bins, what you can see is that there are some signals that the genomic erosion could have started before the 20th century, as the zoological data and the tuna trap catches showed. But all of these tests are not statistically significant, probably likely due to the small sample size. And so the main point here is that the 1970s baseline may underestimate the historical bluefin tuna productivity, and that the genomic data could serve the stakeholders to extend the short timeline covered by fishery-dependent data. So what we propose here is that the true natural baseline could be reconstructed with ancient DNA, obviously with larger sample sizes, and complement these results with demographic inferences from modern data, and then to establish some genomic monitoring to actually track the rebuilding of the stocks, and then combine the genomic indicators and catch effort and environmental indices to set the thresholds. So the main stakeholder for the bluefin tuna is the International Commission for the Conservation of Atlantic Tunas, ICAT, which already has a research program. So in this first here, we focused on obtaining sound results, but we are trying to organize a workshop with the ICAT and other stakeholders, where me, our group, and other researchers from the Norwegian Institute of Marine Research and Canadian Ocean Fisheries are going to share all of our results, but we are not there yet. And then for the communication, we presented our results at several scientific conferences, and we then posted several posts on my social media. On the institutional one, we had an interview with Ikaya, and with this article here, we had a blog post on the University of Bologna website, and then a newsletter, and we are now preparing a post for the sciencenorway.no. And we then presented our project at the European Researcher Night in Ravenna. And this entire work is actually a chapter of my PhD thesis, which I submitted yesterday. So that's why I looked that stressed, because I am. But yes, I'm going to later write down a scientific paper, but the analyses are still going on. And all the data have been published on NCBI. This is the bio project. And what about the next step? So I did not have time to present all the results or the analysis that I obtained, because as I said, our focus was on the non-neutral genomic part of the genome. So I just reported what the main point, but we also did some genetic load analysis, and we are now focusing on the positive selection, on the detection of positive selection signals through observed temporal frequencies and the imputed one from ancestral recombination graphs. But we are still moving on this part. And so this is the program for the next 3 months. And then then we want to write down the manuscript and to organize the workshop. And in the very long period, our idea is to increase the sample size of the temporal beans to get more reliable estimates, especially for the very deep time samples. And then to sequence the true modern samples from 2025 to check the current trend, and then to obtain a similar timeline, more recent one, for the Western stock to see what is happening on the other side of the Atlantic. But yet the main limitation are the ancient samples collection, especially for the Western stock, and fundings. So I want to thank all of the project members and all the ones that joined later, and Christian and Chiara that help us quite a lot. So, and thank you all.
Really cool stuff. Thank you. We have time for a couple of questions. I wanted to ask, you're in a very unique situation here with all of these data samples, and it's quite, I don't know, either exciting or worrying to hear about it, depending a lot on which baseline you choose. So if we have species where we don't know this historical data, how do we interpret what the baselines really are of our...
The ones you mean for the other, all the other species?
Yeah.
Yeah, so actually so ancient DNA could give you the real scenario, but we are moving on with methods to detect the past demography, even the recent past demography with modern data. So that's what can be done. But yeah, I cannot answer to your question because I'm just a bioinformatician. So yeah.
Okay, then I'll ask a bioinformatic question. In that, does all of your data follow this stringent ancient DNA pipeline, including the modern data?
What can you say?
So you said you used this very stringent filtering
based
to capture the ancient DNA, but did you also process the modern data?
Yeah, the same way. Actually,
and are you losing signal potentially because of this?
Yeah, probably yes, because actually we for Adam had to sonicate the DNA fragments of modern sample to make them of a same read length of a same length of the ancient ones. And then we applied all the same filtering. We had to remove the transitions. So yeah, that's why, yeah. So we actually obtained a quite large data set, but yeah, you could, so if we restrict the same filtering on modern data, we get a much larger data set. We still managed to observe what we wanted to observe.
Yeah. Then thank you very much. Oh, sorry, Rob.
Okay, biology question for the bioinformatician. What is the lifespan and more importantly, the reproductive lifespan of the bluefin tuna?
Okay, so I know the generation time actually because I did some simulations, so I had to search for that. I am not sure about the lifespan. I guess it's around 30, 40. Yes, 30, 40 years. And yeah, but yeah, the generation time is around 10 years or even more.
Okay.
This is what is reported in scientific literature. Yeah.
Okay, thank you.
Thank you very much.
Thank you.
We have Lucia. Is Hannes also joining us?
He's joining online, but I'm...
Oh, perfect. Okay. Then it's just the big button, and that's the pointer.
Okay, that's the pointer.
Yep.
Okay, so thank you for having me here. I'm here on behalf of Hannes, and I will present our research that we did together. So just to clarify, I'm a research and teaching assistant at the University of Zagreb. I'm also a PhD student there under Ivana Bui. But a year ago, I started my double PhD together with Hannes from the University of Antwerp, and I'm going to present what we did for our research. So to give like a bit of context, what is going on here with the species that we were working on. So as you probably know, the Mediterranean region has a lot of species that are endemic and also a lot of, sorry, oh no, yeah. Okay, thanks. And a lot of those are also considered to be threatened. So the main group that we will focus on are the sand gobies, especially the Cliporichia species. But I will also include one Orsinegobius species, that is Orsinegobius craticus, just because it was before assigned to the Cliporichia genus as well. But then based on some morphological and molecular analysis, it was actually reassigned to another genus. So although the Adriatic Knipovicea species, which I will focus on, have been described based on morphology and ecology as well, when people were using some standard mitochondrial DNA barcoding, they actually failed to separate them as species. So we can see that here. So we have Knipovicea panice, Radovici, and Mrakovčići, which were actually clustered together. And here we can see the Orcinigobius craticus, which was then considered to still be the Knipovicea genus. So based on all of that, actually, that's why we were studying with the genomic analysis of these species, so we can clarify the problems here. So something about the Knipovicea genus. They're mostly microendemic, not all of the species, of course, but some of them are. So some of them we can find only in one location in a single river basin or like in a lake. So their range is quite small, and we can also call them probably steno-endemics as well. Because of their small range, they can also, of course, be vulnerable to local anthropogenic disturbances. What we wanted to see in this research is actually a possible gene flow that can happen between sympatric populations. I will, of course, explain which ones. So here are the species that we included. We first have Knipovicea panice, which is a euryhaline species, so it is not freshwater as all of the others are, and it is also widespread. We can see that here on the map. So Knipovicea panice is her range is assigned with the yellow color. So then the other species are Orcinigobius craticus. We can see that its range is quite smaller, but it's still not considered to be a microendemic species. And then we have the Knipovicea radovici and Knipovicea mrakovčići. So Knipovicea radovici is here, endemic to the Nori River, and Knipovicea mrakovčići is here, and it's endemic to the Karka River. What I also want to say here is that you can see as the range of the species is declining, it's considered to be more threatened, which of course also makes sense. Okay. So what we wanted to do actually with these populations, we wanted to investigate the gene flow between the sympatric Knipovicea and Orcinigobius species, and we also wanted to investigate population differentiation. I also want to say something about the reference genomes, because a part of this project was actually focused on finishing 2 reference genomes, so the annotation of 2 reference genomes. One was for the Knipovicea panice that I'm working on, and the other one was for the triplophene blenny. So basically the annotations got stuck with NCBI for a lot of months, and we still don't have the annotated reference genomes. So we started to think about the possibility of doing the annotations ourselves, because for further research, we of course need annotated genomes to be able to make some conclusions. But what we do have is the data for the reference genome of Knipovicea panice that was made. So it is still not annotated, but the reference genome was made as a part of a ERGA pilot project, and we are actually quite happy with how it turned out. So the Busco completeness score is quite all right. So I think it's going to be just fine. So for the population sampling, I wanted to show this map just to show how some species are actually quite distant, and how some are quite near. And I also wanted to say how the Norin River, which is here, so we have 2 species there. One is Knipovicea radovici, and the other one is Orcinigobius craticus. And the results will actually focus on these 2, which are sympatric. This is like just the pipeline to see how and what I did. So I did a lot of analysis. I've been working just 1 year on that. Of course, I will not show all of the results. I will just focus on the results that can kind of answer our questions. But for the most important part, we did whole genome resequencing, and the average kind of coverage for the samples was around 20x. So of course, I first started with a phylogenetic tree just to check where and how our species are now distributed. So these are the species that I worked on: Knipovićeva panica, Mrakovčić, Radovići. Orsinivovius croaticus is now here. And if you remember the tree that I already showed, its position significantly changed now once we're using the genome data. And of course, I put them into context. So all of these other species I took from NCBI, and they are all part of a Gobida family. So what we wanted to check here, this is another tree that I did. It's a neighborhood joining tree, which was reconstructed based on the distance matrix that I got, also using whole genome resequencing data. It's of course a preliminary result, but we can here see all of the samples that I used. And basically the questions that we wanted to answer are the problem that we saw while sampling Knipovići Radovići. So we are sampling it at the same locality where we also have Orsinigobius coraticus. And these species, if you don't know, are quite small. So they're like 2 centimeters. And there was some misidentification going on the fieldwork, because you really cannot tell them apart just by looking at them. You have to take them to the lab, use a magnifying glass, and really do some lab work to see which is which. So once sampling the Knipovići Radovići, we actually sampled some of the Orsinigobius coraticus from Norin as well. Another thing is, which we can see here, that both Knipovići Radovići and Orsinigobius coraticus are non-monophyletic, because we have 2 populations now here from Orsinigobius coraticus, and from Knipovići Radovići, you can see that these 2 samples are kind of separated from these 2 as well, even though they were sampled at the same locality. So we were trying to see if there's a gene flow pattern going on between the sympatric ones. We first did the analysis on the whole genome, and we used specific population pairs as shown here. So for P1, we chose Knipovićeva panica, which is more related to Knipovići Radovići than Orsinigobius coraticus. And then we actually saw that all of the 4 Knipovići Radovići samples that we have show some gene flow with the sympatric Orsinigobius coraticus. However, these 2 samples here show much stronger signals than the other 2. We could also see them, how they were kind of separated in the tree as well. So that was based on the whole genome. But what we then did is look at the specific chromosomes. So we looked at all 23 chromosomes, and most of the chromosomes actually showed something like this. So for the admixture proportions, there were really not a significant signal going on here. All of the kind of values are more or less positive. But if we look at this, here we can actually see the differentiation going on, because these signals that are red and blue are for the Knipovići Radovići that were also kind of distinguished in the tree, and for which we got higher samples when we were looking at higher values when we were looking at the whole genome. And the other colors that are kind of not so visible and not so significant here, those are for the other 2 samples. So basically, we do have a differentiation going on here. So some conclusions. The taxonomic misidentification actually shows that during fieldwork, we cannot really assign which species is which in a very easy way, and that could also be a problem later if some stakeholders were trying to do the same thing that we did. So genomics will then allow us to easily distinguish the sympatric species Also, the outlier region that we see on the chromosome 12 can suggest a recent gene flow or maybe an introgressed inversion that is still polymorphic on that site. But of course, as I said, and as you saw, that was only based on 4 Knipovici-Radovici samples. So we need to include, of course, more samples. But we have some problems there because most of the samples from Norin, usually when we check them, turn out to be Orsinobulbus praticus, which can also point to the vulnerability of Knipovici-Radovici that we have there. So it's kind of hard to continue research when you don't have a lot of samples there. But for the stakeholder engagement, we identified who the stakeholders might be. So the public institutions of the national parks and nature parks that are affiliated for those regions. Of course, Ivana will say more about that in her presentation. It's more conservation related. And of course, for the next steps, we need to analyze more samples to further this investigation. We also need to get the annotated reference genomes so we can, of course, then progress with the investigation as well. And we also need to kind of look more deeper into the chromosome 12 and then the results that we got for some potential selections or adaptations, or of course, to look at the evolutionary history of the species as well. And of course, yes, the publication of scientific research as always. So yeah, thank you for listening, and I'm open to questions.
Thank you very much. A question. I know that there are more genomes coming from these species, and how is this going to change your analysis? Or...
Oh, it's going to be great. I could finally look into them in a proper way. Yeah, there's supposed to be like annotated genomes from Knipovici-Radovici, Vrakušići, and all the others as well. So then I will finally be able to look into these questions more thoroughly.
Yeah, very interesting. So just to see if I understood correctly, so you find evidence of introgression when you look at the statistics and you get very high D values.
Yeah.
So that maybe, are you sure that you're looking at the correct topology? But then related with that, just to be sure I understood, when you look along the chromosomes, you only find evidence for introgression in one of them. So it means that the very strong D signal is coming from that single chromosome. Yes, did I get it right? Okay.
Yeah, that's it.
Because otherwise it could be that you have a wrong topology of your P1.
No, it's just the chromosome 12. So all the other chromosomes didn't show such things.
Okay. Yeah.
Okay.
Thanks.
I have a question, like a follow-up on this. So if the signal comes from chromosome 12, have you tried? And so that's the reason. So your hypothesis is that that's why it disrupts the monophily in that in that tree.
Okay. Yeah.
Have you tried rerunning like the statistics but removing chromosome 12 and redoing the phylogeny? Because
no, I didn't.
If it's caused just by that chromosome, right? But it is a strange and interesting pattern, and definitely the reference genomes will help you understand if there is some structural variation in that in the different species in that region.
That's actually a great suggestion. Thank you. Yeah, I didn't do that, but definitely.
Just as a sanity check.
Yeah, it makes sense. Thank you.
Thank you very much.
Yeah, thank you.
Sorry for my multitasking. Which of these buttons means full screen? Okay. Ksenia, are you there?
Yes, can you hear me?
Perfect. Take it away.
Okay, thank you. So my name is Ksenia Saropoulou. I'm a PhD student at HCMR Greece, and today I will present to you our case study called Sponge Genes, Adaptation Genomics of the Endangered Sponge of Sinalis through whole genome resequencing. Okay. So a little bit of context. Sponge officinalis is an iconic species of the Mediterranean benthic ecosystems. It has been exploited since ancient times, and therefore it has high socioeconomical value. And as we can see in the map, its abundance increases from west to east, where it forms sponge grounds, which are aggregations that have very important ecological value to the biodiversity and ecosystems. However, the past decades, the species has been facing many threats, the most important of which being overharvesting, disease outbreaks, and habitat degradation. And this has led to severe population declines, and therefore it has been suggested that these species should be included to the status endangered in the IUCN Red Lists. So despite all these facts about Sponge officinalis, unfortunately, it's a species that still remains particularly understudied. So the goal of our case study is to shed light on the ecoevolution and the current status of Sponge officinalis in the Greek seas using whole genome data. So what we most want to investigate are the patterns and levels of genetic diversity and genetic structure, and also signature of selection and local adaptation, and the adaptive potential of the species. And these are all aspects that are very important for guiding conservation and management efforts. So the sampling scheme of our case study was based on a previous genetic study on Sponge officinalis using microsatellite data that was published 15 years ago. The samples that were collected during this study are kept at HCMR. So the idea was to visit some of the same sampling locations 15 years later, so that we can compare the genetic diversity between these two timestamps, and also go to new additional sampling locations. So this is our sampling map. With dark gray, you can see all the sampling locations that were revisited from the previous genetic study. And with red, you can see all the new sampling locations that were added. And in total, we managed to collect 193 new specimens. So then we went ahead and extracted high molecular DNA both from the new samples and from old samples. And we sent those samples for library preparation and short-read whole genome sequencing, aiming for 30x coverage. Then this data was obtained and processed for population genomics and adaptation genomics analysis, as well as genotype environment association analysis. And the reference genome of Sponge officinalis was produced in the framework of the Aquatic Symbiosis Genomics project and was published last August. So moving to our first results, as most of you might know, sponges have a lot of symbiotes, and this created a problem for us because this caused a very low mapping rate of our reads. So only 20 to 50% of the reads of each individual actually belong to the sponges, and the rest were symbiote reads. So this caused lower quality and coverage than we initially aimed for. And even though our initial dataset contained 179 samples, after filtering, we ended up with a dataset of 160 samples and around 1 million SNPs. This is a 3D PCA plot with PC1, 2, and 3. As you may be able to see, Sponge officinalis samples were grouped based on their sampling location and specifically their island, except the individuals that are in the middle of the PCA, where we have some samples from Crete and some samples from Piperi Island, which is in North Greece. So these were actually Hippospongia communis samples. Hippospongia communis is traditionally considered a different species from Sponge officinalis. However, genetic data so far have shown that this is not the case. On the right, you can see a phylogenetic tree with several Spongia species, and Hippospongia communis is placed in the same clade with Spongia officinalis. So this is also, our results also agree with that. And it was not, it wasn't the initial goal to study this in our case study, but it was an interesting finding. So going back to the Spongia officinalis samples that were grouped based on the region, this is also a heat map of pairwise FSTs. The values range from 2 to 8% and were statistically significant and in accordance with the PCA findings. So we have some indications so far of genetic structure, but of course we have a lot more analysis to do to draw further conclusions. And now I will move to the stakeholders and the engagement. First of all, we collaborated with NECA, which is the National Environment and Climate Change Agency, and it belongs to the Ministry of the Environment in Greece. We got sampling permits from them to do our sampling, and we also collaborated with them for sampling inside protected areas where we went with their boats to collect the samples. Then we organized the Biodiversity Genomics workshop, where we invited several stakeholders, people from an NGO, from NECA again, from the Natural History Museum of Crete, and from other institutes of HCMR. And we got to share our work for the case study and BG in general. And we got to discuss about conservation policies and management in Greece and how important are genetics and genomics for conserving and protecting the species. And finally, we are in contact with a trading company, a bath sponge trading company, the Kypreos France e-sponges, who are interested in bath sponge cultivation. So hopefully we will collaborate with them in the future for a project about cultivation, and we will use the results gained from this case study for this purpose. So regarding policy, in Greece, it's quite hard to make legal policies about conserving species. But what we plan to do is compose a guidance based on the results we get from this case study and send it to NECA. I've already mentioned NECA before. NECA is responsible for everything related to conservation, to protected areas, to implementation policies in Greece. So we're going to send our results and also propose how the species should be managed and protected. And I would also like to mention that the official assessment of Spongia officinalis in the category endangered to the IUCN Red List will be also published by the end of 2025. So hopefully with all this information available, it will become clear that the species should be included in future management planning and MPA networks. So now for the communication, I've already mentioned the workshop, but apart from the stakeholders, it was also open to the public. So a lot of students came and a lot of people from other institutes here in Greece came. And apart from the theoretical discussion on conservation and policies, we also had a training event where we trained all the people about sampling permits, about sampling, how to sample specimens for to get high quality genomic data. And also we did some basic bioinformatic analysis on reference genomes. We also had a press release in a local news site talking about BG and the case study, and in general how important is genomics for the conservation of marine environments. And finally, Teresa Manoussaki, who is the leader of the case study, gave an interview to a local radio station, also communicating to the public our work. And regarding scientific output, we have presented 2 posters in 2 different conferences, and also we had an oral communication to the World Sponge Conference, communicating our results so far. So now our next steps short term are to complete all the analysis we have to do, prepare and submit the respective manuscript for publication, and make all the data available, the raw data to the ENA and the codes on GitHub. And for the long-term goals, we want to plan restoration actions. We already have submitted a proposal about the restoration project of Sponge officinalis in Crete. So we want to employ the results we get from the case study towards this purpose. We want to engage more with stakeholders, as I've already mentioned, with NECA, and possibly plan some citizen science actions to communicate to the public the work and the importance of genomics in biodiversity conservation. So I want to thank everyone in the team. You can see photos of them in the slide. And I also want to thank all of you for the attention.
Thank you very much. We have time for a few questions. You mentioned these symbionts, and do you have any indication of whether these differ across the different sampling sites or time points that you looked at?
Unfortunately, not yet, but it's definitely something that we plan to look into.
Then.
So Tom asked about the endosymbionts. I was thinking more about whether you collected abiotic measurements for each of the sampling sites. So if you did, are they very different? Things like salinity, temperature, mineral contents present in the seas. And if you didn't, do you have an expectation that they would be different or that they are the same?
Yes, we only measure the temperature in each sampling site. I'm thinking that we will do this in the seascape genomic analysis part. So see if there is any association between specific environmental variables to the genotypes. I don't know. I don't think they will differ that much, to be honest. But I will tell you when I found out.
We can't wait. Then thank you very much, Xenia. Thank you again. And I'll ask you to stop sharing so we can redo this switch.
Pull.
Okay. It's okay now? Okay. Everybody hears me?
Yes.
Okay.
Wait.
No, well, I can greet the audience. Hello, everybody. How are you doing? What a day. I mean, we were just talking during lunch. I cannot remember a congress or a day in which I have heard so interesting information. It's not my presentation, but doesn't matter. We can now check our knowledge. Who remembers? What I was saying, so many interesting things on such a huge biodiversity. Thank you. So many animal, plant taxa, so many information. I really don't remember when was the last time that this was gathered. So many scientists for different disciplines gathered together. So huge congratulations to organizations. Thank you for bringing us here. So my name is Ivana Boj. I come from Zagreb, work at the Faculty of Science of the University of Zagreb. And I will continue with this story on the sand gobies, so Knipovicea and related species, that Lucia already gave you really a nice insight into the. So I don't think I need to go too deep to the introduction. This was the goals of our case study. So the majority of analysis regarding the sand gobies were conducted under this case study, 11.1. So we started with these two objectives to use the whole genome data to better describe and recognize different taxonomic units, because the taxonomy was not well resolved, and to investigate adaptations and putative ongoing gene flow in eurythal and freshwater species pairs, because these the species are secondary freshwater species, so they're colonized freshwaters from sea or brackish waters. However, during the course of the project, this third aim actually maybe overpowered or overcome all the other aims. So actually we have really tried to obtain even the additional data and to use this data for conservation purposes. So this turned out to be the most important. So here are the 4 species that are in our focus. We also work with some Greek species, but I will focus on this one because, you know, I have 12 minutes, so cannot cover the whole biodiversity. As Lucia already said, and as you can see, they're all considered endangered in various endangerment categories. With some I agree, with some I think they were not assessed well, but I will leave this story for some other occasions. They're all strictly protected under Croatian law, and they're all endemic. Some are really connected with really parts of river, not even the whole river, and some to the a bit wider area. So actually, when preparing this presentation and when listening to all of you in these 2 days, I actually realized that maybe it has been advocated well enough that the genomics is very important for practical, for effective conservation. So instead of trying to find the conservational purpose of our results, I decided maybe to shift a bit the story and to showcase to you how we prepare a certain conservation plan and in what parts, in what aspects the genomic or genetic is important. Because actually I'm not molecular genetics, I'm not bioinformatician, I'm an ecologist by vocation and conservation biologist by heart and soul. So this is how I have been doing it. And actually, this is why I got involved in genomics in the first place, because in some conservational cases, we needed some answers that could only be provided by genomics. First genetics, and now there's genomics even better. So that is the way how I will present this case. So when preparing certain conservation plan, designing conservation plan for certain species, there are a couple of questions that we have to answer. So first one, yes, the methodology that we have used. As you can see, as we always do in conservation, we have collected even more data. When we are in the field, we are not just collecting samples for genomics, but we always look at microhabitats, collect physicochemical parameters that you ask, take a look at precise microhabitats that have been exploited by the majority of species, etc., etc., and also about threats. That was very important. So as I said, questions. First, and the most important questions, what biodiversity units should be protected? And that is the question that conservation practitioners often ask us. So what have we got for protection? Here I mean species, of course, but also units below the species level. And here, this project and this investigation was really crucial and very important. Here are the Lucia already showed you very similar 3 with which, by using the whole genome sequences, we really corroborated the independent taxonomic status of each species, which was questionable prior to this investigation. I'm very sorry that we didn't have this project some 15 years ago, because if we did have these results, then also Knipovića Radović and Knipovića Mraković would be on Habitats Directive, because that was the time when Croatia was approaching the European Union. Now they are not. And maybe it seems that doesn't matter so much because they're both strictly protected, but in practical conservation, it's a huge difference. It's so much easier to protect Natura 2000 species than species that is not Natura 2000. And now they're not. These two are not. Another important result for conservational purposes is if you take a look at the Orsinigobius croaticus lineage, it is divided into two sublineages. So that is important to know because there is genetic distinctness, slight, but still there is between them. So both should be considered as conservational units. On the other hand, they are similar enough that both could be used as source for transfer in case of need of population augmentation of each of them. So then the second question and third questions that are often asked by conservation practitioners are, what are the distribution ranges? So we have discovered them. They were known even before this project, but we have corroborated them, and what threats are present there. And here it is really important to go to the field. I mean, of course, genomics can explain some threats that are present in the population structure, but usually those problems are consequence of some anthropogenically induced threats. So that's really very, very important. And so going to the field and revealing what's going on with this population also enables discrimination between whether some genomic patterns observed are natural consequences of maybe some historical bottlenecks, like some glaciations or something that we have been noticing, or are really recent and consequence of what is going on now. And then the fourth question out of 5 is what these threats and what these changes in habitat conditions that we are noticing, what have they been provoking in population structure? Here it is, of course, this investigation bring important knowledge, but I think it's the combination of genomic knowledge and ecological knowledge, and what we see in the field is actually how can we really find the precise answer. And finally, the fifth question from our practitioners is what should we do? What conservational activities are required? And of course, that depends on what threats have we noticed and what effects in the population structures also have we described. And for some activities, for example, if the habitat is degraded, I mean, we do not have to wait for whole genome sequencing. We can go on the field and try to repair it. But for population augmentation, it is really crucial to get a really deep insight into population genetics, so we can really precisely identify source populations for reintroductions, for population augmentation, for restocking. Particularly if we want not only to raise population size, but also genetic diversity, which is often the case. So by some artificial spawning here, there are some eggs. But speaking about this question, so these are like 5 groups of questions, and I try to put where we can find answers best or most effective by genomics. But I would like to ask you if maybe you would like to try a wild guess on what is the most often question that was posed by nature practitioners to me in my professional career. So it was not what algorithms did you use, how large are the genomes, what method, what confidence level did you get? It is something completely different. Can somebody maybe take a wild guess?
How much is it to protect it?
That's a good guess, but not. Actually, I remember this yesterday while I was walking, and there is a barrier near the hotel. There is a barrier on this small river. So this is really by far the most often asked question to me. What is the size of the barrier that a trout can jump over? In my private life, it was for lunch. Okay, I have just like a minute or two. So I will now try to go just quickly through the species and show you how the obtained results are being put in practice. So Orcinigobius curaticus is considered vulnerable. Our investigation actually showcased that this is not the exact distribution range, and based on that, because since its endangerment status is based on its distribution range and decline in it, I think it is not correct. These were the threats and conservation measures that have already been proposed for this species. We agree with that, but thanks to this investigation, we have also add some additional conservation measure. And particularly important is this recognition of two populations as two conservational units. Then Knipovichy mrakovčič, it is not a Natura 2000 species. We do not have monitoring of that species. I would like to include it in Habitats Directive, but whenever I ask, I got an answer, please do not touch Habitats Directive or do not touch Habitats Directive. So it's like it's not possible anymore. It is, again, the important input of our results is that it's just a single population. So really, and even more sensitive because of its low genetic diversity. Knipovichy radovici, we have confirmed its presence in one locations. It is possible that it's also in Modrooko, and Knipovichy panici, which is the only that has somewhat wider distribution. This is its presumed distribution. However, actually, this whole range is not inhabited by this species. Actually, its distribution is fragmented. So it is present only on several localities. And for this species, we haven't done genomic analysis yet. We will do that, but it is very likely that it is a complex of species. So you can imagine for conservation how important that will be. On the other hand, this one is a Natura 2000 species. Again, threatened conservation measures that have been already established, and additional ones by this investigation. We have been working a lot with stakeholders, not only in this project, but in many others. 2 workshops, and the most important conclusions of our work with stakeholders is that they still are not very aware of the applicability of genomic investigation, but are interested in conservational measures that are based on genomic results, but if we translate them just to measures. So it doesn't matter what's behind, but let me know what I have to do, how big barrier can be overlooked by trout. And what are our next steps? Well, it's maybe a bit different. We have already started with active conservation. In the Krka River, invasive species are already being removed. This investigation enabled localization, more precise localization. We are not still doing any augmentation. For that, some completely different investigations are necessary. In some monitoring programs, we have already included monitoring of genetic diversity. But of course, we have monitoring programs only for Orcinigobius Knipovichy panici, which are Natura species. And also, we are working with other public institutions to replicate knowledge and to maybe start similar projects and inspire them to use similar investigations. So with that, I would like to thank you very much for your attention.
Thank you very much. A lot to digest. I'll ask. I think a problem we have is that there's so much subtlety, and you showed that just these distribution maps are completely misleading.
Yes.
So how do you start translating this when you go in to talk to practitioners or write these documents to mix this subtlety with what they want, which is black and white? Okay, next talk.
Oh, God. How much time do I?
-10.
Sorry, okay. I mean, I can maybe try one or two sentences to answer. We have been working with nature practitioners for many years now, so we have really good relationship and good connection. So I don't have to start from the beginning every time. So they already know what are the problems, and we already know what they need from us. So that makes it easier. Okay.
Thank you very much. So thank you, Ivana. And Max is going to take us halfway out of the water, I think.
Yeah, exactly.
So we will leave the water.
Yeah. So my name is Maximilian Wagner. I'm a postdoc currently at the Faculty of Civil Engineering in Rijeka. So it's in the very northern part of the Adriatic Sea. And based on my question, I just reflected a little bit what I said, this thing with people don't know with how much it's this. I'm starting to doubt maybe I'm already too long at this Faculty of Civil Engineering, because that was one of the major questions I was recently often asked when I was at the Civil Engineering Conference. Well, you do these nature-based solutions, but how much are they? You know, it's much cheaper to do more hard than soft engineering. So, and my topic is also about what is considered from an engineering point of view as soft engineering practice, but as you'll see, it's also a little bit cynical, what I mean by that. So we go away from the beautiful rivers of Croatia. I think everyone who goes to Croatia should definitely have a look at the backcountry. But most of the people who come to Croatia, especially in summer, come there because of the pristine, crystal clear waters, these beautiful beaches. And the water quality itself is a result of this karstic coastline and also this gravel beaches. So we mainly have gravel beaches along the Croatian coastline. And the most enigmatic for sure is this Zlatni Rat, the Golden Horn on the island of Brač. Maybe some of you have known this. It's really a flagship and attracts millions and millions of tourists every year. But of course, with every kind of development, tourism has its limits. And in Croatia, for sure, it has reached its limits some time ago. And that gets more and more clear by all the sociological, but also ecological impacts this has on the coastline, on the people. And perhaps the most obvious, I'm not sure if I can play this video. Nope. Sorry, there's another video. I have two videos. So I realized you can embed YouTube videos, which is nice. So, so the problem was, so when you look at these numbers here on the right bottom corner graph, you see that's the amount of supplied sediments to beaches in Croatia, and it kind of quadrupled in the year 2018. That's when really tourism started to skyrocket in Croatia, and it's of course really much correlated to tourism. So there has been nourishment activities. So it's called beach nourishment. If you fill up beaches with fresh sediments, you have to do it, especially in urban areas where there are natural imbalances. But of course, it had also been done, and that's a very negative example of this community there. They created, you know, a whole sandy beach, kilometer-long sandy beaches, without considering any oceanographic or natural resources. And talking about this in the next video, nice, but that's it, okay, no more videos then. I might be a little bit biased there, but one of the most important gravel beach components in terms of biological components are these tiny fishes, less than 5 centimeters, of the genus Guania. They are endemic to the Mediterranean Sea. So in general, this intertidal, interstitial lifestyle has only evolved twice on the planet, in Japan, in gobies, and in the Mediterranean within these enigmatic clingfishes. And in the Adriatic Sea, we have 2 of them. And due to this beach nourishment activities, this year's recent IUCN revision set this one of these 2, and this is also the one where it's about this case study, the piglet sucker, as vulnerable. So it's quite interesting, the development, and maybe it's a little bit my fault. The interesting thing is, so not all beach nourishment activities affect these tiny predators. So within my project, my current project, we investigated a bunch of artificial and natural beaches. And in terms of artificial beaches, different sediment types are used. And in some of these sediment types, you can find this guania, this small clingfish, whereas in others not. And that really correlates with the sediment types and the sediment sizes and the associated interstitial spaces. So usually if the sediment size is too small, then the fish and probably the whole community is absent. So the idea was maybe these fish could be nice bioindicators to monitor gravel beaches and to see if the fish is there, maybe the whole community is quite okay because they're basically the top predators there. But of course, that's what my whole postdoc project currently is about. So we're studying in the first work package the whole community and compare it. Then we try to understand what are the trophodynamics in this whole system by gut content analysis of these top predators. And that's where the BG case study comes in. We study recruitment and connectivity patterns in these artificial environments with these 2 clingfishes that coexist, they occur in symmetry in artificial beaches throughout the Adriatic Sea, and especially in the northern Adriatic Sea. So the idea was, we had already a bunch of CO1 data throughout all this radiation. So there are 5 different species of this clingfish, and the top right ones and the center ones, Guania pigra, piglet sucker, and the Adriatic one, these are the ones that occur in the Adriatic Sea. But we see already that they show, at least not with the CO1 data, in the Adriatic, because the Adriatic fell dry, so you have lots of demographic effects there. But we see already with pigra, despite its CO1, it's not perfect to detect population structure, that it separates the southern with the northern parts. And so the idea was, why not using whole genomes to detect this even on a much smaller scale? Like in the Bay of Rijeka, which is where I'm currently based. It's a perfect setting. You have very industrialized parts where there are no beaches whatsoever. Then you have what I call urban tourism, so touristic places with lots of artificial beaches. Some include this fish, some not. Then you have urban areas that are kind of, there are beaches in between, so it's a very fragmented place. And the idea is now to collect populations from all these places. For instance, like here you have a stretch where it's like almost 5 kilometers, the big harbor of Rijeka, where there's no habitat available for the fish. And just as a practical example, because this is one population that's included in the case study, that's a beach because there is one raffinery at the end of Rijeka, and there is a current problem of oil spills. And one happened 2023. And as you can see, they removed the whole sediment, so probably the whole community is lost. But the good thing is, I went there for the case study to see if there are fish. And you see this tiny little recruit here, this larvae, so to say, almost post-larval stage. So the beach is already, after 1-2 years, again, nicely recovered because they used one type of sediment that apparently supports this whole thing. So as I said, we need proper genomic resources to do this. So far, we have already one genome of Guania adriatica. So this one species has already a chromosome-level genome, but we sequenced now the second species as well within this case study. And we're currently at the stage of pre-curation. So we have a scaffolded genome, and it looks quite nice. It looks, in my opinion, even better in the numbers of what we got back then. But of course, that's amazing how quickly the development went in the last 3 years and how cheap and accessible all this became, and also how easy to, like the application of the whole thing, like softwares, amazing. And I just made a quick analysis to show at least some results. So we collected already from each of the two species now 4 populations. So, and we sequenced them around to 20x coverage. And you see AA means Adriatica mapped to Adriatica, PA, Pigra mapped to Adriatica. So that's the old reference genome, and you see already you have a mapping bias there, because if you map it to the scaffolded genome, the mapping fraction increases a lot, and the same for paired reads, properly paired reads, mapping quality, and so on increases. So it's a good sign, it works, and we're on the right way. And if we get post-curation, I think we get a proper genome, which should not interfere for downstream further analysis in terms of population structure, hybridization, integration. Yes. Oh, I have to go on, sorry. So stakeholders engagement. I look like you there. So stakeholders engagement, we had several stakeholders. I portray some of them which already happen now. Of course, one of the most important is the museum in Rijeka. We also have the local authority, which is responsible for Natura 2000 areas and general management, Priroda. And we have regular workshops with a local NGO, and also the Students Association of Zagreb. And we plan to do this, we plan to do more workshops in the future, and also the next steps, so to say, engaging also with higher level stakeholders, which has not been done so far. Impact and communication, aside of some presentations that I made on conferences, we made one little documentary, which is like very cinematic and emotional, I would say, and a podcast about about gravel beaches and my love about gravel beaches and this gravel beach fish. And so the next steps include just finishing up this reference, do some sampling and a little bit more resequencing. They organize a mini symposium, because there is in the meantime a large community of engineers, civil engineers, biologists, NGOs, that because the problem of beach nourishment and coastal destruction is very, very prominent at the moment. So it would be nice to bring them all together, at least in an online meeting, to discuss and update ourselves. And and on the longer term, of course, writing the publications, engaging with higher level stakeholders, decision makers, policy makers. And with that, I want to thank especially also the people who helped me a lot with the project, and also BGE. I mean, this helps a lot, I have to be honest. So my budget on my postdoc grant is quite limited, and this really like almost doubled the amount of money I have left now, so I can do this properly, what I suggested in my original proposal. So I'm very thankful for that, and also for the patience and organizing, and from the management and work package leaders. It's really helpful, and thank you for doing this.
Well, thank you for presenting. I want to ask, are you...
Got it. So it's very particular that you found the species also on artificial beaches. That's, I'm curious about it. So did you maybe explore on which kind of artificial beaches? Because that could be also like a hint for the management, maybe to feed whatever civil engineering with which kind of, I don't know, gravel pebble material is better to create new beaches. Did you explore different beaches and the presence of these, or maybe also associated species?
Yeah. So we did quite a big effort there. So we investigated. There are 3 different kinds of sediments that are used to make artificial beaches. One is natural sediment. The second one is tiny gravel from quarry material. So it's crushed stones. That is, in my opinion, the worst because it's too small. It's very uniform. And then you have a third one, and that's river sediments. So it's basically quartz and metamorphic rocks. They are more resistant than carbonate rocks, so they will last much longer. And these usually also include the fish that we see and the predators. And so it's hard at the moment to give like a proper suggestion there, but we extracted not only the predators, but the whole species community. So now we have all this geological data. So we have the sieves, the fractions, the singular fractions, what kind of, how diverse the sediment types are, the sediment types themselves. And we want to correlate it with this whole species community using metabarcoding and also automatic image detection and counting to really give proper guidelines for the future. But in my opinion, diversifying sediment sizes is the key, because a natural beach has large stones, small stones, different sizes, more or less uniformly distributed. And all artificial beaches have one sediment size, which is, of course, artificially inflated. So this is why I'm very careful, because even though the fish, because they prefer one size class, right? They need a certain interstitial space, and if that's given, the fish is there. So maybe it's not a good bioindicator yet, but that's what we're going to study now. Yeah. Thank you.
Thank you very much, Max. And with Katja, we land firmly back on land.
Okay.
Take it away.
So yeah, I was thinking of how to make the transition from gravel beaches to Arnica. I guess it's now for something completely different. So yeah, actually a lot of you might know this plant, and hands up who's already been using Arnica in some medicinal preparation. And yeah. Did it work? For whom did it work?
I don't know.
Yeah, it's very difficult to tell, in fact. Yeah, so it's no news. Yeah, so for some people, apparently it works, for others not, but still, it's a widely used and popular medicinal plant. So approximately 50 tons of dried Arnica flower per year are harvested. And what, of course, most people don't know is a lot of this does not come from farming. It's from natural populations, because this plant is hard to cultivate. And this is quite worrying when you see this map on the left of the slide. Yeah, here, what you can see. Yeah, so even though Arnica is very widespread, so it's a true European species, I would say, it's declining in a large part of its range. So, and yeah, even though it's still least concern, some countries have already started to protect it. For instance, in Germany, it's not allowed to collect Arnica without a permit. And others not. So there's also quite a large heterogeneity between countries here. And also when you look at possible reasons for this decline, there is some heterogeneity. So some reasons probably apply throughout the range, and others are quite local things. I wonder if you, when we came here, if you saw the bus driving past eucalypt plantations. So this is actually something that is destroying Arnica habitats. So the meadows where it used to grow are not profitable anymore. People are putting trees there. And this is something that's quite typical for this region. So I also saw it a bit north from here in Galicia. But not in other parts of the range. There it's more meadows not being mown anymore. And also, even though that's a bit hidden behind the other effects, probably climate change. Yeah, so shorter winters, drier summers are just changing the dynamics in the meadows, and the Arnica as a rosette plant that is quite late, we're starting to grow in the season, has little chance to catch up with the others. The harvesting is a bit ambiguous in this context because on the one hand, it can be an incentive to keep the Anika populations. On the other hand, we have absolutely no idea on the impact of harvesting on population sizes, genetic diversity, or anything. So there's up to now 0 data about this. And we're with this study at least not going to change it, but maybe in future. Yeah. So yeah, in 2011, there was a European red list. And they said, yeah, Anika, it's declining, it has to be monitored. But they didn't specify how. And I guess there's a reason behind this. So Anika has 2 means of reproducing. It can form seeds, so sexually, and it can also form daughter rosettes clonally, which makes it extremely difficult to judge what is a population size. So concepts like the NE, for instance, do not work in these cases. And this is probably also the reason why already early on, so in 1998, it's one of the grandmothers in plant population genetics, people have started to look at this plant and to study genetic diversity. And yeah, the most widely used marker system is what is shown here on this map. So the different colored dots are structure groups that were determined in different studies. So different symbols are different groups of people who have collected or let others collect samples throughout the range. So the difficulty with this is that it's different studies, even though some of them use the same SSR loci in this case, microsatellites. The results are not compatible. So you cannot just take the results from one study and compare them with the other. You do not know if even if the sampling was almost at the same place, as for instance here, if the different clusters that came out, if it's actually the same or what is going on there. Yeah, so the idea of our project is to establish something that can be used by everyone, where people, also those that maybe just start to look at the Anika populations, let's say in the south of France or in Poland, where they can use something that is standardized throughout the range. And then we can just extend the data set. They have comparison data from other countries and they can see, are our populations more diverse, less diverse, related to this or to the other? And yeah, sort of make this a unified system. And of course, this also means that we have to look at different marker systems, what is representative, what is easy to use, what's the cost behind all of this. And then with this clonality topic, of course, we also have to think when we do genetic monitoring, it's on a time scale. So what should be the time step for which we look and in which we expect changes to happen? So all of these things we want to address with our little case study. So this is sort of the plan. Yeah, to achieve something like that, we had to use samples that were previously collected and extracted. Otherwise, we could not have done it. So we contacted people from all the different studies and were asking them, do you still have your old samples? Can we reuse them? Do you want to be involved? And luckily, except for one study where they unfortunately had already destroyed the samples, they all said yes and were all interested in doing this. So we now have sort of 2 data sets, approaches. Yeah, it's a little bit a complicated setup, so let me try to explain. First of all, what I've been talking about up to now is this space part of the project, where we have a bit less than 200 samples throughout the range. So we also discussed with the sample providers, where should we take the samples and how do we get a good representation? What is more important? Should we take more samples from one location or more locations? And then we have 4.5, I always say, because 2 of them, there was a translocation, populations within Germany, time series samples. Yeah. And for the spatial samples, we wanted to compare also 2 different methods. So rat sequencing was sort of the standard method we wanted to apply in the project. But then since there was this old SSR data, we also wanted to see, can we somehow repurpose this? Because up to now, there have been almost 2,000 samples of Anika collected and studied just with the previous projects.
And all this data that we have, we probably cannot replicate this because the samples either are gone or it's not good quality anymore. So the idea was a little bit, we sample like 10% and then try to extrapolate based on SSR sequencing and what can we do. Of course, there's also science communication part, but I will come to this later. So yeah, now for the bad news. I will spare you the details, but the rat sequencing was an epic disaster for us up to now. So yeah, we have a plan how to get out of there without also bankrupting anyone. So I think funding, we can manage this somehow to still get it done. But yeah, it was quite stressful for us the last weeks because we were always trying to still get something, and yeah, we will have to do this after the project. But luckily we have plan B, we have the SSR sequencing data so that I have something to show you today. And this is it. So this is a little bit like the toy version of what we want to do with the rat sequencing data later on. And it's also a little bit preliminary because we did not spend as much time on this as we would have wanted to. So it's probably still some details that are going to change. I hope you can see on the left here the Annika genome, which should be like 19 gray bars, so for 19 chromosomes. So actually this genome was also done within BGE, but in a different part, not in this case study, which is why I'm not going to talk about it. It's about half the size of a human genome, and it's also a diploid plant. And yeah, we just mapped the different loci onto this genome. And as you can see, we were really quite surprised, but happily surprised that these loci are quite well distributed. So it's not two in the same place or anything. So this is actually quite nice, telling us that we might be able to use this SSR sequencing data. Then, yeah, just to show you what would happen if you just naively took the data that was already there and made a PCOA. The different colors here are like the different studies. So you can see that, yeah, this is not really, it's just showing the differences between the studies and not between populations. And then if you take sequence data, yeah, you see it's all much more intermingled and the colors are together. And then also there's a difference if you look at the sequence data, so SSRs with SNPs, or just at the length data to sort of simulate the previous method. At first I was thinking like, yeah, we can extrapolate and somehow have like ambiguity codes for different, same length for different sequence alleles. But when I was looking at this plot, I thought, well, it's probably not going to work. There's just too much difference. But yeah, maybe in the future. So for the next slide, I have to say I've been so happy to be able to show you this because it's a historic moment. This is the first time we have genetic data from all the different samples, from all the different studies. And it's in one map and it's like comparable across the whole. So yeah. So I only made this yesterday, and I think it's super interesting because it shows a little bit something we had seen before with chloroplast genome data. There's a huge difference between the Hesperian Peninsula and Central Europe. And what is super interesting is this part here. So I'm wondering if we will see this divide at the Pyrenees also in other data. So this is one part of the outcomes, marker recommendations, I already said, and the time intervals, of course, as well. And since this is just sort of a subset of the samples, this will probably just be a direction for further data acquisition. Stakeholders and engagement, I will not talk about all of this way too much. Just two examples. So we will be actually part of a museum, which is something I think nobody else has mentioned yet. So there will be a new exhibition at the Botanical Museum in Berlin, and Annika will be a big part of this. So if you want to come, it's probably reopening next year. You're all invited. The other thing is, yeah, just the amount of people that not just we contacted, but that also contacted us. Yeah, in the map over here.
And there's also even Arnica producers who contacted us and said, we are interested in what you're doing and so on. So yeah, at one point I just felt like I had to be a squid to contact and keep up and do everything, but yeah, I didn't manage. So this is a little bit also something we're going to continue and try to keep up this network and the communication. So in terms of policy relevance, yeah, of course, it's a little bit obvious. So we want to showcase with the study how you can do a transnational genomic diversity monitoring, so also over the time. And then also we want to learn something about the management of Arnica populations. And of course, what's something interesting side effect, we are also gathering a lot of new experience with this. So things we haven't done before, for instance, like working with, I think it was 12 countries in the end and getting permits in all the different languages and formats and everything, which in the end just made me think, well, we've managed with driver's licenses, we've managed with health insurance cards, maybe we should have something like a European sample passport, yeah, where people really don't have to think anymore, and also the collectors should think that they hand on the papers, yeah, and it's not just the people who work with the samples who think about the permits. Okay, just quickly, the students, we had 4 students working on the project, 2 are here, the other 2 hopefully online. And this is something that is, yeah, I mean, they were invited, yeah. So this is something that was very important to me, because if we say genomic monitoring, it's something over time, yeah, and it's not just us who need to know how to do it, but also the next generations. And then also the data that we produce, I think will be quite useful for training people to show them like the differences between different data sets, how you can analyze time series data. And of course, we also want to publish things. There's a couple of things that sort of dropped out sideways already, but there's still more to come. Yeah, and with this, I arrived at the last slide. So showing at least the logos of all the collaborators. I did not put the names there because sometimes it's like really several people and didn't want to clutter up the slide, but yeah, thanks to all of them. So of course, we want to finish with our library and then brainstorm with the collaborators what to do next. And yeah, hopefully it will finish in a sort of consolidated Arnica network where we can communicate between people in different countries and have like a European-wide view about the protection of the species. Thank you.
Thank you, Katja. One last question before we go into coffee. How do you farm Arnica?
Ooh, I have no idea if I tried. But it's, yeah, I mean, so you can apparently buy seed and grow them in your garden if they grow there. Yeah. And I've also seen recently that actually one big pharmaceutical company is trying to establish Arnica fields in New Zealand. So yeah, it must be possible. And that's actually something I would like to ask the Arnica producers, but they already said they're not going to divulge their industrial secrets. So maybe not.
Otherwise, a good excuse to go to New Zealand.
Yeah.
So,
true.
Thank you very much to all the speakers, to Katja, everyone that spoke. And now, Jose will tell us just how short our coffee break is.
27 minutes. Okay, let's reconvene back here at 4:50. Okay, we're running a little bit late, but I think everyone deserves a bit of rest. So that's 5:50 online at CET.
Yeah. So when I came here, I was a little bit stressed.
No, I'm still sure.
Hi, everyone. Can you hear me? So we are starting the last session of the day, finally, after such a long day. And we have a session with 3 talks about plants now. So Katie will start online. Let me. Okay. Can we start, Kate? Are you hearing us?
Yep. Can you hear me?
Yeah. All good.
I'm Katie Hearn. I'm a PhD student based at University College Dublin in Ireland. I'm here to present our case study work on Trifolium dubium, aka the shamrock, where we've been looking to explore its genetic diversity across Ireland. Next slide. So Trifolium dubium, or T. dubium, or the lesser trefoil, I guess is its common name, is a species of clover native to Western Eurasia, but also found elsewhere in temperate climates globally as an introduced species. In Ireland, it's the species most widely recognized as being the shamrock, where legend goes that St. Patrick allegedly used its trifoliate leaves to explain the Holy Trinity to pagans. So you can sort of see in the photo there, we've got the Father, the Son, and the Holy Ghost, but obviously that's a myth, and we don't know if St. Patrick even did that. And I doubt he even knew himself what species he was picking up. But regardless, it's the species designated by the government in Ireland as shamrock for commercial purposes. Genomically, it's interesting in that it's an allotetraploid, meaning it forms through the hybridization of 2 diploids. In this case, the diploid clover species T. campestre and T. micranthum, whose genomes sort of persist today in it as 2 separate subgenomes. T. dubium is the 3rd most common clover in Ireland. After the more agriculturally relevant white and red clover, it's common in grasslands, field margins, disturbed soils, and plays a key role in supporting pollinators and improving soil health through nitrogen fixation as a legume. Ireland, like much of the world, has climate change to contend with. Oh, yeah, go forward one.
Shall I move?
Yep, thanks. So yeah, over the past decade alone, there's been over 30% of semi-natural grasslands have been in decline, or been lost or fragmented. Agriculture makes up a major proportion of Ireland's economy, and intensive farming practices are impacting grassland diversity. And then there's, of course, the threat of invasive species and climate change more broadly, which are sort of altering habitats across the world, I guess. Relatively recently, as part of the ERGA pilot project, there was a high quality reference genome was assembled and released for T. dubium. So this is the first time the species has ever been sequenced. And it means that genomics projects and species which wouldn't really have been feasible before now are. The hope is that with population level data supported by this reference genome, species genetic diversity and population structure can be assessed for the first time, in this case in Ireland. Importantly, the species presents a unique opportunity to link the application of biodiversity genomics with cultural identity, and also sort of link into agricultural sustainability more broadly due to the use of other clover species as forage legumes. So T. dubium would be the 2nd allotetraploid reference genome of the genus after white clover, which is sort of the most commonly used, one of the most commonly used forage legumes in agriculture. So T. dubium acts as sort of a useful counterpoint to better understand the implications of polyploidy within the Trifolium genus. So next slide. So the goals of this case study are to conduct the first population genomic study for T. dubium overall, and specifically in this case in Ireland, using whole genome resequencing. We also look to sort of exploit this cultural relevance of the species to more broadly raise the public awareness of biodiversity genomics. Our key questions are: what is the extent and distribution of genetic diversity within and among Irish populations? And how are population structure and connectivity distributed across Ireland? Next slide. So in terms of methods and data, the reference genome, as I said, is from the ERGA pilot project. It's chromosomal scale. It has a high BISCO completeness. Of course, the majority of the BISCOs are present as duplicates. For the sampling, we went to 5 different sites across Ireland, trying to capture the broadest geographic range, in the end collecting a total of 43 different individuals. We did the extractions ourselves, and then next NGS was carried out by Novogene over in the UK. Our analysis pipeline looks a bit like what's shown on the screen here, where it's nothing crazy, sort of initial QC of the reads, read mapping, variant calling, filtering, and then downstream analyses looking at genetic diversity metrics and population structure. In the end, the reads should be publicly available on Ensemble, and then any scripts used we plan to upload onto GitHub. So next slide. Unfortunately, our analysis is actually largely still ongoing. So over the summer, I guess it's good news, but our department's HPC got a hardware upgrade, which sort of disrupted things quite a lot. I'd hoped to have a bit more for you today, but unfortunately not. So the reads have been mapped. We've got high coverage with a mean read depth of around 31x, and over 98% of the reads are mapping the reference. But so yeah, so far so good, but we're getting there. Next slide. In terms of the broader applications of our results, we hope they'll show the usefulness of population studies of widespread, even I guess, weedy species like Trifolium dubium in Ireland, and how that can be useful in national biodiversity monitoring. Our current data set is fairly limited in size. We had hoped to go to more sites and get more samples, but things happened. We had an early winter here, it snowed, and then after it snowed, there was sort of no hope for anything after that. But this is a good sort of pilot study as to see how this kind of project would work. So yeah, we hope this case study will inform future population studies across Ireland, particularly in plants, which isn't really done here at the moment. There isn't really a lot of population genomic studies overall in the country. And we hope that it provides broader visibility to the use of biodiversity genomics in conservation as well. So yeah, next slide. In terms of stakeholders, as an example of 3 actors we've engaged with, the National Parks and Wildlife Service, we engaged with them to inform us on licensing requirements, if any were needed, which in our case there weren't, and on the locations of special and protected areas of conservation, which we purposely avoided then during our sampling. We engaged with landowners and local residents when we were out sampling. We got permission for access to sites from them, and we handed out and provided informational leaflets for them to learn more about the project. We also developed a project website, basically for any of our stakeholders to engage with, and as well through social media we engaged with them. The Botanic Gardens, they carry out their own research and are interested in the phylogeography and population genetics of Irish plants in general, and we consulted them on taxonomic identification and sample preservation methods. Next slide. In terms of policy relevance, our study aligns with objectives in Ireland's 4th National Biodiversity Action Plan, which came out 2 years ago there. We hope that in addition to the genomic data we've made available publicly, we hope to provide them with relevant governmental agencies like the MPWS and MBDC with a summary brief to inform policy in the future. Next slide. So in terms of our scientific outputs, this is the first sort of population level data set for Trifolium dubium anywhere, I guess, not just in Ireland and not just whole genome sequencing. So that's available publicly. And we're hoping to prepare a manuscript for that as well. In terms of outreach and communication, so attended and presented posters at both national and international conferences. I took part in Pint of Science, so that's where you sort of go to a pub and present to sort of the general public, anyone who's around who wants to listen to you talk for about 10 to 15 minutes. So we had about 30 or 40 people come to that, which was good. And we've also established a collaboration with Cell Explorers. So these guys carry out DNA extraction workshops and teach kids about genetics across the country. So we're hoping to do a couple of workshops with them in the coming months. Next slide. In terms of our social media, we were able to keep sustained communication. So we had at least one post every month, either on LinkedIn and X. Our engagement numbers were, I mean, none of us are social media influencers, but I'm pretty happy with the numbers that we got. Over 11,000 views overall, which is quite good. Next slide. And then in terms of our next steps, so obviously finish the analyses and then draft a manuscript for it. We want to conduct our outreach workshops in local schools with cell explorers, as I previously mentioned, and then more broadly disseminate our findings to stakeholders and policymakers as need be. Then, of course, longer term, continue with that engagement with stakeholders and then continue to sort of exploit the species for what it's worth in terms of being a cultural icon or a national symbol in Ireland. And then just acknowledgements. So obviously I'm working on this with my supervisors, Graham Hughes and Anne McCartney. Nina helped with the extractions, and then Ruth and Len helped with the sampling, going about and taxiing me about places. And I think that's all. Thanks.
Thank you, Katie. Does anyone have questions? We have time for one, at least. Do we have any?
Oh. Hi, thanks for joining us, Katie. So I guess Ireland, for most of us, is famously wet. But in your different localities, is there a significant difference in the kind of climatic conditions, in particular rainfall, that you might then be able to look at some of the genomic differences according to the different sampling sites that you visited?
Yeah, so initially we had sort of been like, oh yeah, we really want to look at environmental gradients and such. But yeah, Ireland's pretty homogeneous in that sense. It is wet, wet, wet. There is a slight bias towards being wetter in the west. I don't know if you've ever been out to that part of the country, but the rain goes horizontal. It's crazy. Whereas the east is definitely a lot more sheltered. But yeah, that's something to look into to see if we do see any sort of correlation with that. But it's not the greatest thing to be looking at here. It would be more geographic distribution.
Thank you very much, Katie. Now we'll move on to Victor's presentation. There is another one.
Okay, can you hear me? Yes. So I guess it's like this. Okay, so I'm Vitor C. Souza, not the Vitor Souza. That's there's someone with a very similar name, and I'm from C3C, or Center for Ecology, Evolution, and Environmental Changes, from the University of Lisbon, and I'm very happy to be here to tell you about the case study. And I also want to make a disclaimer that I think it's the first time I'm giving a talk not talking about demographic inference or hybridization or animal species, which is usually what I work. But I guess that's the beauty of belonging to ERGA. And this is because I think we are bringing together the expertise of different people to trying to address a common goal, which is biodiversity genomics and how that impacts conservation in this case, or many other examples that we've seen during these 2 days. And this is to build the reference genome of Corema album that brought together the expertise of Manuela Cinci, who's a taxonomist, an expert in systematics of plants, and also the curator of the Museum of Natural History in Lisbon, with Helena Trindade, who's a molecular biologist and geneticist working with plants, and then Bruno Nevado, an expert on speciation genomics, a PhD student, João Jacinto, working on these species and trying to be able to grow this Corema album in the lab, and Carla Alegria from the Communication Office, and also an expert in plant and crop biology. And so they did most of the work. The only thing I did was to help design the plan and the study and to present here today. So this was a one-year project. And why is this species important and interesting. So it's endemic to the Iberian Peninsula. This is the distribution area. And another very interesting aspect is that it is a dioecious plant. So there's female and male individuals, okay? And they have different flowers. And so this is also interesting from an evolutionary perspective and about the biology of plants. Furthermore, they have edible fruits that have been, so that I used to eat a lot when I was a kid, because I grew up close to the coast. And so it's a sand dune habitat. I forgot to say that. It's very important for sand dunes, and it's a charismatic species in sand dunes. And we, at least some of us, like to eat these edible fruits that seem to have some health benefits. And so there's some, one of the aspects of this study is that maybe we can contribute to grow them and to restoration. So also this is a priority species for conservation of these sand dune ecosystems, and that can be very different. So we can have this more on the cliff. Sorry, where is the pointer? Is it this? Okay, yeah, so these cliffs, or it can be more the typical sand dunes. And so these coast areas are under very strong disturbance because of tourism, of economic activities. And we've seen that already in a talk this afternoon. Because of climate change, many of these areas are also exposed to stronger water stress and drought and aridity. And some preliminary data suggests that the male and female plants might respond differently. So it also would like to use genomics to clarify that. And they are losing their habitats, and in Andalusia, they are already considered vulnerable. So the goals of this case study was to generate a high quality annotation. And this is because as part of the ERGA pilot project, there was already a genome assembly, and I want to thank João Marques because he was the one who built this assembly for this genome, and also Claudio in Florence that generated the data, and the colleagues from Barcelona that generated the high-C. And so we wanted, so we had this genome, but we didn't have an annotation. So this case study was really to look at the annotation. And we wanted to, at the same time, address these biologically relevant questions about the differences between the two sexes, between the male and female plants. And so to do that, Manuela, João, and Carla went to do the sampling in this location, and we sampled many different tissues at dawn, so at early in the morning and late in the afternoon, such that we could have capture different transcripts, also from the roots, stems, leaves, and flowers. And we here try to maximize our diversity in the population. So we used 8 samples from the male that was used to build the reference genome. So we had 8 samples from the roots, stems, leaves, and flowers in the early morning and late afternoon. And then rather than have the same from the same female individual, we collected from different females. All the vouchers are deposited in the museum and at the biobank. And for the genome annotation, we followed the protocol of Aureliano Bombarelli, and this was done by Bruno. And again, this is another advantage of belonging to ERGA, that we can share these protocols and learn together. So these are the slide of results. So the reference genome was quite good. So 13 contiguous chromosomal pseudomolecules with about 500 megabases, and with an N50 of 40.9 megabases and about 96.7 completeness in BUSCO. And these are the results we have for the annotation, which we found about 54,000 genes. But given the high amount of repetitive regions, so we have 51% of the genome that was annotated as repetitive, we are still trying to improve this annotation. So Bruno is still not convinced that this will be our final list. But the bottom line insight is that by using these different tissues and transcripts from different sexes, we hope that we manage to annotate as many genes as possible and to capture the diversity of transcripts of these species. In terms of stakeholders and engagement, so we've been in contact with national agencies of agriculture and veterinary, the INIAV, also the Conservation of Nature Institute, also with local and regional municipalities, with two NGOs, SPECO and ResEco. So SPECO is about the Portuguese Society of Ecology and the Portuguese Network of Ecological Restoration. And also even with companies of agriculture, because there's this interest of trying to grow these plants to commercialize the fruits. In terms of policy relevance, our main aim is to support future conservation genomics of these sand dune ecosystems and use this as a kind of emblematic species that could raise awareness and help showcase the importance of genomics to conserve these species. So that's why we are trying to develop communication material explaining this in Portuguese and to interact with as many stakeholders and entities as possible. And this is where we enter into the impact and communication, where we've been participating in workshops with stakeholders. We've participated in poster and national meetings of fruit producers, and we've tried to do these talks very often in Portuguese and to write so in the local language, so to write and translate this knowledge into something simple that was more accessible, including, for instance, an article in a monthly trade magazine tailored to farmers. We had monthly posts in social media, and we also participated in the Open Days activities. Also, this was very nice that there's this photo book written in Portuguese describing and summarizing lots of the research that's been done with the white crowberry, covering from seeds to flowers to the fruits to the genomics, and it was very nice that we participated in this photo book. The ERGA blog post also raised awareness about this project within the University of Lisbon, at least, and it was important. And so the next steps, as I said, Bruno is now confirming the gene annotation, especially trying to solve this issue with the repetitive regions. We are making all data available by tomorrow, at least the preliminary data with the help of Chiara, hopefully. And then we want to, in the next months, do this sex-specific genetic gene expression analysis to see if we find differences between males and females in different tissues. And in the midterm, we'd like to publish and disseminate these results. And we are planning to apply for funding to do the population genomics and the interesting, cool stuff that we can do now with this important resource. And I want to thank again the people of the team that actually, they did all the work. It's just that they couldn't come, unfortunately, today to be here in person. Thank you.
Do we have any questions?
There's one question in the back, so.
It's just a short question, and I don't know if you know, but is it actually possible to see this plant if we go to the beach here, or is it only somewhere
I'm
else?
not from here, but it's within the distribution area, so I think you should be able to see it if there's some sand dunes
Get the flashlights
around.
tonight. Thanks.
I think maybe in Vila do Conde is more rocky, right? Yeah. So there you wouldn't see it. Yeah.
Yeah, thanks. Very nice talk. I was wondering, because you mentioned that there are also some companies which are maybe thinking about commercializing the plants. Are they interested in these kind of resources?
Yes. So for instance, João, part of his PhD, he's trying to germinate the seeds under controlled conditions in the lab and to find what are the right conditions to grow the seeds. And the success rate is very, very low. And we are now trying to see if we can develop some markers for candidate genes that then we can try to identify if there's some genetic variation or, you know, if there's a genetic basis to understand when they grow better. So yes, so there's some interest from on these results. Yeah.
So I know that you're still confirming the annotation, but a question we get time and time again is like, how many tissues do I have to sample? How many lifestyle stages do I have to sample, etc.? And you have a really nice range from both sexes and lots of different tissues. So I wonder if I could suggest or request if you could also do like how many additional genes do you find when you add a new tissue?
I see.
Just as a proof of principle here,
yes,
we have few of those, and I would like to see more of those. And this seems like a really good
yes,
case.
That's a very good idea, and for sure we have the data to test that, so we can see. Of course, so here we didn't have seeds because we thought that it wouldn't have enough material, and so we didn't have all the life cycle, but yes, but at least with these 8 tissues at different times, we can see how much do we gain in the annotation. Yes, very good idea.
We have
Thanks.
an online question. Will be amazing. Oh, sorry. Is there phenotypic variability within the species that could be important for local adaptation or food production?
So it would be very nice if these other people were here to answer that question. And I hope I'm not saying anything wrong, but from if I recall the discussions, yes, there's some evidence for differences. So at least in terms of the resistance and tolerance to aridity, there seems to be differences between males and females. I just don't know if that's local and different in different regions of Iberian Peninsula, but I think I recall that there's differences between the south, where it's very dry, and here, this region, which is much wetter. And so I would suspect so, but I cannot answer for sure.
Thank you very much again. And now we have another online talk. Dennis, can you hear us?
Yes, yeah, I can. Can you hear me?
Yeah, all good.
Okay, so, and I see you can see, you can see the screen. Great. So, hi everyone. Thanks first to the organizers for organizing the conference and giving me the chance to talk. So I'm Janice, and I'm a postdoc in Aarhus University. I'm going to talk about work on local adaptation in Thymus vulgaris. So I think probably everyone knows common thyme. So it's a very common shrub in the Mediterranean, and it's also widely used by us for both as a condiment for food, and it also has many uses in traditional medicine. But what is also probably not as known is that its aromatic compounds that we use are also used for the plants for climate adaptation. So there is variability between individual plants in which is the predominant monoterpene that they produce. There are 6 main and quite common chemotypes, which can be divided into 2 main ecotypes, depending on the chemical composition of its predominant compound. So we have on the one hand the non-phenolic ecotypes, which have been seen to be more resistant to freezing events, while the phenolic ecotypes are more resistant to heat stress and heat wave. So these 2 ecotypes vary, we can find local variation at a very local scale. So the place where they have been very studied is in Saint-Martin-de-Londres, that is in southern France near Montpellier, where there is, in a very small geographical scale, in around 10 square kilometers, we can find variation in the predominant monoterpene on local on the predominant ecotype in local sites, where in the central regions that tend to be more cold and have more freezing, we find that the non-phenolic ecotype are dominating the landscape, while in the surrounding regions that are more warm, because there is actually, it's a valley with a temperature inversion, we tend to find the phenolic ecotypes. What also has been seen is that in the last decades, as freezing events has been decreasing in the frequency and the intensity, the phenolic ecotypes have been becoming more common and expanding its range. So it's actually a case where so far there has been a successful response to climate change, where using the ecotypes are able to track the increase in the heat wave frequency, while the freezing events have been decreasing. It has also been shown that the genetic regulation of the ecotype is oligogenic, so it can be almost around 90% of the variation in this phenotype can be explained by three loci that intervene that regulate the ecotype. So, but there are some questions that we wanted to track in this study. One of them is whether this importance of the ecotype variation for climate adaptation is unique to San Martin de Londres, or is it something that we can find in other populations? And also whether the ecotype is the main contributor to local adaptation, or there are other phenotype and genetic variation that are also important. And finally, something for which the genome has been very important, it's to understand what are the genetic variants, so whether there are, as we will see, there are actually structural variants, and whether these variants are shared across the populations or they are different variants that are regulating the ecotypes. So for this, we generated a new data set where we sample multiple altitudinal transects for different populations. We sample between 3 and 10 sites that show diversity in the phenotype, the frequency of the ecotype within each of the populations, where not always, but most often we tend to find non-phenolic ecotypes in the higher parts of the mountain that tend to be colder. From this, we generated a data set. So in the population resequencing data set, we used a gene enrichment capture of around 400 genes with a total of 1.7 megabase per sample. And so from this, we got around 1,000, exactly 1,000 samples for which we have got the ecotype information and genetic sequencing. Then we assembled the genome that using the supported by BG. That so far, because of lab issues, we still have not been able to produce the high-C data for scaffolding, so we have a draft genome, but we are in the process of finally getting the chromosome level genome. But the draft genome is quite contiguous and has some issues with duplication in the BUSCO, because it's a very repetitive genome, but hopefully it will be improved as we add the high-C data. But overall, it's a quite complete and contiguous genome that has been enough for the analysis. And we also generated for some of the site temperature recording data, so some environmental data but for this I will not show results because it was too long to have all. But yeah, so getting into the results. First, if we look at the population structure, we can see that even they are quite geographically closely, especially the ones in the middle. Populations tend to cluster, samples tend to cluster by population, but in that mixture plot and in PCA. And very important, the two ecotypes that in the PCA are distinguished by either it's a cross or a round circle. They tend to cluster together, so at the genome-wide level, the ecotypes are indistinguishable. So it is at the local level, at the local genomic level, where we will be able to find the difference between the ecotypes. So to map these regions into the genome, and this genome is at the chromosome level, but because we used external closely related chromosome levels so far to place the scaffolds while we don't have the high-C. So we map the three loci in this genome. We find three very clear peaks showing association with the ecotype. And within all of these loci, we find genes that are known to be involved in the biosynthetic pathways of these different monoterpenes. So it's what we will expect to be regulating the ecotypes. Next, we look at what it is, what are these variants. So to do so, we had both the two draft reference genome, the one generated with the ERGA VGA, that is GIR5, and then a previously generated also draft genome for another sample, and they are both from the same populations as the study. So what we see, these are dot plots in the local genomic regions where we find the two main hits, and we see that the GIR5 genome have the same alleles, so they have they don't have structural variation, but within all the other, when we compare all the other haplotypes, we find always some complex structural variation. So in the main hit, we can see that the primary assembly of the lin 607, there seems to be a deletion relative to the other samples. While in all the others, we have some other sort of more complex variation, but it's clearly some structural variation. So we can see that there's a structural variation that also seems to be multi-allelic. So we have at least 3 different variants in using only these 4 genomes. 4 haplotypes from 2 genomes. Next, we wanted to explore whether the importance of these variants for local adaptation across the different transects. So we first did an FST scan within each of the 10 transects, and then we integrated these 10 FST scans using Pigmin, that is a software to test for evidence of repeated adaptation. And we do find, so in the plot I have put boxes in the background to indicate the regions where we have the associations with the ecotype. And we see that the main hit determining the ecotype is also has the strongest evidence for local adaptation, but all 3 hits as captures have been involved in repeated local adaptations across transects. We also find 3 other variants, 3 other loci that also have evidence to be involved in local adaptation. So the ecotype is not the only mechanism for local adaptation to climate, which also makes sense, but it has a very high importance. Then I also look at the local clustering of all the samples in the 2 main hits for adaptation to the ecotype. And in the trees, I have colored the terminal branch by the ecotype. So blue if they are the non-phenolic and red phenolic, while the dot in the end of the branch is indicating the population of origin. So what we can see is that within these 2 loci, the ecotype, the samples cluster very strongly by ecotype, indicating that they all populations share the same genetic variation in the regulation or determination of the ecotype. So the 2 main results that can be relevant for conservation management is first that the ecotype is important for local adaptation. So it first provides an relatively easy to measure phenotype to track the adaptive potential of populations. And it is also important to preserve as much chemotype diversity as we can in order to preserve the adaptive potential. And we see that the genetic variants are shared across populations, suggesting that it is dispersion and gene flow that allows populations to have maintain the adaptive potential. So it's also important as much as possible to maintain habitat connectivity. So the outreach that we have done, we have been presented in local and international scientific conference, and also disseminating some of the main results or like the main points in the across social media and the IRCA blog post. Also, two of our collaborators from Montpellier are involved in also in local conservation of time and in general of the different natural parks. So they are in contact with stakeholders, and once, as now that we start to have quite clear results, they can start using it for applying in exploring relevance for conservation policy. So one clear case for locally, it has clear relevance in understanding the importance of the ecotype for local adaptation across all the populations. One that is more speculative, but that we think might be relevant evidence, is in making plans of how to use both combining phenotype and genetic data to track the adaptive potential of populations and predict the potential for adaptation to climate change. But that this is quite speculative since it's just one example, but that we think combined with other examples can be interesting. So, and the plans for the communication, we are making, of course, available all the genomes and the sequencing data. And also this will produce georeference data on local ecotype abundance at this point that can also be useful in the future to track how the populations will and ecotypes will maybe shift as a response to the continuing climate change. So we are preparing the manuscript on local adaptation, and for the future we are also planning to use the reference genome to try to understand better the origin of these genetic variants, and also potential sharing, because there is some preliminary evidence that they might be shared across other species of the Thymus genus and even more further species. And yes, public outreach, once we have some publications, we'll use press releases to disseminate the results. And also, again, our collaborators are in contact with local communities that are, for example, it's quite common for traditionally to harvest thyme for cooking or for medicinal purposes. So they are also interested because the different chemotypes also have different both smells and taste and medicinal properties. And yeah, so the next steps is to finish the scaffolding the genome and publish the first manuscript. And as we've finished the results, we can start communicating and discussing what to do with the stakeholders and how this can be helpful. In a more longer time scale, our collaborators are also working in tracking to track or predict the changes in ecotype distribution as a response to climate change, and that we can also use genetic data for that. And also there's the follow-up research on the origin and the sharing of the genetic variation with other species. And yeah, so this is a project that has involved quite a lot of people. So I thank everyone and also thank BGE for the help for generating the genome and for all the help in organizing everything. And thanks for listening.
Thank you, Janice, for the presentation. Do we have any questions?
Thank you, Janice. I was wondering, do you plan to develop some kind of test based on the genetic data that you have, or is it sort of good enough to use the olfactory method to distinguish the ecotypes in the field?
Yes, so olfactory you can distinguish if you are trained. Like for me, I tried, and between the six, I'm not always successful. But so for this, we have for actually phenotyping the samples, we used like chemical profiling, like no, the name doesn't come, but Yeah, we use like we look at the chemical profile to confirm that what's the abundance of the different compounds. And with the genetic data, you can distinguish, but not with 100% accuracy. So usually you can, it's around 90% accuracy. So it's not, there are either other variants or probably there's a small plasticity. So it's very strongly genetically determined, but not totally.
Anyone else? We're good. Thank you again.
Thanks.
And we conclude it for today.
Yes. Thank you. So just can you hear me? Yes. So thank you all. I would like to ask for a round of applause to everyone that spoke yesterday and today. Not only the case study presentation, but also all the excellent and amazing keynote addresses that we had. And so go ahead, don't be shy. It was a long day, but I think it was, and thank you all for the enthusiasm of the presenters and the audience for keeping active and because this is I think the last time in a conference that we are all gathered here in this room, because tomorrow we'll have parallel sessions. I would like just to acknowledge, well, João Pimenta here, come here. Yeah. Yeah. Well, he organized this, so I think thank you, João, for all the effort, and of course, Christian, who's been instrumental. All the people from ERGA and BGE, Chiara, Diego, Tom, Rita, and Luisa, that were very important for organizing this and moderating the sessions. Thank you. A round of applause. And actually, these guys are very important driving the committees of ERGA, and so their work has been very, very important over the last years. And also, finally, the people from here, from my group, that I didn't name in the beginning, but Joana, Liliana, Inês, João. So we have three, and Pedro, who have also helped a lot during this organization, and thank you for all your help. Okay. I hope I didn't forget any name of people. Okay. So just final logistic remarks. So for people that are online, don't forget that tomorrow this won't be streamed in YouTube. You have Zoom links to access the sessions in the program, right, Luisa? Yeah. And for the people that are here, so today we will have our dinner at 8 in Hotel Santana. Don't forget to bring your badge, and if you don't remember what you chose as a meal, it's written on the back of your badge. Okay, so the bus should be outside, yeah, waiting for us. We're a little bit late. And so have a nice rest of afternoon, and we will see each other at 8. Thank you.